Hello. Good morning, everyone. Welcome to the second day of the 9th Annual Conference of Board of Genetic Counseling India. We are delighted to welcome you all. This session, we are going to focus on the genetic diagnosis, counseling, and gene therapy. I am Upasana Mukherjee. I am a Level 2 Certified Senior Genetic Counselor. And with me, I have Ms. Ashwarya Panampil. She's a Level 1 Certified Genetic Counselor. And we both are working respectively in Bangalore and Kolkata. So, these areas which we are going to talk, that is genetic diagnosis, counseling, and gene therapy, are at the forefront of personalized medicine, transforming how we approach the genetic disorders. Genetic diagnosis allows for early and precise identification of the genetic conditions, setting the stage for the targeted treatments. Genetic counseling offers the vital support and information, helping individuals and families to understand and manage their genetic risks, while gene therapy aims to correct the faulty genes causing the disease and hoping for many. Today, our expert speakers will delve into these topics, addressing the challenges, ethical considerations, and future advancements, and how they can collectively enhance the patient care. I would like to first introduce and call upon our chairperson, Dr. Usha Dhave. Dr. Usha is a very known and dear ma'am to all of us, and I must not specify her credentials, but she is a PG research grad at Mumbai University for PhD, honorable medical geneticist in Vadia Maternity Hospital, Mumbai. She is also research director for Mills International India, Mumbai, national professor and visiting scientist for Hafkind Institute, Mumbai, and the laboratory director for Navigen Labs, Mumbai. Her career highlights are she is the president for ISPAT from 2021 to 2023, vice president for NPI, which is from 2018 to 2022, founder member for ISIEM, that is the Indian Society for Inborn Error of Metabolism. And many awards and recognitions has been always enlightened her career, special awards, recognitions from invited guest speaker for international and national scientific meetings. And her research interest lies on the clinical and biochemical genetics, cytogenetics, molecular diagnosis, and translation genetics, and so on. We welcome you, ma'am.
Good morning, everybody. It is my honor to introduce our co-chair, Dr. Abhishek Mohanty. He is the program director and pan-India head for the Center of Biorepository and Biobanking at HCG Cancer Hospital, Bangalore. He is also the chairperson of HCG Scientific Research Committee. He is the regional director, South India, for the Biobanking India Foundation. He has also held his previous positions as the Principal Research Officer and Head of Research, Rajiv Gandhi Cancer Hospital and Research Institute, Delhi. He is also the founder, Head of Molecular Oncology Division, MVR Cancer Hospital and Research Institute, Calicut. Speaking of his academics, he has procured his doctorate in biochemistry at IISc Bangalore and has his postdoctoral fellowships from Ottawa Heart Institute, Canada, and Institute of Stem Cell and Regenerative Medicine. Speaking of his additional qualifications, he has more than 15 years of experience in teaching research in molecular and translational oncology. He has more than 30 publications in peer-reviewed international journals and has authored a book chapter in Springer Nature. He holds one Indian and one US patent as well. He is the editorial board a member of international journals like Frontiers in Immunology and Public Health, to speak of a few. Speaking of his career highlights, he has led various research and biobanking institutes across India and has contributed significantly to scientific research and publications in the field of oncology. We welcome you, sir.
Now, I would like to request Dr. Usha Dave ma'am to please introduce our first speaker, Dr. Professor Parimal Das.
Good morning, everybody. Nice to see so many people right in the morning, 9 o'clock. Our first speaker here is Professor Parimal Das, and I believe he is unable to come and it's online. Am I right?
Yes.
Okay. Nevertheless, you are reading here also. He is a senior professor, Center for Genetic Disorders and Institute of Science, BHU Varanasi. He is BSc from North Bengal and later on PhD from BHU. His additional qualifications are postdoc research on recombinant DNA-based malaria vaccine development in NIM, NI, Institute of Immunology National. Also PCP NDT elected member, he's member secretary of Human Ethics Committee in BHU, and also Dr. Mohan Malviya Cancer Research Center, Netrudaya ICT Hospital, Varanasi. His career highlights are his leading research on genomics of polycystic kidney disease, renal cell cancer, congenital missing tooth, Parkinson's disease, ALS, autism spectrum disorders, and so on and so forth. His co-PI in UMID, that is, I think all of you should know, these are DBT-initiated programs, and so must be Center for the Genetic Diseases in Newborn Babies supported by DBT. Actively integrating human genomic knowledge for practicing integrative personalized medicine and incorporating aspects also of Ayurveda and holistic medicine. So we welcome Professor Parimal Das, and I think the team will be having his online talk. Dr. Parimal?
Please play the recorded talk that I have sent. Well, I will be present on.
Can you hear us? We cannot hear you. Are you mute?
No, I can hear you.
Yeah, now you can.
Well, sure. As I have mentioned that I will be remain online while the recorded play will be there, which I have already sent last night, so that you can save time, and I will be ready to take the question.
Yes, sir. So we are just starting with the recorded version, sir. Thank you.
This work has already been published in novel patent-induced putative kinase 1. Good morning, everyone. Thanks, Dr. Ali Hassan and other organizers for giving me this opportunity to present our work on genetic regulation from NGS, expediting the scope for personalized medicine in neuro-related conditions. Neurological disorders is an umbrella term for a wide range of diseases, injuries that primarily affect the nervous system, including the brain, spinal cord, and peripheral nerves. It can ultimately lead to a number of neurogenetic and neurodevelopmental disorders, including Alzheimer's, Parkinson, amyotrophic lateral sclerosis, epilepsy, and various neurodevelopmental disorders like autism spectrum disorder, cerebral palsy, etc. There are no treatment capable of modifying the course of neurological disorders. The identification of effective drug depends on the understanding of the pathophysiological mechanism of underlying disease. So, the important step in these directions depends on the availability of genetic screening that can identify genetic predisposition, enable early intervention, and proactively preventive strategies provides targets for gene therapies. Discoveries in vitro and in vivo models have added greater understanding into it. Animal models have enabled the field to develop taste and refine target therapies. Genetic forms of human disorder do not always perfectly phenocopy sporadic diseases, but in many cases, there are excellent surrogates providing intrinsic validity and genetically based model in neurodegenerative disorders. In fact, the models based on genetic forms of these disorders have provided both insight into the molecular mechanism and temporality of changes of human diseases and help on identify candidate potentiality disease-modifying therapies. Our lab is currently focused on understanding the genetic basis of various neurological disorders like Parkinson, amyotrophic lateral sclerosis, and autism spectrum disorder using Indian patient and families. Developing drug-induced animal models of Parkinson's disease, finding different gene expression and in treated and non-treated conditions, and to explore the effect of Ayurvedic formulation in disease conditions. Also, want to aim at the establishment of patient-specific cell line with a specific disease gene mutation, which has been revealed by sequencing in our lab. Parkinson's disease, which shows an overview of the Parkinson's disease, as we all know that the Parkinson's disease, the pathological hallmark are the loss of dipostasial dopaminergic neuron and in the presence of intraneuronal proteinaceous cytoplasmic inclusions terms Lewy bodies. It's a hallmark for Parkinson's disease. This pattern shows the pathways of gene action which ultimately leads to the Parkinson's disease, shows the interaction of different candidate genes like PINK1, DJ1, and ultimately the complex interaction of different candidate genes in the mitochondrial membrane. A oxidative stress is get involved and finally leads to Parkinson's disease. And it shows the experimental setup of the workflow. Human blood is the subject for these things. We have so far been able to collect 289 sporadic cases of Parkinson's disease and 2 familial cases. We were clinically confirmed by our clinical collaborator. Then blood sample collections, screening of the known and unknown variant, data analysis in silico and in vitro analysis, and visual correlation of the scene. It's a list of variation which has been found in the candidate gene parking. And the shown here is the amino acid change and the nature of the change like a non-synonymous condition, and applying various prediction tool, it has found to be disease causing. Same slide in the variation in PINK1, we also find nonsense and the non-synonymous changes, which also leading to the disease causing condition. List of one of the major candidate gene, leucine repeat kinase 2 gene, we also say it's synonymous disease causing and then synonymous disease causing mutation. And in vitro also there were PIN1 variant. This curve is showing the change is from the normal to the mutant condition is shift from C799C to T. And its conditions find across the species that it is in a very conserved region of this PG. What we find ultimately, due to this change in the DNA, the blueprint of life cause ultimately amino acid change following a primary, secondary, and tertiary structure of the protein, which gives a 55 kDa mutant protein, whereas the normal one is 100 kDa. This work has already been published in novel mutant-induced putative kinase 1. Good morning, everyone.
Professor, can you hear us? Professor Parimal, can you hear us? Hello? Sir, can you hear us? Yeah. So there is a technical issue with the video which you have, which we have received. Could you kindly continue from the slide which was seen last? Hello, sir. You are in mute. Can you unmute yourself? So could you kindly unmute yourself? You are on mute.
I can hear you.
Yes.
Yeah, I can hear you.
Sure. So could you kindly start presenting from the last slide which was seen in the screen?
Okay, now
I will tell you the slide number, sir. 1 second.
hello. Yeah. Yeah. It's on here. Can you hear me?
Yes, sir, we can.
Yeah.
Sir, the slide. Yeah, pink one variant ke baad, after that, sir.
Yeah, but I have to bring the slides into my things, so that's something to manage.
Okay. So. Okay. So could you kindly show it? Is it coming, sir?
So the problem is I have to bring my slides into
Okay.
No, I don't understand that. But how do I bring that slides with me now?
Hello? Dr. Parimal, you can share your slides here, and wherever you have left from there, can you start it?
Yeah, I'm trying.
Share screen.
So go to a little bit here, and then share screen is coming to the present now.
Yes, I think you can forward those slides. We saw your first slide and you can talk parallelly.
Yeah, I'm trying to do that.
So we are really sorry. I will get back to you in some time. Before that, we are proceeding with our next speaker, sir, and we'll just sort the technical issue, sir. Please be with us.
I think so, because I'm not being able to bring the slide and present more from my skills at the same time. So, yeah.
So we will get back. I'm trying. I'm connecting with you, sir. And I would like Usha ma'am to introduce our next speaker.
The technical issue of this presentation. I think without wasting time, we will move on to the next speaker. Our next speaker is Dr. Anand Kumar. He is a consultant pediatric hematology, oncology, mainly, and the bone marrow transplantation specialist at Sparsh Hospital, Bangalore. His academics is the MBBS 2005, then King's Bangalore, and later on MD in 710. BJ Medical College, Ahmedabad. Oh, I see. Additional qualification, he is hematology training, Gangaram Hospital, New Delhi. So, of course, he will be authoritative on his subjects. His career highlights are consultant hematology MD/MT at BGS Hospital, Fortis Hospital, Bangalore. We welcome you, Dr. Anand, and look forward to hear something new.
Very good morning to all of you. So I would like to thank the organizers for giving me this opportunity to interact with the genetic community as such. So we as clinicians, we communicate most of the time with the geneticist or the genetic counselor over the phone. Face-to-face interaction is quite less. So here I present a few of the experience from the hematology side, wherein we have relied on genetics to lead us to the diagnosis, and how it has helped us in managing the patient as such. So next slide. So, like we all know, hematopoietic disorders are always driven by a lot of genetic mutations, and the genetic technologies like NGS, the ultra-deep PCRs, whole genome exome sequencing, are helping us to detect several mutations that are involved in the pathogenesis of these disorders. And there is emerging evidence that these data can be used for the clinical practice, including diagnosis, prognostication, and to predict the response of specific treatments, and also guiding us in the novel treatment targets for the patients with hematological disorders. So, like I said, genetics has a key role to help these patients right from the diagnosis, sometimes risk stratification in our leukemia patients, and of course, like I spoke about, the targeted treatment approaches that we have, and ultimately to decide whether the patient is responding or not, and to help in prognostication for these patients. So, so I would like to start my thing with starting the root of hematology. So we all know that all these the blood cells that are produced in our bones in a soft spongy structure known as the bone marrow, it is like a blood factory for us. So in this, there is an engine known as the hematopoietic stem cell, from which various cell lines are produced. We are all aware of this. So today, I will take you to each of the cell line, and I will present a case scenario wherein we have used genetics to either diagnosis or arrive at the risk stratification or prognostication treatment approaches. So it will be a case-based approach kind of a thing that I'll be doing. So first, let's go to the red blood cell disorders. So the most common single gene disorder in the world is thalassemia. It has a huge burden, about 240 million of the world's population of thalassemia carriers. Every year, about 1 lakh thalassemics are born, and 80% thalassemics are born in Asia and Middle Eastern countries. So in India, around every year, about 10,000 children with thalassemia are born. So, like I said, the carrier rate in India varies from 1 to 17%, average being 3.3%, and 40 million thalassemic carriers are present. It is a sixth of the world's, 1/6 of the world's thalassemics, and nearly like 10,000 thalassemics are born every year. So it's a huge burden, and only 10 to 15% receive appropriate treatment for this disorder. So to briefly tell about the thalassemia types. So earlier we used to classify them as thalassemia major, intermediate, or trait. Now, with the help of the genetics, we are able to classify them into transfusion-dependent thalassemias and non-transfusion-dependent thalassemias. And this has kind of clearly demarcated the treatment approach for us. And this is a brief, this is a slide which briefly tells about the pathophysiology of thalassemia. So as we all know, there is an excess of, so there's a deficiency of beta chains. So the alpha chains are excess, that creates the imbalance, and there is a hemolysis that's happening. As a consequence, there are a lot of things like jaundice, gallstones, hepatosplenomegaly can happen. And to overcome this, we are giving a lot of blood transfusions, so iron overload. Ultimately, this leads to cardiac failure, so organ involvement, and finally death. So this is what happens in thalassemics, right? So, like coming to the genotypes, like about 200 mutations have been reported worldwide, and the gene is the HBV gene on chromosome 11 is responsible. And in India, these are common mutations, and some newer mutations have been discovered. So let's take up a case scenario to help us understand how genetics play a role in these patients. So in one of the clinics, one of the antenatal mother came for an antenatal visit, and she had beta thalassemia trait, right? So MCV was less and hemoglobin A2 was 4%. But so what we do, we check for the husband's thing, whether he has a thalassemia trait or not. Fortunately, the husband was having a normal blood counts with a normal MCV and a normal HbA2 levels. So mother is a trait, father, the husband is normal. So don't need to, because the would-be child would be fine, right? So that's what the clinician thought, and no further tests were done. Normal delivery happened, healthy baby boy was born. So, but what happened later, 4 months later, is baby had progressive failure. The hemoglobin dropped to 5, and when they did the hemoglobin electrophoresis, it was suggestive of thalassemia major. So HbF was more than 90%. So again, it improved with transfusion, but now but the family is really disturbed now. Like they're saying, so the obstetrician reassured them, saying that there is nothing to worry, and now we have a baby child who is having thalassemia major. So how can we explain this, right? So when we did a mutation analysis, the mother was turned to be, of course, thalassemia minor, but father had a silent mutation, cap site mutation, which was not detected because the complete blood count was normal, no microcytosis, HB electrophoresis was normal. So fortunately, and because of the consequence of both of them having this thalassemia mutations, the 1 in 4 chances that the child could develop thalassemia major, and unluckily here the child developed a thalassemia major. So beta thalassemia carriers, silent beta thalassemia carriers is something that we miss in our day-to-day practices, and that's when the genetics do come to play important role. So like I said, clinically everything is normal, and of course it can be diagnosed with some of the alpha beta globin chain synthesis ratio. That is quite, we can do it, but ultimately we rely on mutation analysis to arrive at the proper diagnosis, so that we can prognosticate, saying that look, they have a thalassemia trait mutation. So there is a 1 in 4 chance that the child that could be born will be having a thalassemia major. So let's go to another case scenario. We had a 23-year-old male who presented with anemia, abdominal pain, and jaundice. He had received a lot of transfusions from the age of 12. So he had hepatosplenomegaly. Hemoglobin was low, MCV was low. So we did a HB electrophoresis wherein we saw a high level of HbA2. So how to interpret this HB electrophoresis given the clinical context? So usually we rely on this thing that if the HbA2 levels are more than this thing, it could be HbE syndrome, HB Laffour, and so on. But again, we are not clear that what kind of a thing, particular type of hemoglobinopathy this patient has. So here again, molecular diagnosis comes to our rescue. We had this compound heterozygous for IVS15 and HBE. So the diagnosis is HBE beta thalassemia, and they can have varied phenotypes, and mostly they are managed as thalassemia intermediates. So these are some of the scenarios wherein genetics really plays an important role in thalassemia management. Of course, diagnosing in early life, like I said, in early life, the shift has not happened from HbF to adult hemoglobin. So the interpretation of HbF levels when are very early, in 1 or 2 months of age, is difficult. So at that newborn life, mutations could be helpful. And then, of course, in very difficult, unusual features, and especially this scenario we face quite often, the patient comes with severe anemia, someone in the district place has given already blood transfusion. So now how would you diagnose those things? And of course, in thalassemia intermediates, and of course, like I said, in prognostication, some of them, thalassemia intermediates having XMN1 polymorphism, they respond very well to hydroxyurea. So doing the genetic mutations in such patients would help us in guiding the treatment and prognostication as such. Like I said, prenatal diagnosis and to detect silent carrier mutations, and of course, other hemoglobinopathies like HBD Punjab I spoke about. And of course, nowadays, we in our practice, we recommend it to all new diagnosed thalassemia patients for accurate genetic counseling and helping in their understand the disease and further deciding about the treatment course and everything. Okay, so let's take up another case. There was this 39-year-old lady who was referred to us in view of low hemoglobin requiring blood transfusion on and off. She is a known case of thalassemia trait. So as we all know, thalassemia traits, minors, they live a near normal life. They don't require any blood transfusion. But the question here is why this lady is requiring hemoglobin transfusion, requiring a lot of, so there are number of reasons why middle-aged, a young adult lady would require blood transfusion. So we ruled out all other causes, and then we did a genetic test because we were suspecting something was alpha triplication in these patients. And this is one of the conditions wherein thalassemia trait behaves as a thalassemia intermedia requiring blood transfusion. So this helps us reassuring that the diagnosis that, okay, you don't have any major problems, you don't have any bone marrow problems, it is your own thalassemia trait with this variation causing your current problem, and you just need some blood transfusion requirements here and off, and there's nothing to worry. So this is one of the scenarios again wherein in thalassemia the genetic diagnosis help us to arrive at the diagnosis and properly guide the management. So this is the same genetic report that for this patient. So thalassemia mutation carrier state is present, and of course alpha triplication is present here. So let's go to another case. So this is a 19-year-old. So this is the red blood cell disorders again. So we finished the thalassemia part. So let's go to another red cell disorder wherein we took the help of genetics to arrive at the diagnosis. A 19-year-old girl presented to us with recurrent anemia requiring blood transfusions. She also had insulin-dependent diabetes mellitus and dilated cardiomyopathy. So she was evaluated in multiple places, but the cause was not identified. So there was no any fever, jaundice, blood loss, bony pains, recurrent infections, no significant family history. So there was some history of second-degree consanguineous parents with her, and on examination, failure was present, and there was a hepatomegaly, and other systems were normal. So here, whenever there is a anemia case, we do this workup of blood counts, iron profile, and everything. So you see the low hemoglobin is there. MCV is low, again indicating a microcytic hypocrite picture, wherein you think there is iron is a problem. So we did a ferritin, which is very high. But again, the other causes of anemia like autoimmune hemolytic anemia, thalassemias, hereditary spherocytosis, PNH, even bone marrow was done. But in the bone marrow, if you see, there is an increased iron stores. So two things in this slide. One is the ferritin is high, MCV is low, ferritin is high, and the iron stores are high. So what do we do next? So we did a fasting iron profile. If you see that serum transferrin levels are low, 20, okay, but all the other parameters are increased. So there is only one condition wherein you have a high ferritin, high iron, low transferrin, and a microsodium picture. So we suspected congenital A transferrinemia in this patient, causing refractory iron deficiency anemia. So hence genetic testing was done, and she had this homozygous transferrin gene mutation on exon 12. So now we know that this patient has this thing. So the beauty of having this genetic diagnosis, now we know that this patient doesn't require iron therapy, doesn't require blood transfusion therapy. It just requires plasma transfusion, wherein we can supplement the transferrin, and whenever the hemoglobin is high, we have to go phlebotomies to cut down the iron overload. So this case really helped us how genetic has played a role in diagnosing this patient, so that she was really struggling with a lot of diagnostic dilemma till she came. She was requiring a lot of blood transfusion. People were transfusing her left and right, giving iron because MCU was low and everything. So this is how genetics helped us in arriving at the diagnosis in this case. So this is the same genetic mutation panel that we got. Transferrin mutation was present. So now we are done with the red cell disorder line. Let's go to the white blood cell disorder that has helped us in arriving at the diagnosis. So this is a very young girl who presented to us at a 5-year age, born out of a 3rd-degree consanguineous for the consanguineous parents. So she was normal till 3 years of age, when she developed fever on and off monthly and subsided with antibiotics, not requiring any oral or parental antibiotic as such. But she had recurrent aphthous ulcers in the mouth, and around 4 years of age, she had some rashes in the infancy, mostly responding to moisturizers. So the first serious infection that she had was in 2017, February 2017, when she had this suppurative cervical lymphadenitis, which showed staph infection, which is recovered with 7 days of IV antibiotic. So second serious infection occurred in May 2017 in the form of necrotizing gingivitis, requiring 7 days of antibiotics, from which she recovered. So on both these occasions, when the blood count was done, her neutrophils were very low. So she was supported with granulocyte colony stimulating factor. So here, this is a case of severe neutropenia. So we are trying to figure out what is the cause. Is it a congenital nature, or to rule out any other cause of neutropenia in such a young child? So other causes are like cyclical neutropenia, benign familial neutropenias, and of course, we did a genetic mutation panel that is available for congenital panel, and we did a workup for inherited bone marrow failure syndromes. So it was negative. When we looked at the autoimmune profile, we looked at the primary deficiency profile, even we did a bone marrow aspiration biopsy to look at if there is any myeloid arrest. There was no any drug history, and it is unlikely to be infectious for such long process. So therein, we were stuck with this severe congenital neutropenia, and no known mutations were detected. So like as a clinician, so what should I do in this case? So I know that this patient has most probably severe congenital neutropenia, but I don't have a genetic diagnosis to proceed. So I cannot confirmly say that, okay, this patient is having congenital neutropenia and requires bone marrow transplant as a curative option. So if you see the literature in congenital neutropenia, the indication for bone marrow transplant would be one or more than one life-threatening infections. Until then, you have to just support them with granulocyte colony stimulating factor. So that's what we did. But we side by side, we kept on counseling that this patient would require bone marrow transplant, and we started working up for that. So meanwhile, we took the help of Christopher Klein from Denmark, from Germany, wherein they were able to diagnose this ADA2 deficiency. So it's again a primary immunodeficiency which can explain this neutropenia in these patients. But unfortunately, in these patients, the timelines got jumbled up. We were waiting for this report. Meanwhile, the patient was in the community, succumbed to a serious infection. So this is one of the cases wherein although we sought genetic diagnosis earliest, and whatever tests that are available in India, we couldn't arrive at the diagnosis. We couldn't properly guide the treatment, and we were just stuck with the conservative management. But this child definitely required bone marrow transplant. Maybe the availability of this whole exome sequencing would have helped this patient earlier and would have guided to the patient to the transplant, and maybe this child would have lived a long life. So this is again a case scenario wherein, so primary deficiency as such, the genetics plays a huge role. There is a wide spectrum of inborn error of immunity which are diagnosed through genetics, and it has really revolutionized that part of specialty. So let's go to another. So I spoke about the red blood cell part, white blood cell part, and next let's take up the platelet disorders. So this was a 9-year-old girl from one of the districts in Karnataka. She was referred for recurrent epistaxis, So this symptom was onset from 7 years of age, and there was no any trauma associated. Always there are third degree consanguineous parents, and any family member did not have any similar history. So the initial screening test, whenever a child comes with bleeding problem, we do is what is a platelet count, that is normal. We do an initial coagulation screen, that is PTINR, APDT, that is normal. And so after this, then we suspect, okay, this patient now may be having a platelet function disorder or a von Willebrand disease kind of thing. So we wanted to do a complete bleeding analysis. So we did a complete bleeding analysis. In that, so if you see this this thing, it's a platelet aggregometry studies. Usually, we suspected platelet function disorders. So in Glanzmann's thrombocythemia, usually ristocetin is not affected, but all others should be affected. But this is again not affected. So it's a kind of a variant of Glanzmann's thrombocythemia that we were suspecting, and hence we got a genetic diagnosis. So here we have ITGA2V mutation, giving us the diagnosis of Glanzmann's thrombocythemia, which couldn't be diagnosed through the blood test that we are having, that is the platelet aggregometry. So therein again underlying the importance of genetic diagnosis in diagnosing this case of bleeding disorder, right? So these are some of the just a few case snippets. In everyday aspect we are dealing with such disorders, and we are using genetic tests to arrive at the diagnosis. So let's take up another case. 24-year-old trainee in the last trimester was referred with respect for.
Dr. Anand, you have 2 minutes. Okay. Can you wind up early, please?
Yeah, sure. So this lady had a problem with low hemoglobin, low platelet, and she had also had seizures, but was found to have a DVT, thrombosis of the lower limb also. So it's a very rare scenario wherein we have thrombocytopenia with thrombosis. So we did an initial workup. These were the few differentials. So, but simple peripheral smear gave us the clue that there are these cystocytes. It could be a case of TTP, right? Congenital, so thrombotic thrombocytopenic purpura, right? So we did this workup. We have this ADAMTS profile here. But here what you see is, of course, the ADAMTS activity is low. But usually in ADAMTS acquired TTP, the antibody levels are high. So here the antibody levels are low. So wherein it led to a suspicion that it could be a congenital TTP. So therein again we did a genetic workup suggested to have a congenital TTP. So this is another case that we had a pediatric child similarly treated for isolated thrombocytopenia as ITP for several years, and then when he came to us he had renal involvement, high blood pressure, and we again suspected congenital TTP, and ADAMTS mutation was positive. So these patients were diagnosed with Upshaw-Schulman syndrome, that is a congenital TTP. So simple treatment is just supplement the ADAMTS with FFP transfusion, it will improve, and these children did remarkably well and on follow-up. So like I said, we are also dealing with hematological malignancies, the role of genetics in hematology, in diagnosis, risk stratification, and management. So we had this old lady with a very high white cell count and this peripheral smear picture. So when we did a FISH test, it turned out to be BCR-ABL positive. So she's a case of Philadelphia positive CML. And so this BCR-ABL being positive led us to the diagnosis of CML. And since we have a BCR-ABL positive, this is targeted by tyrosine kinase inhibitors. So this is the case again wherein genetics has helped us in both in diagnosis and treatment, the targeted therapies. So again, we had this middle-aged lady from one of the districts in Karnataka, came with very high white cell count. The bone marrow shows promyelocytes. So this is a case of acute promyelocytic leukemia, wherein the mutation that is present is PML rara. So again, this is the only kind of blood cancer wherein you don't require chemotherapy to treat. You can just treat with vitamin A, that is this thing, atra and arsenic, right? So survival rates are close to 90% in this kind of scenario. So this was this lady, and of course we can go on and on this thing. Just 2 minutes, just 2 minutes. So one of those cases we diagnosed with AML. So this patient, completed his chemotherapy, was doing well, attained remission, but I just want to underline the importance of genetic diagnosis here again. We got a ASXL1 mutation that came positive here. So this patient was risk stratified to higher risk, and even though the patient was in remission, had to undergo transplant to achieve complete, further prolonged remissions. So this is again a very interesting case. I think to the lack of time, I'll wind up my talk. Thank you.
Thank you so much, Dr. Anand. I think in the end of the session, we'll have questions. I hand out my mic to the co-chair, Dr. Mohanty.
Yeah, good morning all. And so we go ahead with the next speaker for this session. Next speaker for this session is Dr. Sunil Shakya. He is a scientist too in the Department of Genetics and Genomics at the Rajendra Institute of Medical Sciences in Ranchi, Jharkhand. And his academic credentials are, he's a bachelor's in biology from Bikaner University, Rajasthan, a master's in biotechnology from TM College of Biosciences, University of Bikaner, Rajasthan, and a PhD in neurology with specialization in neurogenetics and genomics at All India Institute of Medical Sciences, New Delhi. His career highlights include being a scientist C in an ICMR project at All India Institute of Medical Sciences, Delhi. And his work mostly revolved around doing the molecular genetics of thyroid cancers for a period of 3 years, 8 months. And he's been a senior research fellow in the ICMR project at AIIMS, Delhi, wherein he has looked at the molecular genetics of spinocerebellar ataxia. So his research specialization focuses on movement disorders, molecular genetics of spinocerebellar ataxias. Over to you, Dr. Sunil.
Good morning, everyone. First of all, thank you for providing me an opportunity to interact with you, the wonderful genetic counselors. So here we'll start. The title of the talk is A Dilemma in Genetic Diagnosis, Prognosis and Counseling for Rare Neurological Disorders. So I think almost everything has been covered if we talk about the mutations in last 2 days. So here the area which is least covered in this conference is that the expansion mutation in some of the patients. So I will try to cover these types of mutation. So first of all, the rare neurological disorders. Actually, as 2 days back, Dr. Vinod Sakare said that there's nothing like rare in the genetic disorders. But if you talk about the rare neurological disorders, these are actually the phenotypically and genotypically they're rare because most of the cases remains undiagnosed genetically. So rare neurological disorders, these are extremely heterogeneous group of disorder, clinically as well as genetically. They affect the brain, spinal cord, peripheral nervous system, and the muscles. Actually, they represent almost half of all the rare diseases that are present in here. So, and out of these 80%, more than 50% of the cases remains undiagnosed, even in specialized center where broad genetic testing is routinely performed. So, genetic underpinning of rare neurological diseases. So, genetic defects are the researchers are continuously working on the rare neurological disorders, and they continuously uncover the new genetic defects that helps to understand the disease. But the number is very high. That is why we are not able to, make some profile that will help us to, deconvolute the disease. There are some genetic modifier like other than the main the causal mutation. There are some secondary mutations are there that actually modify the disease phenotype that makes more difficult to the clinician to make the clinical diagnosis. Other than the secondary mutation or secondary variants, there are some other factors like environmental factors. They contribute to the disease phenotype. The factor that also affect to the disease phenotype is the mental condition of the patient that that also contribute to the genetic modifiers. So other genotype-phenotype correlation is also very important to solve these types of cases. So the rare neurological disorder diagnostic challenges and unmet needs. The diagnostic gaps are there because after we have the next generation sequencing or other advanced technology, there is the diagnostic gaps. So we have to, a large proportion remains undiagnosed even after the advantages in the genetic testing. So there are some technology, there are many technology that helps to diagnose genetically, but there are some gaps like the profiling of large repeat expansion mutation that cannot be done using the conventional method like the Sanger sequencing or some even in the with the NGS. There is a new technology that called long read sequencing that only helps to explore some novel types of repeat expansion mutation patients. So the carrier screening is also a challenge because we do not know even the primary mutation. Then how can we do the carrier screening? So first of all, we have to have the reports of the primary mutation. Then only we can go for the carrier screening in the neurological disorders. So rare neurological disorder may be broadly divided into three parts basically: the neuromuscular disease, neurometabolic disorders, and neurodegenerative disorder. As I'm working on the neurodegenerative disorder, basically the spinocerebellar ataxia, I will talk on this only. So the spinocerebellar ataxia, these are clinically and genetically heterogeneous group of neurodegenerative disorder. These are a common disorder in a rare neurological entity. So these are basically the common symptom of the spinocerebellar ataxia is the ataxia. The ataxia may be the gait ataxia, or maybe other ataxia means incoordination basically. It may be the gait ataxia, the limb incoordination, any in the upper hand or upper limb or lower limb, maybe the dysarthria or other symptoms. The ataxia is one common thing that is present in all the spinocerebellar ataxia. There are approximately 100 types of SCA types are there in the world. And autosomal dominant cerebral ataxia may be divided into 4 broad categories. These are the ADCA 1, 2, 3, and 4. And in India, SCA 1, 2, 3, and 12 are the most common ataxia subtypes. So, so we do screening only for these types of ataxia in the primary screening. In the secondary screening, we include other types of less common ataxia like 6, 8, 17, and DRPLA. So if we talk about the inheritance of spinocerebellar ataxia, there are inheritance of autosomal dominant, autosomal recessive, sporadic, or maybe the X-linked inheritance. The mutation types are basically the tandem repeat expansion mutations in SCA1, 2, 3, and 12. And there are also the conventional mutation like in SCA28. And there are some tandem repeat mutation in autosomal recessive ataxia also, that is called the Friedreich's ataxia. So the mutation present in almost throughout the genome, that the exonic part, intronic part, 5' UTR, 3' UTR, as well as in the promoter region. So what happens at the time of screening, what we do in the clinic? So first, when the patient comes to the OPD, clinician makes the clinical diagnosis on the basis of the disease symptom, family history, biochemical testing, and the radiological findings. Then after after making the clinical diagnosis, he refers the patient for the genetic screening to us. And we take the informed consent, collect the blood, and extract the DNA, quantify, then we check the quality of the DNA. After the quality check, we do the gene scan. For the primary screening, we first of all screen the patient for SCA1, 2, 3, and 12. And if they got negative, then we further consider this patient for 6, 8, 17 DRPLA. For recessive ataxia, we do screen for Friedreich's ataxia. You can see most of the cases remains undiagnosed after the genetic diagnosis. We further divide this patient into 3 categories on the basis of their inheritance, that is autosomal dominant, recessive, and sporadic. We use the panel for the autosomal dominant as well as for the autosomal recessive. And the sporadic cases are considered less than 5 years, is less than 25 years. We consider these patients for the recessive screening, and if age at onset is more than 25 years, then we consider this patient for the autosomal dominant screening, basically for the repeat expansion mutation. So I would like to discuss some cases that will probably help to do the genetic counseling. So there are some interesting cases that we experience in the clinic. So here a 35-year-old male came to our clinic with the symptom of progressive cerebral ataxia. He was having muscle stiffness, rigidity, and sensory neuropathy. He was diagnosed, clinically diagnosis was SC1 or 3. So after making the genetic diagnosis, we found that this patient is positive for the SCA3. He is having heterozygous expansion mutation in ATXN3 genes. Actually, the story begins here when his younger brother came to us and said that I want to know about my son, that is there any probability that he is also having these types of symptoms, because all the siblings of these patients were affected with the same disorder. The clinical symptoms were almost same. That is why he refused to go for the genetic test because of the financial, the constraints. We did the genetic counseling on the basis of his brother's report, and we counseled him that we counsel like the prognosis for the SCA3, like SCA3, and inheritance, how will he be inherited the disease. But luckily we considered this patient for the panel-based sequencing, or panel-based the diagnosis. We considered this patient for SCA1, 2, 3, and SCA12 screening, and we found that this patient is positive for both SCA2 and 3. He is having heterozygous mutation in both SCA2 and 3 genes. So here, the counseling entirely changed actually. So we cannot do the counseling for both the patients simultaneously, because both are having the different mutation, although the both patients are siblings, but the counseling will be entirely different. The probability of getting mutation in the next generation will be changed from 50% to 75%. The similar case, see, in this case you can see the family, the mother and father's side both are affected with the same disorder. Here the same, almost the same family was there. So onset was 22 years and having the cerebellar atrophy, dysarthria. The patient was having SCA2 symptoms, and the clinical diagnosis was SCA2 and 3. And when we diagnosed this patient, when we did the molecular screening, this patient was positive for SCA2. He was having heterozygous mutation in the ATXN2 genes. You can see the threshold of the disease is 32 repeats, and he was having 39 repeats. We considered this patient for the panel-based sequencing and found that this patient is having homozygous mutation in the ATXN2 gene. So his sibling was having heterozygous mutation, and the patient, the youngest brother is having the homozygous mutation. So the genetic counseling will be entirely changed here also, because the getting mutation in the next generation is actually the 100% in second case. So here the third case, this was also an interesting case. 29 years old male came with us to with the feature of progressive ataxia, dysarthria, abnormal finger nose test, impaired tandem walk, decreased reflexes, and slow saccades. The symptoms was typically SC2. As this patient was this comes from the Agrawal community, we first time saw the patient with Agrawal community with these features. So we decided to go for the panel-based screening, and found that this patient is positive for SCA2 and 12. See, the SCA12 is exclusively present in Agrawal community, not in Agrawal communities. That is why we did the panel-based the screening for this patient. So we have to consider the community also, like as in some of the community, like in Jat community, the SCA7 is the more prevalent. I mean, almost 99% cases comes from the Jat community of North India. Another case is the 22 years onset, having gait ataxia, tumor, dysarthria, and hyperplexia, was having slow saccades. The family history was autosomal dominant, was showing the autosomal dominant inheritance, and clinical diagnosis was SC2. And he was diagnosed with SA1 and 2. The meaning is that that may, and the feature of clinical feature doesn't matter in the spinocerebellar ataxia. There may be the concurrence of the ataxia. So the panel-based the screening is most important. Another interesting case was there. The family history was negative. Age at onset was 32 years, slowly progressive ataxia, and he was marked as SC query. So the symptoms were like for SC17 seizure and the forgetfulness, early sign of dementia are present in the SC17. So we did the secondary screening and found that the patient is having the expansion in the SC17 gene. The cutoff of SC17 is 47 repeats, but we found that this patient is having 42 number of repeats. See, the patient is showing typically SC17 phenotype. That is why we consider this repeat as a positive repeats, because these are the intermediate repeats. Sometimes these patients, show the phenotype, but these same repeats are present in the control population also, in healthy population also. So we have to consider such things also in the genetic counseling. So there are multiple reports are there in the co-occurrence of the mutations. So if you talk about the prognosis of the disease in the single trinucleotide repeat expansion disorder, the number of disease severity increases with the number of repeats, and this is also dependent upon the subtypes of the ataxia. But what in the 2-in-1 mutation in the co-occurrence? So you can see here that all the ataxia genes are interacting with each other. So we can't say if there are two mutations in a single patient. We can't say we can't talk about the prognosis. We can't say anything about prognosis. That may be the more severe or more lighter symptoms. So in the genetic counseling, basically in the autosomal dominant disorder, if one is having the single expansion mutation disorder, their progeny will have 50% chance to get affected. If one is having 2 mutation, the progeny will have 75% chance of getting mutation. If there is homozygous mutation, then the 100% progeny will get affected with the disease. So the co-occurrence of the causal mutation must be considered during the generating genetic reports and the genetic counseling. So genetic counseling should be done carefully in the patient carrying the homozygous mutation in basically in the autosomal dominant diseases. Any combination of ataxia subtype may manifest with or without clinical features. You have to take care of that. Panel-based screening of SCA loci in the affected families with SCAs is recommended. A detailed family history should be taken during the clinical examination, and all affected individuals must be screened for the panel-based genetic analysis. In such cases, prognosis, disease inheritance option always vary, so genetic counseling must be done accordingly. Other contributing factors like environmental factor, other mental condition or financial condition, or other secondary variants should also be taken into the consideration during the genetic counseling. So long-term regular follow-up will definitely help us to take the better decision in the future. Thank you so much.
Thank you, Dr. Sunil, for highlighting us on the importance of genetic screening in differential as well as primary diagnosis of neuromuscular disorders. Thank you. So we move on to our next speaker today. Our next speaker is Dr. Rohit Karyappa. He is the lab director at Neogen Labs, and his doctorate is in the field of biochemistry and molecular biology from University of can't read this, University of Florida, I think, yeah, University of Florida from US, and he is also a board certified clinical chemist, and current position is, of course, is the lab director, and his previous positions were at Medtronic, Minneapolis, US, Johns Hopkins Medical Institution, Baltimore, US, Washington University at St. Louis, US, and Heart Research Institute, Sydney, Australia. Over to you, Dr. Rohit.
Thank you, Dr. Abhishek. Good morning, everybody. This is a talk on NBS, newborn screening. We set up our lab in 2006 in Bangalore, and we've been doing this for 18 years now. It's still, the uptake is very minimal. Awareness is low. So hopefully when you leave here today, have a good awareness about newborn screening, what the lab does, the important, so the awareness, the setup of a lab, the management of the lab, and the very last part, my last slide, might be interesting from the genetic counselor point of view. What happens when newborn screening, the baby definitely is sick. The newborn screening is normal. So the newborn screening looks at treatable disorders. And my last slide says, what happens when the newborn screening is normal? So for the past maybe 10 years, I've been telling them, the parents, or the doctor who calls me and says, your report is normal, that this is a small subset of treatable disorders that we're treating. So definitely do genetic analysis of the baby's DNA. And I think in the future, if newborn screening becomes like more universal, we'll have a good pickup, and that's great for the population. You get treatment for those babies. But at the end of it, 99% are going to be genetic, untreatable, and they'll come to the counselor. And that's when, I think my last slide refers to that. What happens when the newborn screening report is normal? As a newborn screening lab director, I'm telling them very strongly, please do genetic analysis. And it's great news that a lot of doctors are very receptive nowadays. They take the responsibility to counsel the parent or send them to genetic counselors to do that piece. And as you've already seen, there are many, many reports of rare mutations being found, which is it's evidence-based medicine. Now the family knows there's something in their family, but this is a bit of a distraction. Sorry, move on. So the importance of newborn screening is that it's a set of treatable disorders, that's the whole point of this, that are captured early in the baby's life, soon after birth. And inborn errors of metabolism, that's what they are. And not all of them, just a small subset. Totally about 60, 70 disorders that we pick up with our testing, which I'll just go through briefly. And when you miss it, if you don't do screening, the outcome for those babies with those disorders, even though it's treatable, is terrible. So therefore, it's a powerful test. If you catch it early, very beneficial outcome, there's treatments, therapies, etc. Not all disease disorders are treatable, and therefore, they're not in the newborn screening panel. What's in the newborn screening panel are the treatable but undiagnosed disorders. So the history, briefly, is the Guthrie test for PKU. Dr. Robert Guthrie should have won the Nobel Prize for that. He didn't. Today we realize the importance of that discovery. So it started for one test for one disorder. And then tandem mass spectrometry, which I'll talk about in the last part of my talk, very powerful technology. From one spot of blood, you're able to diagnose about 40 treatable disorders. So that's being used quite a lot, and it's re-energized the field of newborn screening. New technologies are coming out, multiplexed assays. From one blood spot, 3 or 4 biochemical tests can be done. Microfluidics, moving fluids with electric current. Those are being used for lysosomal storage disorders. Hard to treat, but can be diagnosed early. ACMG stands for American Council of Medical Genetics, has a panel formulated that all babies should, it's recommended, please screen for these disorders in the newborn period. They're all treatable. And a refinement of that is the recommended universal screening panel. So we do that in our lab, almost all those disorders that are in the RUSP panel for newborn screening. So this is just for awareness. I'm going to get to setup and management of the lab. But when should a newborn screen be done? So 24 hours after birth. That's really soon, but you should wait for this because there's an endocrine surge that all of the doctors know happens, which settles very quickly. About 24 hours, the TSH values come down to normal. You allow metabolism to settle after the birth trauma. And basically, it's about 3 to 6 feeds in that time period. So you've challenged the metabolism, the enzymes in the baby's digestive system, or et cetera, metabolism. And it's a very good time to collect the sample. So please remember, after 24 hours. And then up till when? Well, the baby's asymptomatic usually, So maybe the first week of life is a good time. Up to the neonatal period, up to about 28 days, it's beneficial to do the test. If you catch something, you can save that baby's life. The types of samples, dried blood spot, that's it, only the dried blood spot. Collected on filter paper, an excellent sample. I'll get to that on the next slide. Urine, very unreliable, not for screening at all. Do not use urine. Cord blood, for 1 or 2 disorders only. Venous blood depends on the contamination. Ideally, it's the heel prick from the heel, the baby's heel, collected on the filter paper. So this is just some worldwide. This is from the CDC Atlanta. Newborn screening is the most successful public health program. There's no second place. Maybe polio, but that's probably the #1 most successful program. More than 75 countries do it, and it's mandatory. So every birth, it's happening by law. It's like a baby's birthright to do a newborn screen. In fact, in Singapore, etc., you have to opt out. If the parent does not want to do the newborn screen, they have to sign a declaration. We are not doing the newborn screen. We'll take responsibility. So it's automatic default opt-in. Every baby gets screened. Let's look at our situation in India. Approximately 27 million births. There are newborn screening programs for a long time in Chandigarh, in the district of Chandigarh. The government does it. In Goa, there's a private-public partnership, private and the Goa government. There's an MCGM program in Mumbai that was run in the municipal limits, 4 hospitals. Again, that was public-private. And then Kerala government is the first big state other than Goa. In 4 cities, they're running newborn screening. But there are issues with that program. There are tendering issues, etc. So it stops and starts. I don't think it's started again for some time. The end number that I calculated was about 0.01% of that number undergoes screening. So the huge market, it's just awareness. It's mostly the corporates and a few enlightened cities or states that are screening at the moment. So for setup, I've just put things that when we set up the lab, what do we think about? It's not easy. You have to think it through, and it's not at all easy to make any money in it. We're probably one of the only labs that's run by a very intelligent man, my business partner, Thomas Mookin, father of a child with an IEM, and he thinks through the business side and the testing side with the high quality. So it's very well run, and that's credit to my business partner. So personnel is very important. People, you have to be passionate, be trained well, you have to understand the importance. It's not the doctor, it's not the parent, it's the baby. We're collecting money from a newborn baby. We have to give you a good result, or as good as possible. High quality filter paper to collect the sample, not any old paper. Has to come from a well-made source with barcodes. Location of your lab, a central place, so couriers, patients don't come, it's couriered sample, the dried blood spot. This one kills all sorts of programs. If you're not aware of it, annual maintenance contracts. So equipment's expensive to maintain, easy to buy. So Bangalore City decided to buy 2 fancy GSP, genetic GSP machines. I don't know what SP stands for. They're from Perkin-Elmer. The 2 white elephants. They bought it, collected the kickback, and the machines are not working. So like 5 years ago, big announcements were made. I think the annual maintenance contracts, if you don't pay for that, your quality just falls off. So that's a big cost that people don't look at. Accreditation. Is your lab following established protocols? Is it, running in the right manner? Are rules being followed? Is there quality? Have to have accreditation for a newborn screening lab. Because it's a one-time test, you have to get it right with the screening to get good quality screening. Kit supply, crazy. So many suppliers. Logistics, are they working? Do they not work? Continuous supply. You can't tell your client, we're not giving you this result because we don't have kits. And then other reagents that are not with the kit. They're all expensive. And there's huge customs duties, etc. But, it's unfortunate But those savings and those costs can't be passed on to the customer. Labs have to pay that. Equipment is voluminous. Fluorescence readers are very sensitive, better than the spectroscopy readers, spectrometry readers. They look at fluorescence, which is makes it a much more sensitive and specific test. Washers, automatic washers, shakers, incubators, evaporators, auto samplers, tandem mass spectrometers in the more advanced labs, nitrogen generators, HPLC for hemoglobinopathies, dried specimen punchers. If you have large numbers of samples, it's tedious and difficult to punch by hand. So automated machines do that. And you have to have a very good laboratory information management system. That's really important. Human beings make errors. You have a high thyroid value, the report goes out as normal, not with a good LIMS. The LIMS catches it. So you have to invest in a good LIMS and keep refining it. So you need an IT professional also. So the lab setup, temperature control lab, these are sensitive tests, so you can't go above certain temperature in different seasons. Bangalore's lucky, it's quite temperate, but still it gets hot during summer. Ancillary equipment to run the lab, people don't think about this. You need generators, you need UPS, compressors, pure water, safety equipment, and again, safety, annual maintenance contracts for all that equipment. So it adds up the costs. Okay, so that's in the setup side. It's a short talk, so that's it for the setup. Management, this is my goal as a laboratory director. What do I think about every day? Reduce false positives, unnecessary anxiety. If I tell the doctor the baby's positive for the screen, please do the confirmatory test, and that's negative, that's really irritating for the parents, for the doctor. So you try to keep false positives as low as possible. On the same hand, you still want to have a false positive bias. You'd rather call a baby maybe positive and ask for further screening, a repeat specimen, or confirmatory testing, rather than a false negative. And that's, oops, sorry. Oops, I'm going the wrong way. Yeah, you want to eliminate the false negative, and that happens rarely, and that's part of screening. You can't get rid of the false negative for various factors. So all our reports have a big disclaimer saying, you know, positive may be negative. And when it happens, you take it very seriously. You do a root cause analysis, you do a CAPA, corrective and preventive action, in your quality control, you be transparent about it with the doctor, and you proceed. Turnaround time is really important. For labs, you can't take time. If the baby's truly positive, you can't be taking 10 and 12 days to give a result. It has to be within 24 to 48 hours from the time it reaches the lab. Did I do that? Oh. Power? Oops. Came back to generator. Oh, it's come back, yeah. Yeah, so that's my bottom line. To deliver a result that a doctor can trust. If you don't trust it, call me, because then we reach a consensus. Usually if I get a positive, I'm calling you to say, does it fit the clinical picture or not? Sometimes I'm telling you it's clearly positive and the baby is asymptomatic, so that's a wake-up call to the doctor. So the technologies that are used are the biochemical assays. These are ELISA or immuno-based assays. I'll talk about those disorders. Tandem mass spectrometry, which looks at a bunch of analytes in the blood. And then HPLC is used for the hemoglobinopathies. I'll just talk briefly about the biochemical assays. That's the automated instrument, and it works. You put the samples in for all those disorders that I'll show you, for about 6, 7 disorders in plates. In the morning you come and all the results are out. The software decodes it and tells you what's positive and what's negative. And that's about 1 or 2 crores for that machine, but the annual maintenance contract for that is 20 lakhs. So it costs 2 crores, but to get the technicians to come every year and service it is 20 lakhs. So it's really a government that should be doing this test and not private labs. Biochemical assay screening, these are the classic disorders that all doctors know, and these are very easily treatable. Congenital hypothyroidism, a model disorder. If you have it, the IQ, and you don't screen it, don't diagnose it, every week the baby's IQ drops 10 points. At the end of a month or 2 months, you have a developmentally delayed baby for the rest of its life. Very unfortunate. Treatment is 15 paisa, and the test is about 250 rupees. So beggars can afford to treat their babies, but it's not being done universally. That's very sad. The rest, you've heard of them: CAH, galactosemia, cystic fibrosis, rare in India, but there's a kit, so we do it. G6PD, very common. The other extreme, extremely common in India. Have some incidence data on the next slide. Biotinidase, easy to treat, but rare. And PKU, easy to treat, but rare again. These are done biochemically on that machine I showed you.
Dr. Rohit, you have 2 minutes.
Oh, 2 minutes, wow. I've got lots to go. So this is, again, just the biochemical assays. Stay away from false negatives as far as possible. Some incidence data, multiply the last digit by 100, you get 46 per 10,000. That's how I read it. TSH, 46 per 10,000. CAH, 22 per 10,000. All the way down to PKU, 1 per 10,000. 5 years data, random 5 years. Hemoglobinopathies, this is what the instrument looks like. Excellent. You pick up trait and sickle cell, all the other variants, structural variants of hemoglobin, the tandem mass spectrometer, that's what that looks like. It's super sensitive. It's very high sensitivity and specificity for a bunch of disorders: organic acids, fatty acids, amino acids. Extremely robust. High throughput, very low false negative, false positive rates. So NICU babies, go here first, whatever it costs, 3,000-4,000 rupees. Do a TMS on that baby for sick babies. This is what I deal with every day. If I'm stuck, I call the treating physician. Between him and me, we can figure out what to do next for the baby. If I have these false up or down analytes. I mentioned these. These are examples of each class of disorders. 50 more, so about 12 there, 15 here, 15 or 18 here. If you get a positive, screen first, that's with us. Confirm it next. Before you tag the baby, go to the next level, which is either GCMS with the urine specimen, HPLC, amino acid, again, urine or plasma, or DNA for certain disorders. In the newborn screen, for like CFTR gene for cystic fibrosis. Go to a lab with NABL, make sure it's accredited to get good results. A team in the lab, scientists, some sales guys. That's my talk. That's my address, if you ever want to contact me. And then the final slide, 2 types of panels. A simple panel, cheap, about 1,000 to 1,500 rupees, basic panel. If you include TMS, it goes up to like 3,000 or 4,000 rupees. And that's definitely for NICU babies. What happens when this is normal? That's where I was going with my last slide. Pretty happy I got here. If you're normal for TMS, HB, GCMS, urine and blood panel for a newborn screen, but you continue to suspect an IEM, that's when you do this, which has relevance to this talk. So thank you very much. Yeah. It'll get there at some point in the future when the costs drop, I think. But then you have too many VOUS. What you're doing with the blood, you actually have quantitation of the analyte. You have excess citrullinemia, tells you like that. And you have all the other amino acids normal. That's ASA, lyase, ASA synthetase for sure. So the blood and the basic panel that we do with TMS or biochemicals is a quick, direct, cheap cost. That's why when the costs, maybe 10 years from now, when we drop, go to that, and you have better knowledge of all the pathological mutations, maybe it'll get there.
Okay. We can have the questions at the end. We will have a panel discussion. We can have it. Okay, thank you, Dr. Rohit, for highlighting us on the on the NBS screening platforms or initiatives taken on Neogen Labs. Thank you. So I think we can resume the next talk, that is Dr. Parimal's talk. Can have him online again. You can have him.
Hello, Professor Parimal, can you hear us? Hello, sir, can you hear us?
Can you hear me?
Yes, sir. So go ahead with your slides, sir. We are ready. I hope you can see the slides on the...
Right.
Yeah. Yes, sir.
Okay. So that's why that I get lost with the slides, and when we see what we found from there, can you make it a full screen or something? Is it possible to make it a little bigger? Yes, that's probably I can it's ready. Okay, so as I said that our work has been published in that you have seen here that instead of a wild type 100 kDa protein, we got a truncated product of 55 kDa, and you can see the lab data showing the beta actin in that. Next slide. So, two other mutations are written over there that's also found pathogenic in that. We have already been working on that, and the functional studies on that is on, undergoing. This has already been published in IGMI. Next. Here, as typically when we work on here, even if you respond from the deep sequencing of next generation, or nowadays it's called even third generation sequencing, it's always better to validate the data with a conventional technique like standard dioxy sequencing, and that's what is happening over here. We have cross-checked it, cross-validated by forward and the reverse sequencing, and see it has been correct. Next. Now, following this as the way of the day is coming with a prediction by using biometrically and in silico prediction of these things. So that's what it's showing the wild type and the mutation. Next. And, couple of things that we are now doing it using this information tools to see the primary, secondary, and tertiary structure. Next. This is again the continuation of the same thing to show the mutant protein that how it takes a tertiary structure, and that's why it's become, difficult for us to do the correct job. Next. So in that we have shown the ISND value and the noise fluctuation value to see the tragic end that it is a finding from the mutant protein. Next. So, in brief, we found that a nucleotide substitution in exon 8 of the ring domain of part 2. This substitution, obviously, as I said, consecutively was difficulty in having the original functionality structures of the protein. And this ultimately leads the pathophysiology of the Parkinson's disease. Next. Now, as we all know that now there is Indian traditional medicines long been said, but there was some lag in between the experimental data, but they're widely shouted over here and throughout the world. So we wanted to see the impact of this kind of Ayurvedic product, Ayurvedic formulation is called Rasachandru with drug-induced Parkinson's disease model. Next. It has been explained over here, so I'm not going for that. There is a time constant. We made a MPTP induced, which is a false narcotic Parkinson's disease model, mouse model. Next. There are different kinds of models being used. A few of them are shown over there, and recently it is coming the 3D model or the organoid model. Apart from this is genetic model and then drug-induced model for Parkinson's disease. Next. The same kind of things is shown that how genetic model is different from the neurotoxins model and what is its advantage or disadvantage. Both have its disadvantage and disadvantages as well, but both are very effectively used to study the impact of a mutation which is found from your population in a specific mutation of a specific candidate genes. Next. This shows that MPTP action is showing the production of the superoxide radical is very much found in Parkinson's disease, which inactivations of the tyrosine hydroxylase enzyme. And it causes the DNA damage and depletion of this cellular energy states. And that's how it works in the nerve cells. Next. So Rasachandru and Ayurvedic organomaterial derivatives of the mercury. And you know that mercury has a strong effect on releasing these things in the body physiology, and it acts for several neuroprotection kind of things. So mercury has a high impact in Ayurveda. And that's why we wanted to check it now how it's really work. And earlier, the Rasachandru has already been seen in a study in our from BHU Bangalore University that it has a protection against ALS and Alzheimer's disease. Next. The study that we have planned in experimental setup was with 3 cells of mice. Those are 8 to 10 weeks old, and essentially those all are male because the occurrence of Parkinson's disease has been found more in the male. And the condition was a empty vehicle, and the condition 2 was the experimentals with MPTP, 15 mg per kg of the body weight, twice a day for 2 hours, and it was given intravenously. The condition 3, the questions of Rasachandru, so all are like in experimental 2, plus the Rasachandru 1 gram per kg of Rasachandru per body weight for the 4 days. And mice were all sacrificed 2 days after the last injection. Next. And this is the workflow that how it has been done, which logical sections have been done from the brain with a specific precision, and then immunostudies were done to see. Next. This is a histological study of neurological design to find out the dopaminergic neuron in the section. Next. This shows the microscopic observations in case of condition 1, 2, and then condition 3 in 2 different magnification, 4x and 10x. And pretty clearly you will be able to see here this condition 1, which is empty vehicle, you find all the neurons over there. But when we treat it with MPTP, the neuron has died, and again it has been restored when you giving that Rasachandru. This clearly indicates that Rasachandru has a immunoprotection B. This is having been cross-validated. Next slide. By counting the neurons in the dopaminergic specifically, what we found over there from the web, this is the MP control, this is with MPTP, and this is MPTP per Rasachandru. So average cells count from there that implemented experimentally for each condition was compared, and this result significantly recovers the dopamine state neuron in condition 3, which means presence of dosa syndrome. Next slide. This again, as to see it, the real data in terms of its RNA by using semi-quantitative RT-PCR. Next. And it's fun using the specific use of our affirmatics. We have done the experiment twice, as we very typically done in the research lab, and it shows the overexpression, low expressions of several genes which are connected with the neural development. So in first case, it is MPTP versus saline, and then in second one, it is MPTP plus Rasa-Sindhu, and to see its effect in terms of this MPTP versus condition 2. Next slide. The list of differentially expressed gene identified by the microarray, which clearly shows that pexin B3 stimulates neural outgrowth and interacts with the ring and a neuron specific and small place which is involved in the
Can you hear us? You have 2 minutes to wind up session. Yes, please.
Okay. We'll go first. Next slide, please.
You can conclude your.
With all of these, the validations of the same thing that we have found in the section. Next. So definition of expression of the new setups of genes which helps in neuron development and in condition 3. Next. And this is amyotrophic lateral sclerosis again and devastating neurological disorder. We have collected patient samples both familiar and sporadic cases, and we have done to see if its expression of and in a specific mutation in a specific candidate genes, so particular candidates are identified to see if any change in the DNA, the blueprint of life, and how this could be useful for designing, specific experiment to recover the condition or restore the condition. Next. We found one level mutation, which is shown over there in NEK1 gene. Next. And this has already been published recently in 2024. And there are other mutations also in the candidate gene, which has some potential to give the condition worsen, and that is under study now. Next. The third really that we are also working on is autism spectrum disorder. You all know about autism spectrum disorder. Well, plenty of samples that were coming from that, and Ocanis is very much good there. About the phenotype and other things, parents found it, this patient had a loss of eye contact and gave him.
Dr. Das, I would request you to conclude, There is a debate, discussion, and time is out.
Using this stringent condition. Next. Results showing again in 2 patients, mutations in different genes. So likely one pathogenic mutation has been found in a gene, which is this is being published. Next. We are talking about different models. I'm skipping all these slides now and then go to the summary again, if there is more time. Yeah. So neurological disease has been found in a spectrum of things in different phenotypic conditions. This image may vary in severity. Genetic factors play a crucial role, which, in genomics radiation, next generation or latest generation sequencing has put up a proof to find out the changes in the genetic blueprint, which could be cautiously used for using in personalized medicine, and that's what we are targeting it. Next. So using all these classical to the high throughput techniques, we have found out a few very crucial mutation, and we now next aiming to make individual, IPA cells so that we can study it more properly to find out that what is happening, what is the exact mechanism, and how we can design for practicing personalized medicine. Next. So this is my bill. Professor Deepika Joshi from Neurology Department, IMS BHU, and this all for my PhD students. And thanks for your patient hearing. Very happy to.
Us? You can stay with us. There are question answer sessions now.
Okay.
Hello. Thank you, ma'am. I would like to call upon our speakers, Dr. Anand, Dr. Rohit, and Dr. Sunil to come up on stage for the panel discussion. A short one. We are already delayed, and we will come forward.
That is right. Karyappa. इसको तो पूछना नहीं है अभी ना. No, it's okay. Yes. Welcome, speakers. I think one speaker is online. Is Dr. Das there?
Yes.
Okay. Can you hear us?
Yeah, I can.
Yes, okay. Now, what we would like to ask you, as I think we are taking the opportunity first as the chairperson, that you be doing this wonderful research and experimental work in neurological disorders, especially late onset. How do you position, when you are so busy, the role of genetic counselors, you know, in clinical practice? Yes? Can you hear us? And we are just asking you, but how do you position the role of genetic counselors in your practice, where there is a late onset neurodegenerative disorders or other neurological disorders? Can you see something different or a great role in comparison with the clinicians?
You know, role of genetic.
Go ahead, sir. We can hear you. Sir, would you like to answer the question?
You know, the role of genetic counselor is the important thing as we all know. Now, there is a scarcity of the genetic counselor. Can you hear me?
Yes, sir.
Yeah. So, obviously, the role of genetic counselor is, yeah. Can you hear me?
Yes, sir. We can hear you. Go on. Okay, I think there is, we'll take the questions. So can we ask this next speaker the question? I think there is a problem.
The next
I
question.
think there is a time lag. And that's why it's not.
Yeah, okay.
Ma'am, please go ahead with the next questions.
The next question is for Dr. Anand, that you have been doing such a wonderful job for the bone transplants also, for the, especially to prevent the motility in hemat disorders and cons. But when you are so busy in your surgeries and clinical things and to give so much of time to the patients, don't you think genetic counselors will be very important there, and how do you justify their roles? Especially when there are very complicated, complex genetic variants and conditions, you know, where you have to explain personally, you know, to the parents in their own language, taking care of their emotions and ethics.
So rightly as pointed out by you, definitely the role of genetic counselors, I mean, as clinicians, we are less aware of genetic counselors, but as and when we are using more of genetic tests to diagnose these disorders, and we are stuck in those gray areas wherein there is a variant of uncertain significance, but the clinical picture is matching the disease, like I showed in some of the cases, there we have to explain them that there is a possibility, and then the genetic counselor's roles become important. To share an example, I had a case of Fanconi anemia. Clinically, she was fitting the profile, but the genetic diagnosis didn't show the thing. So we kept on pursuing that this could be a case of Fanconi anemia. So then we sent them to a genetic counselor so that they understand this importance of why we are so persistent on finding out the genetic problem for this child.
So I had a comment, it's a teamwork of the genetic counselors and hematologists like you to actually make sure that we actually report the silent mutations or incidental mutations to the database. Like in your case, you have to go back to the international literature and come back to this thing. So when you do a reverse phenotyping, you actually have to go back. The team is actually between the doctors and the genetic counselors to go back and make sure that that is reported. If it's a VUS, if it's an incidental mutation, if it's a silent mutation, so that the others who are working in the field can make sure that they have that, and it saves time for the diagnosis also. So just a comment.
Absolutely. It's not, and one more thing, it's not only that there is a difficult case which needs counseling.
Our case.
Because when you are talking about whatever variants you have spoken about, each required counseling.
Yeah, I agree.
Correct? And because they all required cascade testing, so which you haven't done. So you have done focused on, I don't blame you.
Yeah.
Clinicians are trained to look at a patient.
Yeah.
Counselors are trained to look at family.
Correct.
Okay.
So we had one interesting case like a T53 mutation. It was a germline. Yeah, I agree.
When it's not interesting, it's still important.
Yeah. Hello. I think you can now speak. Any questions from the audience? Yes. There. Mike, please.
Yeah, so there are two things. One is consultation, other is counseling. We should not get confused with the two. Because when patient comes to the clinician, he is oriented more to the problem, what patient is suffering from, what is the management and what are so. But counseling needs the psychosocial support and then understanding of the patient's situation, the family and so on. So I have been part of both. I did my clinician part, I did some counseling part also when I was pursuing family medicine. So I realized the patient's grief and their anxiousness and all those things only counselor can handle, and that too, counselor is not fully fledged when he starts the counseling. He also learns over the time. And I think when clinician is doing some part, that has to be contributed by the counselor. So we should distinguish these two things first, that consultation is different from the counseling, so that we can create more space for counselors in the clinical setup. And I am emphasizing this in my institute, that we should have a counselor separate post. We should not get confused with
Yes, I
consultation.
think we agreed on that yesterday, and we are with you. So what we can suggest, Dr. Anand, is that he should appoint a genetic counselors. Okay?
Ma'am, can I ask a question? Yes, so my question is to Dr. Sunil. So you have mentioned in most of your cases that counseling is different for both these siblings in many of your families. So how counseling was different? Can you explain in one or two lines, like in summary?
Actually, in the rare cases, the counseling cannot be done in all the cases in the same way, actually. So as you've seen in my slides, that there was a family that is having the same symptoms, and the both siblings are affected with the same disorder, and another sibling was having two different types of mutations. So counseling should be done in a different way. We cannot do the counseling at the same time with the, both patients, you know.
Okay, so by different, you mean that separately?
What you mean is at the same time, they should be differently tackled because variants are different and the role of counselor is different. That's all.
And at your center, like, do you counsel the patients, or
Yes,
is
we
there any
do the
genetic
counseling. We are
counseling?
the 4th member, and we do the counseling for the patients. We are not yet certified, but we do that.
Yeah, I think the situation becomes complex when there are two mutations in the same patient. And you have to diversify the counseling based on the mutation.
Yes.
Thank you.
We should include other factors as well, like environmental factor, if you can include that, the working conditions, the mental conditions, because that is very important for the prognosis, if we describe the prognosis of the patient. So that also can be taken into account.
So, yeah, this is something for Rohit that, India is a culture diversity capital and we have socio-economic landscapes so much diversified. So how do you think that with this kind of challenges, with India broken into urban, rural landscapes, how do you think newborn screening can actually reach to the millions at their doorstep? What do you think?
So I've been proposing for a long time, the dried blood spot is extremely robust. We could collect the social workers, Anganwadis. We've made a national program. They've got so many disorders on that RSY something, I can't even pronounce it. They just focus on CH, the thyroid. It's universal. That's a crime that's being committed. There should be a universal thyroid testing program. You collect blood spots when the baby is born, send it to a state lab, well controlled, well managed. On that blood spot, you piggyback other tests, so if you roll out a universal screening program, Pakistan has started,
Yes.
Bangladesh has started, Singapore, Australia, New Zealand, Middle East, all mandatory for thyroid and a whole bunch of other disorders. That should be prime concern. Somebody should drive that. Dr. Dave, you have a lot of contacts. That should, it can be done. And then these babies are life and death. It's
Yes.
in that first 2 weeks of life.
Dr. Karyappa, I agree with you. Last more than 2 decades, I think we have been, when the days we were pursuing newborn screening, when you go to the clinician and in the end we would hear that, yes, when I get a patient, I will send the test to you, So still it has been going on as a high-risk screening. I think Karyappa will agree that when the baby is sick, they will send it to TMS. They will compare with the clinical, phenotypes. But all over the world, even Asian countries, poor, they have started. Why can't we? I do not know. Sometimes I heard in the international, there has to be a political will. There has to be affected baby in the political families. Or even the national policies, also in the guidelines where those people who sit over there, I think when Sitaraman said sickle cell has to be, eradicated by 2047 has come. Suddenly from all the states, the projects of sickle cell disorder have started coming. Okay? So I think maybe such segments are there. But since this is a BGCI genetic counselors, people are over here at the genetic, after the newborn screening, positive or a negative, you are going to have a role, as a genetic counselors there. You have to be well versed with metabolic and other preventable conditions, because still I said that gene therapy is a distant hope. So prevention is better. And that's why newborn screening came as a policy, early detect, asymptomatically you detect, and that's why then further you can, in case detected, do the metabolic diet, and then further molecular genetic, and prevent the disability and recurrence in the family. So that's the whole goal which genetic counselors have to be ready. Okay, thank you.
I think just to add on, rare diseases initiative from the Government of India and the newborn screenings will always go hand in hand. So that it's always saying that whatever we find NBS should always report it in rare diseases, at least in the manual for people to refer to it. We don't lose time on that.
Yeah.
One more point that is very, here in the front row, ma'am, for newborn screening is, as a genetic counselor, when we see that the sample of the baby cannot be taken, the doctors are really busy treating the babies, and then the patients come back to us, ma'am, we lost a child, and what to do? Our first question in certain times were, did you do an NBS? Because you can isolate DNA from the dried blood spot, and I can go for an exome sequencing. Otherwise, the only choice left to a counselor would be to do a carrier screening test in the parents, which may or may not give an answer, which is again costlier. But if I have the baby sample, then it would be so nice to look into the baby. So the DBS is really a good thing. It would also help to store the samples.
Store the samples. So
in many of these IAMs.
Yeah, these are the points which these laboratories they always give. And I think few years back, we had pressed 5 years back DBS sample, which he sent the dried blood spot to us, and we further did the molecular mutation and prevented the next baby. So it's okay, he and me felt very happy, at least we saved that baby. But so much of our work is, involved in that. It's not that easy. But at least you should give a message properly how to collect the sample, take proper dried blood spot, and DNA can be stored.
Is there a policy to store these DBS slips, sir?
Yes, ma'am. So because of this extracting DNA, like she said, parents are upset, they leave the hospital. They don't even
Keep the documents, nothing.
they don't keep samples. If they've sent the newborn screen to us, we store that to 5 years. And then we
5 years, that's really good. That's
we
really,
kept it
really
cold
good.
rooms. We have thousands of samples. We started pulling out the positive samples and keeping it for research purposes, but we throw it at the end of 5 years. It's tragic.
So for all the counselors, your colleagues anywhere, so if some parent comes to you and you ask this one question, that by chance did your doctor send anything that was taken as a heel prick? Did they pick the heel and then take a blade and was it sent? Ask this question first and then send for carrier testing. But if that sample is not there, then anyway we'll have to test the parents. You'll save that money when you say that the cost is there. So testing the index child is always going to help. Okay.
And another thing is the fetal death. You may come across, and then that time the parents are in a very vulnerable position, not in a mental shock and trauma. You can't talk to them that give us the fetal tissue and give it the fetal sample. But that time you talk to the clinician nicely, keep some EDTA sample, keep some skin biopsy sample, keep it in the fridge. Maybe after 2-3 months or something, they are going to come. That time you can console them. That time you can counsel.
Yes.
So in fact, Dr. Karyappa was mentioning this morning that he feels really bad about having to discard samples after the 5-year period. And we were just discussing that the consent that you take must include for research purpose, that leftover sample can be used, that your storage policy is 5 years, after which it can be either discarded or used for research purpose. And we can help you with the wording and also the wording of what it should be, that what kind of research and whether it will be genetic research, so that they know, and then later on, neither of us is kind of held responsible that you didn't inform, even though the intentions were right. But yeah, this is a very stable and cost-effective way of storing samples, and really very important.
We've exchanged numbers. So.
Thank you so much. I think we are already, yes.
One last point since you had said, but remember there will be always a medical legal issues and ethical issues. The cases have happened in Australia and all where there are 10 years, more than 15 years, DBS there, and the children are grown up, there is a paternity dispute, okay, and then the children come to know who is a father, who is a mother, and then the medical legal cases, and Supreme Court there had given the order to just demolish, just burn all DBS cards. So when anything can happen like that, you have to be very, very careful.
Thank you so much. I think we are already running behind right now. It's late already. It is going on. So I would like to call thanks to all our speakers and Professor Parimal sir. We are sorry there's a little technical glitches happened, and I would request our chairpersons to please hand over the a minimal memento to our esteemed speakers. I would call Dr. Usha to present the memento to Dr. Anand.
Dr. Abhishek, could you please hand over the memento to Dr. Sunil? Dr. Usha, could you please hand it over to Dr. Rohit? We also request the speakers to stay on stage for a photograph. Professor Narayan Ran, we request you to please present the mementos for our chairperson and co-chair. I request you all to please stand on the stage for a photograph. Thank you.
Oh, yes. And after this, we shall take a small tea break for only 10 minutes. Sorry, we are running late right now, so I would request all the participants to come back by 11:10 to the auditorium. Thank you so much. Thank you, everyone.
Test. Change.
Good morning, everyone. Thanks, Dr. Annie Hassan and other organizers.
Test. 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 Test.
Check.
I request all of them in the hall to settle down quickly. We are shortly going to start the session. Please keep your cell phones on mute or switch it off if possible.
Maybe it's on. Okay. Good morning, everyone. Welcome to the second session of day 2 of our conference. So this is on reproductive genetics, PGT, prenatal. So we'll have some interesting discussions. I hope you are expecting. Okay, and I want your full participation. So the two chairpersons for this particular session are Dr. Vijayalakshmi Kodati and Dr. B. Bhaskar. And the moderators are myself, Dr. Madhavi, and with me is Ms. Pooja. And we will be taking you through the sessions. And please keep your notes open. Keep all the doubts and questions and we'll discuss, okay? So I would like to invite first our chairperson, Dr. Vijayalakshmi Kodati. She's a general physician, obstetrician and gynecologist at Vasavi Hospitals with an experience of 39 plus years in her field. She's completed her MBBS from Osmania Medical College with PhD genetics in Osmania Medical College and has an MR COG from Royal College of Obstetricians and Gynecologists. Her services is dedicated to pregnant women counseling actually on abortions, in discontinuations of pregnancy, fertility, conserving procedures, premarital counseling and complicated pregnancy treatments. She's a member of the Telangana Academy of Scientists and has a Distinguished Scientist Award in the year 2001. Welcome, ma'am. Over to you, Pooja.
Yeah, I'm here to welcome this second chairperson for the session, Dr. Bhaskar sir. Dr. Bhaskar is currently working as a consultant in laboratory medicine and heading the Department of Genetics and Molecular Medicine at Kokila Ben Dhirubhai Ambani Hospital, Mumbai. His previous affiliations include Shankara Netralaya as a genetic scientist, consultant, head of Department Medical Genetics, Apollo Hospitals, consultant and head at Madras Medical Mission Center for Genetic Studies and Research. With over 2 decades of experience in the field of genetics and 25 plus years of research professional experience in the field of human genetics, he has a PhD in genetics from Utkal University. He holds the UGC DSA Junior Fellowship from Mysore University, ICMR Junior Research Fellowship and CSI Senior Research Fellowship at Gujarat University, Ahmedabad. Additional qualification of his includes Stephens Shapario Memorial Fellowship by the Board of International Association, England, and IUBMB Young Scientist Fellowship from the USA. With a lot of poster and oral presentations given at national and international conferences. He's authored 70 plus original research and review articles and chapters and books. He's been awarded an AMP International Membership Grant Award from the Association of Molecular Pathology USA, ISCA Young Scientist Award in the section of Medical Sciences from Indian Science Congress Association, Calcutta in the year 2022, Precision Med India Awards, Medical Geneticist of the Year from BOH Next Edge in 2024. We welcome you, sir.
Over to the chairpersons.
A very good and happy morning to all of you, coming from all over the country. I'm from Hyderabad, and my co-chairperson has come from Bombay, Mumbai, and we are in Bengaluru. Good. Okay. I can see all the enthusiastic upcoming genetic counselors, as well as very nervous also the genetic counselors who are going to appear for their viva. No need to be nervous at all. See, you enjoy these talks and you can just walk through your viva. So I just want to reassure you all to be happy again. Well, there is a slight change in the order of the talks today because of the delay of the arrival of one of the speaker, and as well as a glitch in one of the speaker who is going to come online. So we will keep that towards the end. So I would like to start with the persons who are already here with us. I would like to invite Dr. Veronica Preeta Tilak, the first speaker today. Please come on, Veronica. Veronica is got her affiliations, as you can see from there, to quite a few organizations and institutes. She is a clinical geneticist and genetic counselor for more than 31 years. Wow, that's good. You have seen before the genetic counselors have come and now afterwards. That's good. You have good experience in that. She has done her MBBS from KIMS Bangalore, and we are proud to say that she's a certified genetic counselor level 2 from the BGCI. And her additional qualifications, she is an ABL internal auditor and program director for certificate course in clinical genetics. Teaching experience in genetics is 30 years. And career highlights are publications, both national and international. She is an advisory member to the Molecular Labs, MedGenome, Newberg, Supratech, and Strand Diagnostics. Great. Okay. And the member, Christian Medical Association of India. That's very nice, Preeta. I think you have a lot to offer to BGCI and hopefully the genetic counselors of the future. Okay? Yeah, you can start your talk, please. Thank you.
Thank you, ma'am.
And try to keep to your time.
Yes, yes. Thank you so much, ma'am. And I'm really grateful for this opportunity which is given to me to talk to the counselors, the genetic counselors, as well as the future genetic counselors. So most of my slides are going to be out of my experience, and I have a great, I feel very honored to be in front of Dr. Annie, ma'am, and Dr. Vijayalakshmi, who were my examiners when I took up the exam. Okay, so I am very happy that this opportunity has been given to me. So as mentioned, I have been in St. John's Medical College for the past 30 years. I joined in '93, and this was like a pre-genomic era, I should say, because those days we were doing karyotyping, and then after that, little by little, we went on to fluorescent in situ hybridization, molecular cytogenetics, and from there, we went on to the molecular medicine part of it. So I have been seeing the difference in the training, in the diagnostics, as well as in the clinical trend of genetics. So I just like to go to the first slide. First slide, please. Yeah, thank you. So in the first slide, I put up this about a difference. We always say genetic disorders. But I think there is a demarcation between a chromosomal disorder as well as a genetic disorder. So as all of you know, chromosomal disorders are involved with chromosomal abnormalities. But when you talk about genetic disorders, we are talking about inherited conditions with a single gene mutation. Okay. Now, most of the time I work in a clinic. We have a genetic clinic which is happening in St. John's, and we have referred patients coming from different specialties for the genetic counseling and genetic evaluation. So the most important thing which I have noticed over the period of my experience is, as the patients walk in, they have this sort of a uncertainty and anxiety in them. So they feel the curative and the definitive preventive measures are unavailable. Okay, this is what they feel. Oh, genetics, I don't think it can be cured. I don't think there is any treatment. So, but I'm going there to find out what is there and what can be done. Inability to affect the outcome and absence of treatment modalities. This is the mindset with which most of the people who come for genetic evaluation is seen in our genetic clinic. But of course, we know that this whole concept is changing, and it has changed, and we have to understand that the amount of information that we give to the patient is more important. They have to understand what they are going through. So the families, they pursue the genetic counseling in an effort to demystify the mysterious. They are not sure. They are not at all sure about what genetics is, what it involves, and how it can help them. So as a genetic counselor, it is my primary responsibility to help them to understand this. They seek answers and information, an honest evaluation and a clear explanation of all the possibilities, and a sensitive exploration of all the available information and knowledgeable decisions about their future family planning. Because this is a subject where we are not talking about only the present, but we are also talking something about the past, and to take the future also into consideration. Next slide. Okay, so this is my clinic where I see my patients, and as a genetic counselor, I feel that there are certain attributes which we have to have when we are practicing this profession. So be interactive and empathetic. Now, these are the two most important factors which, on a personal basis, which I have felt. Be interactive. Allow the person, the others, to talk. Listen to them first, okay? So that you can respond to their need. This is what is most important. And an empathetic attitude is very important. They don't come with a very big burden, but the burden increases once they get the information and they understand what their problem is. So it's a stepwise manner in which you have to deal with these patients. And it is a challenging scientific and a structural conversation between a genetic counselor and a patient. It is challenging because we do not have enough genetic counselors to help the amount of patient load that we are having. The patient load is increasing, and it is very important that you have you're placed with a challenge as genetic counselors to do or carry out your profession. It is scientific, it has got a lot of scientific basis, and it is a structured conversation because it is not only the scientific information that you're going to give to them. There are more points, there are more things which will come out in the process of the conversation with your patient. Essential components would include identifying the patients with genetic disorders, your communication skills, and clinical management. Now, one might ask, I'm a genetic counselor, how do I deal with the clinical management? Yes, you do have a role to play in the clinical management. The patients who are referred to from clinicians, they are the primary physicians who are going to take care of your patients. But at the same time, I am sure that your role as a genetic counselor is going to greatly influence their decisions as well as the decision of the patient. The theme of the conversation should be multifold: psychotherapeutic, education about the disease, and control over the situation and helping them to adjust and cope with the situation in a better way. This is totally in the hands of the counselors who can help them to overcome their fears and anxieties. And it helps the patient to make informed decisions. Okay, everybody has a right to information, and it is very important that they make an informed decision so that they are happy with whatever they have done. They should not feel guilty at the end of it, or they should not feel that they have made a mistake. And it's an ongoing process through the lifetime of the patient, because we are talking about generations together who will come back to you if they trust you, and the doctor-patient relationship will continue. Next slide. Yeah. So this is, I'm supposed to be talking about ethical dilemmas. Okay. So I just wanted to put a definition to it and tell you what is actually described in the textbooks about an ethical dilemma. So it involves the need to choose from among two or more morally acceptable courses of action when one choice prevents selecting the other, and the need to choose between equally unacceptable alternatives. This is the definition which is given. But in a clinical setup, in a genetic consultation in the clinic, both the doctor as well as the patient has got a moral responsibility, an ethical dilemma which the doctor also goes through, the counselor also goes through, as well as the patient. So what are the ethical dilemmas which the patient undergoes? There are a lot of attributes which are important for them to make a decision. There can be legal implications, religious, cultural, social, and the family pressures which go along with it. Now, in our Indian society, I have never or very rarely seen individuals coming on their own. It is a family affair. There are people who are within the family who come along with the patient. They may be well-wishers, they may not be well-wishers. I don't know about that. But, or to complicate the situation, I'm not sure about that. But still, there is a support within the family, and they look forward for this support, and they come together for the counseling. Now, from the doctor's point of view, we have to maintain our ethical principles, okay? At the same time, look into the legal aspects and respect the religious background with which the patients come from. So this is very, very important. Religion may not, or maybe you might say that it should not, make a big impact. But in our society where everything is in balance, your social, your family, as well as your religious concepts or your belief is very important in your life, because we are such a diverse population, and a diversity exists not only in our social setup or in our economic setup, but also in our religious setup. So it's very important that we respect that. So what are the 5 basic ethical, I'm sure all of you would have seen this, but I'm just sort of reiterating the principles. So this is patient autonomy, beneficence, non-maleficence, justice, and confidentiality. Of course, I have described what each one means. Next slide. Okay, so there was a study, actually, as a matter of fact, yesterday one of our speakers had also spoken about this low and middle income countries where we are finding a lot of findings are coming through, and at different levels of scientific research as well as population studies. So within this group, there was also a study which was done about the ethical, social, and cultural issues which are related to these countries. And as you can see in the map, the blue color is our India, and almost around 20 to 25 studies have been done under this area, and they have come out with certain key points which seems to be similar from all these studies. And some of these key points are the consensus, the total consensus after the study is that to bring genomic medicine from the lab to the village. How do you do that? Okay. In between, there is a huge area where the scientific research is going on, new discoveries are being done, treatment modalities are there, and there are a lot of diagnostic investigations which are being done. How are you going to bring that to the village level? The in-between level is the urban area. We are quite aware of it, okay? And the doctors and the people who refer the cases to us are also aware, so they're very happy about sending samples to the labs, informing the doctors, probably contacting a genetic counselor and taking their help. But when you go down to the level of an uneducated or less aware area, how are you going to do that? That is the challenge that all of us need to face, not only as doctors, not only as counselors, but all of us need to face that. And I feel that it is a personal responsibility on our part to, advocate this so that everybody in this nation is involved. So some of the points which they came out was genetic counseling has a tendency of being directive. Now, our universal consensus is non-directiveness, but there are people who can make the decision for you. People are confused with the amount of knowledge which you're giving to them through counseling. So they look forward for a moral support, somebody who can decide for you. You're not sure, you're in a dilemma. You don't know what decision to make. So they look forward for, and I've had many patients who ask me, Madam, what will you do if you were in our place? Okay? It is a very practical thing. I'm being very practically telling you, or I'm going to tell you what all is happening in my clinic. So do I have the right, or should I make the decision for them? No. Please don't do it. However much the pressure may be, do not make the decision for them. You will be biased in doing that. Okay? So, but the general or the conclusion which they have got from this study is that it is being directed. So there are people who say, okay, you have a child, or the ultrasound report shows that it is a fetus with multiple congenital abnormalities. Please go ahead, get it terminated. Okay, there are people, there are situations where this happens, but you do not have the right to make a decision on that pregnancy. Please remember that. So genetic services have psychosocial consequences that require improved support. Now, what do I mean by this support? There are organizations where we have, or associations which we have seen throughout the world, who give a lot of support, not only psychologically, even in terms of education and financial support. This is not there in India. How many societies do we have? How many associations do we have? There are institutions, there are hemophilia societies, there are thalassemia societies, there, of course, but not as an association which can cater to the needs of the patients who do not have the facilities for these sort of societies or associations. That is what I'm talking about. So they do need to improve even at that level, to create support groups who can help them to understand their situation. Medical genetics training is inadequate. This is a very pathetic state. I can tell you that in our medical curriculum, we still do not have genetics. Okay, whether it is in first year or in the final year, it is not adequate. Part of it is taken in pediatrics, part of it is taken in obstetrics and gynecology. Oh my God. Okay, I'm sorry. Okay, so then I'll just go on. So genetic services are difficult to access. The social determinants affect the uptake and understanding of genetic services, and the social stigma is often associated with genetic disease, and family values are at risk of disruption by genetic services. Religious principles pose barriers to acceptability and utilization of the genetic services, and cultural beliefs and practices influence the uptake of information and understanding of genetic. Can I go on to the next slide? These are the conclusions which they have identified. I'll just quote two examples and I think I will finish. As all of you know, as far as reproductive counseling is concerned, we have to find out some carrier status, and that will influence in the prenatal diagnosis and the pregnancy, possible pregnancy termination in the case of an affected fetus. Now, this is a case where the couple came to me with a history of the first child having congenital deafness. We did the genetic screening. We found a mutation, GJB2 gene, which is quite common, and the parents wanted to go in for the second pregnancy. They went in for the second pregnancy, amnio was done, and they identified the same compound heterozygous mutation. And so they decided to terminate the pregnancy. Now the mother is so petrified that she says, no, I will not go ahead with another pregnancy. I know that this is, both of us are carriers. So at least let me have a baby through IVF with the PGD, okay? And she opted for a donor egg. Now, I work for a Catholic institution where prenatal diagnosis is not at all spoken of, okay? And nothing is practiced. So I could not actually, confide with her and sort of have a consensus with her saying that she can go ahead with a donor egg, because she was capable of having a pregnancy by herself, and prenatal diagnosis would have been sufficient enough for her to make a decision. Next slide. Now, this was another case of ectodermal dysplasia. The father had ectodermal dysplasia and he got married, and in the first pregnancy, we had told them that they can go through amniocentesis or prenatal diagnosis to identify the same mutation which the father had. But the wife was so, confident. She said, no, I do not want to do any prenatal diagnosis. When my mother-in-law has accepted the child with ectodermal dysplasia, I too am willing to accept whatever the outcome of my pregnancy is. So she went ahead with the pregnancy. She delivered a child, a female baby. But after about 1.5 years, they came back for evaluation because the child had features of ectodermal dysplasia. So we did the testing and we found the same mutation even in the baby. Okay. Now the story doesn't stop here. Everything seems correct. Got married, he has the mutation. The first child has also got the mutation. But what I wanted to tell, the interference of the family. The mother was not happy with what happened. That is the boy's mother. So she got him married to another person. Okay. Ma'am, this is a real life story, ma'am. Really. She got him married to another person, and she, the second wife, became pregnant. She delivered a boy baby who was absolutely normal. Okay. So now this was done without the consent of the first wife, and the first wife filed for divorce, and also for the case is still going on, and this is the complication which took place. Now the first daughter was willing to accept whatever the outcome of her pregnancy was, but the mother-in-law did not want to accept it. It was the same like what happened to her son. So she went ahead, got him married to another person, and luckily, it was a boy, okay, which was normal. This is an X-linked condition, okay? But if the second wife had also got a child with the same problem, I don't know what she would have done, okay? Yeah, we will get him married again, okay? Now, next slide. Okay, so these are some of the things which I wanted to tell. Again, there's a shortage of time, but probably we can discuss it. These are some of the conditions, these are some of the situations where we come across for reproductive counseling. And to reiterate, your pedigree analysis, your history taking, and if there are any red flags which you can see in the history, please be very aware of it so that you can pursue the counseling with the couple. Next slide. I'll finish in 2 slides, ma'am. 2 slides. So in reproductive counseling, there are 2 important legal aspects which we come across in our clinical practice. One is the medical termination of pregnancy. As all of you know, it is going to be extended, or it is already extended from 20 weeks to 24 weeks. My only concern here for this is medical termination of pregnancy should have some ethical values before you really decide on it. And I would request, with all due respects to the obstetricians who are seeing these cases, to refer them as early as possible for the prenatal diagnosis. Because we've had situations where they come at 20 weeks, 22 weeks, and we are not able to conduct the investigations to confirm the diagnosis and make an informed decision in case they want to terminate the pregnancy. Next slide. Okay, surrogacy is another thing which is a very controversial situation and has got a lot of ethical and legal issues. One of the reasons why I put this up was to give you some examples about cases where they come with Turner syndrome or Mullerian agenesis. Surrogacy is required. And as a matter of fact, one of the examples that Dr. Anand gave was Glanzmann thrombasthenia, where they can have a lot of obstetric complications at the time of delivery. So many of them choose, they choose to go through surrogacy, and there are certain laws and restrictions which are required if you want to adopt this particular procedure. Next slide, my last slide. So in summary, I'm sorry, again, there's got a lot of material here, but again and again, I just wanted to reiterate, please, as you're going through your counseling sessions with your patients, please make sure that you consider it as a from a holistic point of view. Please do not rely only on the scientific information, because even the scientific information is becoming very ambiguous, and it is very important on our part as genetic counselors to give the best option to our patients, because genetic counselors are now becoming very important. Because I always tell my students, you think of a cell with a nucleus, you have to be knowledgeable about any tissue which becomes pathogenic. So you have to know the background of the medical situation and help the patients to make an informed decision. Thank you.
Dr. Veronica. There's a slight change in the order. I can see Dr. Vandana Hegde has arrived. So I would like to invite her. You ready with the talk? Yeah. I would like to invite Dr. Vandana Hegde. She is the clinical director and chief IVF consultant at Hegde Fertility in Hyderabad. And her talk will be role of PGT-A in reproductive medicine. And some cases are presented so that, following that, we can have the genetic counseling in pre-implantation genetic testing. That's what the order we have put. Okay, you want to introduce? I would request Dr.
Hello. Yeah. So it is a great pleasure in introducing Dr. Vandana Hegde, who has established Hegde Fertility Center at Hyderabad since 2010, and also received multiple awards, Excellence of Fertility Care, and running 1 year full-time fellowship in art, the program that she has been doing since 2019. And her academic, coming to academic, she is a MS OBG from GSS Medical College, postdoctoral fellowship from reproductive medicine from RGUHS.
University, yeah.
I do not know. Anyway,
Bangalore.
diploma in reproductive medicine, and embryology from Germany, and various additional qualifications starting from Padma-Bhushan and various things.
Okay.
I think it is on the screen. Over to you, madam.
Yeah. Very good morning to all of you. So it's nice being a part of the BGCI conference, and this is my first time over here. So it feels nice to be among all the brains, all the PhD scholars over here, a different doctorate. So I'll just start my talk. It's the role of PGT in reproductive medicine, where I'll be describing partly PGT and little association with the cases that we have seen. So, so there are a few cases which I'll just run through so that you can, apply your mind and think of the answer or solutions for these patients. So case one, we had a case of 29-year-old with married life of 5 years, presented to us 3 miscarriages, all 3 spontaneous miscarriages between 7 to 10 weeks. And on evaluation, all her RPL investigations were normal. Just the husband karyotype showed Robertsonian translocation. This is case one. Case two was a 37-year-old, slightly on the elderly age, with 5 years married life, little diminished ovarian reserve, AMH of around 1. She conceived on IUI the first time, had a blighted ovum. Second IUI led to a biochemical pregnancy, and then she opted for an IVF conception. She conceived in the first attempt, but had a miscarriage at 9 weeks, and we sent her POC for microarray, which showed multiple aneuploidies. We had a case 3, a 30-year-old, as doctor has already just presented before me. It was an affected child, previously affected child, Duchene's, and she came for planning her next pregnancy. So we need a solution for her. And case 4 is a 40-year-old, slightly more advanced age, with married life. It was a late marriage, so just 2 years married life with an AMH of 0.8. That means her egg reserve is declining rapidly, with all other fertility parameters being normal, her uterus, her tubes, her male parameters. She opted for 2 IUIs which failed and went immediately for an IVF cycle, where she got 8 oocytes. She had 3 good embryos, blastocyst seen. She had a first transfer with a single blastocyst which failed, followed by we did 2 blastocyst transfer in the next cycle after checking her hysteroscopy, evaluation for her, implantation issues. But again, she has failed. So what is the option for her? Is it a donor gamete? Is it a repeat IVF? So these are things that we'll have clarity. So now what are the questions from my patients, what we see regularly is, why is this implantation failing in spite of you doctors telling I had good grade embryos? Then other question is, why am I recurrently aborting? So these are constantly, and I can only tell you that these questions last 5 years, I'm seeing an increase in miscarriage rates, and even, the women have started presenting towards later part of their life. Now the mean age of my clinic has increased from 28 to 33. So definitely we are heading towards. So these questions, I think we all know necessity is the mother of invention. So it's these questions that you start thinking, and then you realize that at the end of the day, the embryo is nothing but it's partly the mother and the father's DNA over there. So that means if something is happening, probably there are changes at the oogenesis and the spermatogenesis stage itself, where any alteration in the genetic makeup will lead to disturbed function of genes and the chromosomal pattern. So any error in this development of the early egg, sperm, or the embryo could lead to an abnormal number of these chromosomes and could be one of the factors. So the question comes, should you screen embryos for chromosomal abnormalities? So one thing we have understood today with all the evidence over the last 20 years is abnormal embryos don't implant, and they do end up in an abnormal baby. So we have been checking embryos on day 3 and day 5. And if you look at it, as the age is advancing, definitely the percentage of embryos are increasing to a staggering, by the time a woman is reaching 40-80% of her embryos are going to be abnormal. Even at 35 years, 50% of the embryos are abnormal. So when it thinks of day 5 also, they match the day 3 status. So if what we are more worried about is patients, if they are landing up late to the fertility clinics, if they go through an IVF cycle, what is the thing is the miscarriage rate. They have a higher miscarriage rate, and that's something that is very worrying, because every miscarriage we cannot, she can't conceive for another 3 to 6 months. Again, her age is advancing. So we need to optimize their pregnancy at the correct time. That's very important, that timing. So with all the evaluation of RPL, we have now understood that 80% of the problems lie within the embryo, and in the embryo, 90% of the problem is with the maternal oocyte. It's only 10% of the issues where the male sperm parameters or the uterine factors are reasons for the recurrent pregnancy loss. Now, what we do standard whenever we in fertility clinic is the morphological grading, which has been, the standard for the last, 40 years that we have been practicing IVF. So this morphological grading, if you look at the picture here, this is the 2012 picture where they have done 2 blastocyst pictures. They look absolutely the same morphologically. But if you look at the CGH, the data, you can actually see that it is only on CGH that you actually pick a good quality, because we routinely tell patients that your embryo quality is good, though we are seeing it actually morphologically. It is only, you, the genetist, who actually added value to the work that we are doing, that where you actually go today and you have answers to the problems, where we didn't have, the absolute answers. So today we can explain patients why she failed in spite of her embryo grade being good. So now couples seeking fertility treatment, it is not just about getting pregnant. They do want to have that one healthy child. This is something that we routinely hear in clinics today. Doctor, just do something that I have a healthy child. So looking at that now, if you see, we have to follow guidelines. We have to follow evidence-based medicine. So the common societies that we follow, the ASRM guidelines, the RCOG, NICE guidelines, and the ISHRAE guidelines. But what they have been telling for the last few years, that you start evaluating recurrent pregnancy loss only from the 3rd loss and above. And this was creating a lot of angst in the population, young, as I told you, for the age of the pregnancy is going on advancing, and this delayed diagnosis, the poor diagnostic experience, the dissatisfaction with care has all been increasing internationally. And there's a lot of genetics that has advanced over the last decade. You can see your, the platforms that have moved from FISH to CGH to NGS today. So the way genetics has progressed, all this new evidence that ISHRA had to go back, and they've updated their recently, 2022, they gave a recent update on the guidelines, mainly because of the advancement in genetics. So what they recommend today, the first test to do in a patient with RPL is the POC testing, because this is the only test that gives you a lot of information. So the first test that you do, POC, so these are graphs that they've put, that if the embryo comes euploid, just look for other factors. That is the major, the one important thing they tell. Other part they tell is, if the POC shows an unbalanced translocation or an inversion, then you do the parental karyotype, and based on that, you send them for a genetic counseling for PGT-SR. I'll just come back to the terminologies. Now, if the POC comes aneuploid, should you jump to PGT-A? No. They tell do not do, because we know that lot of patients do have, abnormal embryos. That's a norm in human pregnancies. So the first time it comes aneuploid, they tell don't do anything. It's an expectant management. But again, we need to consider what is the age of the patient and these other factors. But if there is repeated aneuploidy, recurrent aneuploidy, then they do tell consider PGT-A. Now, IVF, I'm sure you're all aware of the technique, what we do. The only thing is here, when we stimulate the ovaries, we collect the oocytes, we fertilize the oocyte with the partner's sperm in the lab, that is through intracytoplasmic sperm injection. And then these embryos are cultured in the lab for 3 to 5 days. So right now, we are doing PGT only on blastocysts, that is the day 5, day 6 embryos. These embryos are then biopsied. So we use laser on the outer profectoderm layer. We put a shot, we just open it a little. We aspirate few cells, about 5 to 6 cells. And then the embryo that is being biopsied is then frozen in our cryotanks under liquid nitrogen. And the biopsied sample is then tubed and it is sent to the genetic lab. Where today what we follow is the NGS technology, and the report comes as either the euploid embryo, aneuploid embryo, mosaic, or sometimes, you may have a low DNA, it could be inconclusive result. So now, is it safe? We are putting laser, we are doing biopsy. So there's a lot of literature and evidence that has been, got for the last 20 years that PGT is absolutely safe. So we ourselves in our clinic have been practicing for the last 6 years, and we have got good outcome in PGTA patients. And there has been any abnormality seen where we have followed up the children also. So definitely PGTA is very safe to choose a healthy embryo among all the good morphological grade embryos. So coming to PGT, the terminology, what was initially PGS and PGD, the new terminology is the PGT-A, PGT-SR and PGT-M. Now PGT-A is only for the aneuploidy testing, where you look for the presence or absence of the chromosomes. PGT-SR is where you look for the structural rearrangement that detects translocation, inversions, deletions, and PGT-M is for the single gene diseases. So if you look at the cell from the embryo, so when you actually look at only the chromosome numbers, it is PGS. When you come to the end, you separate the DNA, look at the gene, is the single gene diseases, PGD, the previous terminology. Now, balanced the ISHRA guideline specifically looks at this because couples could have balanced translocation, but when by that time the progeny that could lead to unbalanced translocations. Now, why they tell this is mainly because of the recurrence risk. See, once there is an unbalanced translocation, the recurrence risk for pregnancy loss can be somewhere between 50 to 70%. So that is the only indication they tell that where, it is recommended to go for a IVF with a PGT-SR. Now, will PGT in general, will it increase the live birth rate in the general IVF population? The question everybody asks. So this is SART data, which looks only at the RPL, that's patients with recurrent pregnancy loss. So if you look at it, they have studied almost 40,000 embryos. So in this lot, you can see the live birth rate. Definitely this is the live birth rate, which has shown without PGT, if you do in RPL patient, the live birth rate is 33%. But if you add PGT, it nearly goes to 47%. Very significant increase. So in RPL patients, definitely there is a role for PGTA. Now, what they did in US is, once they got all this data coming, they started applying it to in general population. So they started thinking that this is going to improve the live birth rate and all. But what they saw was the absolute opposite. That means the pregnancy rates, in fact, started decreasing for the women who were younger. So PGTA is not a solution for all the problems, just doesn't improve your IVF success rate. So once they saw this, they have a STAR trial which was released recently, where they took all the patients aged 25 to 40 and everybody was included in the PGTA cycle. What did they see? Between 25 to 35, there was no improvement in any result, but as the age crossed 35, between the 35 to 40 bracket, they saw significant improvement in the pregnancy rates with PGTA. So if we look at PGT as an answer, it's probably only for patients with advanced maternal age of more than 35 years. Repeat implantation failure, there is more than 2 IVF failures where you have transferred good quality blastocysts of at least, 4 in number. Recurrent miscarriage of more than, 2 or more losses. Previous trisomic pregnancy and probably a severe male factor that could also be a reason for aneuploidy. Now, PGD for single gene diseases, yes, this is an accepted modality today, where it allows selection of the non-affected embryos before implantation in couples who have risk of having an affected child. So in this, we have autosomal dominant disorders and autosomal recessive disorders, with dominant having a chance of having 50% of affected children and the recessive having the 25% risk. Same thing goes with X-linked diseases, where we can have the risk with X-linked dominant and the X-linked recessive. So these are accepted modalities of treatment. Now, when the PGT in today, we are moving from the invasive biopsy to non-invasive, just like how we have the non-invasive screening for prenatal testing. We do have this non-invasive coming up in a big way now. A lot of research is being published, but it is still under research, because right now the concordance and there are many labs which are, not yet confirming whether, it gives the same result as a non-invasive. Now, there are a lot of pros and cons with invasive versus non-invasive PGT. Because what they tell is non-invasive is where the embryo is in the culture dish and we are taking the cell-free DNA. That is, we are taking the culture medium and sending that for testing. Whereas in invasive, we are actually touching the trophectoderm. So controversy is that does trophectoderm actually is it totally indicative of the inner cell mass? And in non-invasive PGT, what they tell is because it gets the DNA from the whole embryo, it may be more indicative of the euploidy status. But where we have done a trial of non-invasive PGT, we found a lot of higher mosaic rate of these. We've been doing invasive PGT for a very long time. So we as a clinic are still not satisfied with the non-invasive PGT, and many genetic, even the diagnostic centers have still not yet moved to non-invasive. And oh, okay. Yeah. So just going back to the case, now that we know that terminology is everything, I think now we have the solution for our patients. The first case, who was just a 29-year-old, 3 miscarriages with Robertsonian translocation, what we did was a PGT-SR for the patient. And this patient with miscarriages with an advanced age. So considering her, we presume this is probably because of recurrent aneuploidy, we did PGT-A for the patient. And the previous ditchinies, we did a PGT-M. And this patient with advanced age of 40 years who had recurrent implantation failure, the next cycle of IVF was done with PGTA. So now this is a 40-year-old. So people keep asking me up to what age. So in our clinic, the highest age of the female where we have a live, pregnancy going on, presently she's reached 30 weeks, is 43 years. Now what we need to understand is, as a woman's age is increasing, she will not produce that many oocytes. Now the euploidy rate is age specific. That means by the age of 42 to 43 years, her chance of getting an euploid embryo is only 10%. That means for 10 blastocysts, she will have 1 blastocyst normal. So they need adequate counseling, that they will need more number of cycles to be able to achieve this. So this patient at 43 years, her AMH was 0.3, very low egg reserve, and other parameters were all fine from the male factor. And when she went with IVF cycle, she needed 4 stimulation cycles. We did 4 egg pickup cycles for her to pool the number of blastocysts. And we sent around 5 blastocysts for PGT, out of which she got 2 embryos being euploid. We transferred a single blastocyst and she's conceived and she's reached 30 weeks of pregnancy. This is the highest age at our clinic. And so this is a paper that we published at, this is presented at FIGO Foxy conference. So this is a presentation on recurrent pregnancy loss. Actually, we were presenting it on thrombophilia, where we divided the patient into non-PGT and PGT groups. And if you look at actually, when a patient is having RPL, where we rule out thrombophilia, give all the adequate support, It is only when we added the euploid embryos, the pregnancy rates were very high in these patients. The continuation of pregnancy. So everything they had a better outcome, which crossed almost 70 to 80%. So many patients with RPL, definitely PGTA is one of the first recommendations today when it comes to RPL. So this is by accident, I would say, this case, because today we are in the BGCI conference, which is mainly a conference for genetic counselors. So this patient just came to us, and she was just a 26-year-old with 3 years married life. She was a primary subfertility, and she had just failed multiple TI cycles, IUI cycles. She had borderline sperm. Last slide. Yeah, she just had borderline sperm parameters. So we were thinking, from IUI, let's start IVF. So while just, thinking and talking to her about IVF, that's when a small, the husband had a slightly, intellectually, he was looking a little low. And, when you started questioning, then you came to know that even the sister was similar. So then the wife realized that, okay, there is some issue. And because we have an in-house genetist, that's the added advantage. You just send any doubts you have, to them. So that's where, you realize that there could be an issue. And this couple, though they were young, with no other history that they were actually aware of, the genetist just told that, yes, it looks like there could be a problem. So they received, a proper counseling, and with their, consent, they were sent for whole exome sequencing. And, accidentally it was discovered that the husband is the carrier of the VAC gene mutation. And this could be, autosomal dominant mutation, which could lead to, severe intellectual disability in the child. So I would tell this was just an accidental discovery. And then what we did is, we went for an IVF cycle, which they were anyway planning for. We did a biopsy of their embryos. So the 7 blasts were sent for PGTA. Out of the when it was sent for PGTA, we got 4 euploid. These 4 euploid, when we sent for PGTM, they got 2 healthy embryos. She underwent a single euploid PGTM normal embryo transfer, and she conceived in her first cycle, and she is now due to deliver next month. So I would tell that today, in this era of, with advancing genetics, I think that this case would have been missed, So we have got ART law in India today, ART Act, and it is very unfortunate that genetics are not a part of it anywhere. So that's something that I think Dr. Rani is here today, and we need to, I think, put across, there are a lot of, seniors who tell that, see, every parent would want to have a healthy child, and we are all aware that in US, any couple that goes for IVF carrier screening it's a routine, the couple carrier screening, which we don't do it here. And the most important part is Dr. Veronica just mentioned a case where the patient just opted for a donor oocyte, right? So I have a patient who has had a donor oocyte with an affected child. We have seen, many of you would have seen. So donor oocyte is not an option. This also I want to tell genetists that please convey this message to your patients. In fact, we got a lot of educated patients who want their donor screen because they're aware. I think we have screened 3 donors. All the 3 had some issues and we could not take up the donor. Because now we are very careful in suggesting donor gamete also. So coming to the ISHRAE guidelines, this is my last slide. So if you look at the ISHRAE guideline, how they have modified today is, the most important is this, where they accepted that genetic tests are, coming to first line today. And the most important thing is genetic counseling plus, it is not plus or minus, it is always a plus information on PGT. So it's something that I think your genetist, your pen doesn't go towards writing that PGT, because I've seen a lot of, counseling sheets coming from genetists where they tell the recurrence risk, 50% you're good, and they don't tell recurrence like that. They tell 50% chance of having healthy child. So that is something that you stress upon, I agree, but we stress on the 50% chance of having an unhealthy child. I think that is where we need to come to terms, and these are platforms where I think we get to interact with each other, because this is the first time that I'm getting to interact with all of you. So this is something that is mandatory as genetists, that you give this option also. Yes, ethical dilemmas, we have our own religion, St. John's Hospital, yes, we all agree that, but I think when we are doctors, we leave all that behind, When we counsel patients, we should give evidence-based and, what is the best for them. Yes, technology is important because it creates the future. We are able to be a part of the next and create things that don't exist. So that's the most beautiful part of you, genetics. And yes, technology is always a double-edged sword. People can use it in the wrong way. That is what we are all scared. I think genetics are very scared of clinicians, that we may use it, where it is not indicated. And that's a fact, because I have patients coming with, hardly 28-year-old, 32-year-old, first IVF, PGTA done, failed and come, where there was no absolute reason to do PGTA. So there are clinics doing that. There are clinics, somewhere at 40-year, AMH is 1.2, actually good for a age, having good egg reserve, 5 and 5, still doing donor gamete. So this is where that, because people are not having, clarity on, because the technology is not being spoken and not being told where to use, where not to use. That's something that we should know. So if we accept this PGTA fast, I think we're getting ready for the future of gene therapies. I think that's in the pipeline. So this is something that is very much necessary today, that we, start collaborating fast and give this hope to many couples. And probably that is the need of the hour today, that we collaborate. Yeah, thank you so much.
Thank you. Thank you, Dr. Vandana, for an exciting talk. I think we are going to have a panel discussions and we can have some, clarification on this.
Thank you, Dr. Zubeda, for the organizing secretary, for showing in touch with me till I landed here. And Dr. Vijayalakshmi Kodati has delivered me actually. So nice to see all of you over here. And I would actually like to thank Dr. Sachin and Dr. Sodhi, who are my pillars for genetics in our center. Thank you so much.
Thank you, Dr. Vandana. Yeah. Dr. Vandana, be available for the panel discussion
at the end of all the
and the end of the talk. Now I'll call upon the third speaker, Dr. Pratati Chaudhary, who is a scientific advisor and genetic counselor in iGenomics, and a consultant, senior genetic counselor, and a visiting genomic consultant in Hegde Hospital in Hyderabad, and a genetic counselor and service compliance manager at GeneTech Hyderabad, and a molecular biologist in Dew Point, India, Hyderabad. And she has completed her postdoctoral fellowship from IIT Hyderabad. Additional qualification, she has got number of publications in national, international conference journals and book chapters. She is a reviewer of articles in various journals in human genetics and genomics, examiner of Indian Society of Assisted Reproductive Gurukul program for medical students, and she has 9 years of experience in genetic counseling. Over to you, Dr. Pratati.
Thank you, Dr. Vibhas, for your kind introduction. Good morning, everyone. Before I start my talk, I would like to thank Dr. Annie and the organizing committee for giving me this opportunity. And the topic for my talk is genetic counseling in PGT. So I will take this opportunity. I chose this topic to speak to the respected clinicians that we have here, as well as the genetic counselors who are at different clinics or labs, to communicate what are different types of PGT. I think there is, in the introduction already Dr. Vandana gave us, to differentiate the different types of PGT, and also what reports and what information we look for as a lab who is offering the PGT services. So these are the three types. The previous terminologies were PGS and PGD, which we still keep hearing when the patient comes, that there's a confusion about the PGS or PGD. They want, actually there is indication of PGT-SR, but they'll come that we have to do a PGD. So I'll just explain about these three types, what exactly we do and what it covers. So PGTA, we can use it as screening based on the obstetric history for screening for aneuploidies, checks for all chromosomes. PGT-SR, structural rearrangement, if one of the partners is identified with a clinically significant rearrangement like translocation inversions or Robertsonian translocation, and PGTM, which is for monogenic disorder, if there is a risk for single gene defect identified for a couple, then we offer PGTM. So this is the overall sequence of events that happens. This is as per the ESHRE guidelines that happens. So I have categorized it. So basic requirement for the IVF and PGT center for all the three types of PGT that is offered. So patient inclusion and exclusion, which I will explain in details. What are the criteria? How do we decide and accept a PGT case? Counseling and informed choice. So we empower the patient to take informed decision as genetic counselors. Then the biopsy part, the embryology team's part. Previously it was polar bodies, cleavage site, D3, biopsy. Now we do blastocyst stage biopsy. Sample collection. In some cases of inconclusive result, they may also go for a rebiopsy. Sample is sent to the genetic lab and we do the testing. There is a pre-examination process, examination process, and post-examination process. And finally, follow-up after the selected PGT tested embryos are transferred, following up till the birth of the baby. I'll start with PGT-A and SR. So, regarding the pre-test consultation, we get from the clinicians or sometimes directly the patients are referred to us for this consultation. We may have different indications like RAF, RPL, recurrent pregnancy loss, severe male factor, or advanced maternal age being the primary indication for us. We look for the karyotype and CMA reports of the previous pregnancy. This is very important, especially for the PGT-SR cases. If one of the partner is identified with a clinically significant rearrangement, we should have the previous, it's very helpful. And we always, apart from the PGT counseling, when I do other specialty counseling, we should always insist on if there is a pregnancy loss, a POC should be evaluated by chromosomal microarray. So that if the parents have a rearrangement, looking at the breakpoints of that translocation, we can correlate that whether it's the same breakpoints which has resulted in the segmental aneuploidy that we see in the pregnancy or not. Depending on the CMA and the karyotype report, in some cases, we may recommend them for going for another testing by a higher resolution banding. Maybe your patient has a regular banding karyotype. This is helpful. So if we have these previous reports. And this gives us the accuracy and when we review a case for PGT-SR in our platform. The family history, obstetric history, this is very important. And whether to go for self, because many a times the couple, after hearing all this that there is risk, they also ask us whether is it safe, is it okay to go with a self cycle or we should go for a donor. So it is our responsibility to tell there is a percentage risk. We know there is a percentage risk of abnormality, but we have this percentage chance of getting a normal embryo also, where you will have a healthy baby. So with this, we check for the PGT-SR technical feasibility to accept a case. And also one more important point, a karyotype with a normal polymorphic variant, usually for this, we have to go for a PGT-SR because there is no clinically significant breakpoints that we have to check for. But in view of obstetric history or advanced maternal age, they can go for a PGT to rule out aneuploidies. This is generally the sequence that happens. Once the genetic lab receives the biopsy samples, we do the NGS analysis, and with the commercial available software, we generate the PGT-A and PGT-SR report. Now the post-test consultation or counseling with the patient. These are the types of results that we may see in the PGT-A or SR report. Euploid, aneuploid is very clear. We know if it is aneuploid, we don't recommend for transfer. Euploid, we can go ahead with the transfer. This one factor of aneuploidy has been screened. They can go ahead with this. There can be results of complex aneuploidy. Again, a no for transfer. The other types are mosaic embryos. So this is mixed type of results that we are seeing, which is again categorized as high mosaic or low mosaic. So as per the recent guidelines by PGDIS, which came in 2021, the recent one, and ESHRE guidelines, the low mosaic, high mosaic, we still don't have evidence. Low mosaics can be considered for transfer if you don't have normal euploid embryos or patient cannot or do not want to go for another cycle. This can be considered, but it has to be strongly followed up with a prenatal diagnosis, screening and diagnosis. We should confirm the final chromosomal makeup. Two non-inclusive results, non-informative or no DNA. So this is again up to the clinician, the clinic team, embryologist. If they feel it's okay to go ahead with a rebiopsy, this is one option that is given to them. We can do a rebiopsy and a reanalysis, and we can rescue some of these embryos. of the inconclusive ones. And finally, the transfer recommendation as per the result, and always any PGT, which again for PGTM I'll say, any PGT we should reconfirm. So there is a difference when we do in a case of natural pregnancy or where the PGT is not done. There when we do the prenatal diagnosis, it's confirmation. With PGT, there's a technical limitation of maybe 1 to 2% that it might miss out. So we suggest to do a reconfirmation by the prenatal diagnosis. Now coming to the PGTM for monogenic disorders. For all patterns of inheritance, we can offer PGTM. These are some of the examples that I have shown for autosomal dominant, recessive, X-linked condition. Along with PGTM, we can also plan a HLA matching for conditions where there is severe immunodeficiency or hemoglobinopathies, where they have the couple, they have an affected child with thalassemia, for example, and they are planning next pregnancy to have a child without thalassemia, and also this sibling, future sibling can be a donor for bone marrow transplantation, where HLA matching is helpful. So we can test for both the parameters, the HBB gene variant which is identified in the couple, and also check for the HLA region for matching. Now, the pre-test consultation for PGTM. There are many factors we see. So one first point always for PGTM. PGTM is not offered for a condition. PGTM is for a gene variant at risk identified for a family. So we get some queries like there is a couple, previous child has autism, they want to plan PGTM for their next pregnancy. Then we have to dig into what are the genetic reports do you have for the child. They don't have in some cases. Maybe for few only Fragile X or CMA report is available. So we know as genetic counselors the sequence of testing that we have to recommend for these children. If there is a chromosomal abnormality, further testing as per the findings for the couple to confirm the carrier status. If it is Fragile X, confirm in the mother if she is a carrier. If there is a single gene defect, confirm whether it is de novo, which is mostly the case for autism, or a susceptibility factor. So this is important. We should check all the genetic reports that are available for an affected pregnancy or an affected child, and also check if the parents are confirmed carrier or not. That is, it is de novo or there is recurrence risk. So the genetic reports are evaluated, family history is taken, and the genetic counseling letter is very important here because they finally what the patients they decide to go ahead with. Always we give, if there is no fertility issue, but they have an affected child, the couple is worried, they don't want another child with a condition. They have both the option in that case. Natural pregnancy with a prenatal diagnosis, but as genetic counselor, it is also our duty to tell them this PGT testing, this is also available based on your reports. The main difference being natural pregnancy, until you do the prenatal diagnosis, we don't know if the pregnancy is affected. In PGT, you have the option before the pregnancy happens, we are ruling out. It's more than 98 to 99% accuracy testing. We can rule out, and then you do a reconfirmation, that's all. So this difference has to be explained, and it is up to the couple now finally they take an informed decision. Also, one very important point, for a PGTM case, pre-PGTM workup is necessary. So, there are some cases where I get a call from a clinic that, we are going to do the biopsy. There is this mutation that is identified. We are sending you for PGTM. It was not consulted before. So we have not done the pre-PGTM workup. So this is very important. If the moment you see that a patient has indication for single gene defect, immediately this should be consulted, and we should work as a team to work on the pre-PGTM workup that we have to do, where couple sample is always mandatory. Depending on the variant, the inheritance pattern, we decide the additional family member sample that should be provided for all the available family members. Certain other important points. Definitely, it has to be non-directive approach. We do not have any guidelines about the inclusion or exclusion of a PGTM case. So our duty is to inform all the possible testing, all the benefits, limitations of our testing, the PGT testing. And finally, it is up to the patient where there are many factors. Can be financial factor, many factors where the couple finally they decide, take a decision which one to go for. We cannot tell that PGT is the only option. So this should be non-directive. Once they decide to go ahead, the counseling part is not only with the genetic counselor. This is very important in cases of PGT. The counseling with the consulting IVF specialist, the embryology team, and the genetic counselor. Because these are different sets of questions that these three teams will get. And we should all work as a team in helping and addressing all the questions that the patient has. The indications, there are some indications like inconclusive, VOUS, variant of uncertain significance. We should do clinical correlation for those cases. Or conditions where there is a predisposition factor, multifactorial conditions, like ankylosing spondylitis, rheumatoid arthritis, where we know there can be a genetic factor, but we also know if there is a genetic factor identified in a person, he or she may have other family members who have the genetic variant, but are not affected. So these are just susceptibility or predisposition factor and can be managed with lifestyle modifications. So for these cases, there's ethical issue. We do not offer PGTM because we cannot be sure if the variant is there, the child will be affected or not affected. And this can be managed. In cases of VUS, if with the pretest counseling, we come to a conclusion that we may change the VUS to pathogenic or likely pathogenic, and we come to a conclusion that this variant is significant, even though the report says it's VUS, but for this family, this is significant. And we plan to go ahead. Patient also wants to rule ahead. Imagine these couples, if they have a child affected, they are seeing the child, they want to rule out that variant because they are under this impression that this has caused the condition. In these cases of PGTM, we always add one, disclaimer and explain this to the patient with the clinical correlation and our assessment as genetic counselor.
Both of
this looks
2 minutes.
Yes, I will conclude. I don't have many slides. So, we are going to plan the PGTM for the variant of uncertain significance and not the condition, because we know maybe there is something unknown which we are not able to identify. So this limitation has to be explained. And carrier screening report, another challenge in some counselings. Couples come with a carrier screening report, maybe 2-3 conditions, both the partners are carrier for all VUS, no significant family history. Does not make sense. But when a patient gets these reports, they are really anxious and worried. Seeing conditions like epidermolysis bullosa and something very serious that they are carrier for, they get really worried. So we have to explain what are these classification and how that impacts. There's no family history, we cannot correlate. So this we have to decide. Or in some cases, if they're going for a donor, the recipient say they're going for egg donor and the male partner is carrier, they still want to rule out that carrier status. Again, we cannot accept these cases, does not make sense. Ethically, again, not right. Because one thing is just the carrier status, and if we don't have the donor's report or donor is negative, then there is no risk of being affected, the affected embryos. But if we don't have the donor's report, in that case, we don't know if the donor is also carrier for any mutation on the same gene or she's not. So if that unknown mutation is there in the donor, we cannot screen. So we cannot offer PGTM for these cases. In cases of donor, we always tell the recipient partner from the couple should also be screened. For example, recessive condition. The donor might be identified as carrier for multiple recessive condition. We should also then know about the male partner, saves time. So this should be informed to the patient that we can do this. And with PGTM, we can do PGTA on the same biopsy sample. So we are ruling out both aneuploidy as well as the single gene defect risk. is the report interpretation, which one to transfer, and always prenatal diagnosis after PGTM. There's a difference again in the HLA matching cases. We always hear what is the accuracy, what is the percentage, what guarantee you're giving about the HLA matching in the sibling. So when we do this PGTM with HLA matching, this is different. We cannot really define and give a percentage. Final matching is decided after the baby is born and HLA typing is done. But with the PGTM and HLA matching, it's almost like 100% because we are checking all the antigen, HLA region antigens. So in summary, PGT consultation and counseling, this is a teamwork with the clinician, embryologist, and the genetic counselor. Pre-test counseling, chromosome analysis reports for a PGT-A and SR is very important, and the previous reports, if available, for an affected child or pregnancy. Pre-PGTM is always needed when we plan to offer PGTM, and PGTM and PGT-A can be offered on the same biopsy, can be done for the patient together. Post-test counseling for mosaic PGT-A or SR, non-informative or inconclusive results should be offered, and prenatal screening or diagnostic to be planned and explained to the patient when they come to you with the PGTM report for a decision which one to transfer and what to do next, and follow-up of PGT pregnancies for each case as much as possible. Thank you. This is my contact address.
Thank you, Blatati. I think you should join us for the panel discussion. Please be available after the next talk. The last talk we have is about expanded carrier screening for reproductive genetic counseling. This is an online talk by Ms. Juliana Lee, a genetic counselor. Which place is she? Yeah. Yes, her biodata is available there. She began her practice at the University of Malaya Medical Center, and then she founded the Genetic Counseling Asia, okay? And she's also a visiting lecturer for the Master of Medical Science Genetic Counseling Program at the National University of Malaysia, member of the Rare Disease Committee under the Ministry of Health, Malaysia. She has done her BSc Honors in Genetics from National University of Malaysia, completed a graduate diploma in genetic counseling from University of Melbourne, and graduated with Master of Counseling from University of Malaysia. And her additional qualifications are, she is a GSA certified genetic counselor from Malaysia, over 20 years of clinical experience. Her career highlights are, she is the founding member of Malaysian Rare Disorders Society, past president of the Professional Society of Genetic Counselors in Asia, and secretary of the Genetic Counseling Society Malaysia. Yes, I welcome Dr. Juliana Lee. We can start her talk without any delay. Thank you.
Good afternoon, everyone. Firstly, I'd like to thank the BGCI organizers for inviting me to speak on the topic, expanded carrier screening in reproductive genetic counseling. I've been practicing as a genetic counselor for the past 20 years, and I spent the first 10 years in public health hospital in Kuala Lumpur, Malaysia, and another 10 years in a private setting from 2014 up to present. It was in 2014 that expanded carrier screening was first introduced by commercial laboratories in Singapore and Malaysia. Over the last 10 years, there has been a lot of changes in terms of test coverage for carrier screening, and in recent years, there has also been an increase of cases referred for genetic counseling, especially preconception carrier screening by fertility centers in Malaysia and Singapore. In my talk today, I wish to share my thoughts gathered from managing cases that require expanded carrier screening, present a case that highlights the genetic counseling issues, and end the session with challenges and future directions. So let's start with the introduction to carrier screening. Carrier screening is a type of genetic test that can tell whether you carry a gene for a certain genetic disorder. is also known as preconception carrier screening, as it is ideally done before pregnancy to allow an individual or couple to find out their chances of having a child with a genetic disorder. So carrier screening can be approached by two ways. One is targeted, whereby the known genetic variants based on personal or family history is actually ordered. Secondly, expanded carrier screening, whereby there will be a list of autosomal recessive and X-linked conditions that will be tested in the gene panel. So the goal is to provide the couple with information on their reproductive risk of having a child with a genetic condition and options to reproductive genetic testing, such as prenatal diagnosis, pre-implantation genetic testing for monogenic conditions, and considerations of egg and sperm donors. Many professional societies have shared their position statements for carrier screening, but I wish to highlight the statement by the American College of Obstetricians and Gynecologists, also known as ACOG, in 2017 and reaffirmed recently, as I believe this has influenced the uptake of preconception carrier screening, especially in private fertility centers. So ACOG recommended that information on carrier screening should be provided to every pregnant woman, and carrier screening and counseling ideally should be performed before pregnancy to enable couples to learn about their reproductive risks and consider the most complete range of reproductive options. Next, we'll see what are the genes or conditions selected in the coverage of carrier screening. So a guideline for selecting conditions was developed by American College of Medical Genetics, known as ACMG, and the American College of Obstetricians and Gynecologists, known as ACOG. However, due to the lack of clarity, these recommendations may be interpreted differently by carrier screening providers, resulting in discrepancy in the available screening panels on the market. Therefore, in 2022, ACMG developed a tiered approach to screening, whereby there are 4 tiers, starting from Tier 1, which comprises of cystic fibrosis, spinal muscular atrophy, and risk-based screening. Tier 2 comprises of autosomal conditions with not more than 1 in 100 carrier frequency. Tier 3 would be the recommended tier for all pregnant women and those planning for pregnancy. Tier 3 comprises of autosomal recessive conditions with not more than 1 in 200 carrier frequency and X-linked conditions. So for Tier 3, there are 113 genes that comprises of 97 autosomal recessive conditions and 16 X-linked conditions. Lastly will be Tier 4, whereby the list of conditions would not be more than 1 in 200 carrier frequency. So genes and conditions will vary based on the lab providers. And Tier 4 will be recommended when a pregnancy arises from a consanguineous relationship, from second cousins or closer, and when a family or personal medical history warrants further risk assessment. Also to keep in mind, ACMG does not recommend offering Tier 1 and Tier 2 screening because these do not provide equitable evaluation for all racial and ethnic groups. And ACMG also do not recommend routine offering of Tier 4 panels. Next, I would like to share with you on the development of carrier screening for reproductive planning as observed in my clinical practice as a genetic counselor. I started practice in 2004 at the genetic service in a public hospital in Malaysia. And we were at that time only focused on offering targeted carrier screening for known variants detected mostly in pediatric patients diagnosed with rare diseases or for individuals with genetic conditions or with a family history who are planning to start a family. Targeted carrier screening was offered for parents planning for their next pregnancy after having a child affected, and for them to consider prenatal diagnostic testing, which are mostly sent to overseas laboratories. At that time, pre-implantation genetic testing was not yet available. Thalassemia is also offered as a premarital or preconception screening, and for pregnant women, as it's a high carrier frequency in Malaysia and around Southeast Asia. 10 years later, in 2014, when I started to move to private practice, that was when expanded carrier screening was first introduced by a diagnostic company based in Singapore for the test to be conducted in the US. There were only 102 autosomal recessive conditions, including some X-linked conditions, that were offered in their panel. Every subsequent year after that, the gene coverage for lab providers of expanded carrier screening began to increase to 200, 300, 400, 500 gene coverage. And as of last year, I came to know of providers covering up to 1,200 genes and 2,500 genes, with options to test for SMA, DMD, and Fragile X syndrome. In 2023, there were also some labs offering whole exome sequencing and whole genome sequencing-based carrier screening. As a genetic counselor, in comparing the test options available in the market to present to our patients, two main questions arise, which are, firstly, how many genes will be enough for carrier screening, especially to plan for a pregnancy? Secondly, if more coverage is preferred, should we replace expanded carrier screening using NGS panel with whole exome sequencing or whole genome sequencing? Currently, less than 5 commercial laboratories are offering carrier screening using whole exome sequencing and whole genome sequencing for healthy individuals. Most clinical labs will only test for symptomatic patients using whole exome sequencing and whole genome sequencing. The varied test options should be curated to provide the couple with the most suitable test options to determine their reproductive risk by highlighting the benefits and limitations of each test. And test options should also meet the patient's needs on gene coverage and also comparing cost of testing. Next, I'd like to share with you on the utility of expanded carrier screening in Asia. So, targeted carrier screening for thalassemia is widely available in Asia due to the high prevalence, unlike expanded carrier screening. So, as mentioned earlier, expanded carrier screening was only introduced in 2014, and even then, it was actually promoted by a Singapore diagnostic company for the test to be conducted in the US laboratory. And since then, most of the expanded carrier screening actually sent out to US or Europe laboratories, as only a small number of laboratories in Asia have the capacity to conduct this test locally. So of late, direct-to-consumer web-based companies have also started offering expanded carrier screening, although not extensive, and a handful of those labs actually conduct the test in-house. Because of this direct-to-consumer testing, new issues have arisen based on the clinical cases that I've managed, whereby there are inconsistent report formats, and some of the tests actually could only be accessible through mobile applications, and some of the test reports are actually not of clinical grade testing that can be used for prenatal testing or PGT. And also there is limited genetic counseling support, especially after the test has been done. In 2017, after the ACOG recommendation, it has really influenced the uptake of expanded carrier screening, especially among couples that seek for fertility services through IVF and PGT, and also considering egg donors. Recently, some countries in the Middle East have also started offering preconception carrier screening due to high consanguinity in their population, with a good support from their local government. Next, I would like to share an interesting case managed for preconception carrier screening using an egg donor. Presenting this case because it had several turn of events that was finally resolved at the end of the case management. The case was referred by an IVF specialist at the IVF Center for genetic counseling to review carrier screening reports. for patient's husband, a 34-year-old Caucasian man with Irish Western European ancestry. His wife is a 42 years old Chinese woman, previously diagnosed with breast cancer at the age of 25. So this couple have tried to conceive naturally for several years but was unsuccessful. So in the end, they decided to consider an egg donor who is also Chinese, and the patient plans to carry the pregnancy. So I was provided the expanded carrier screening report for husband and the egg donor. At this stage, I know that the husband is a carrier of Wilson's disease and the egg donor is a carrier of hemophilia C. So the first issue detected was that there is a different coverage of expanded carrier screening between the genes tested for the husband and the egg donor. So their tests were actually sent out to 2 different laboratories in the US. So the husband ordered the expanded carrier screening panel from a genetic testing company offering web-based service. And he was tested for 265 genes at a US lab that I know is currently offering up to 569 gene panel. So the question is, why was he only offered 268 genes from the test ordered from the third-party website? So upon checking with the couple, it turned out that 268 genes were the highest option available when they ordered a test from the website in February 2023. And after we checked in December 2023, when the case was referred, their website was actually later updated with the expanded gene panel. So unfortunately, the lab in the US is unable to expand the sample for testing because the sample has been discarded. So the second issue is that the husband's test did not include coverage for hemophilia C, and the egg donor was detected to be a carrier of the likely pathogenic variant in F11G. So the test options to be presented in the pre-test counseling with the couple would either just test for the first option, hemophilia C in the husband, or 2, to consider a more comprehensive expanded carrier screening that will also offer coverage for F11 gene. So in the pre-test genetic counseling, the first thing I did was gather personal and extended family history. So the third issue that came about that I did not expect was that the husband has a history of premature ventricular and atrial contractions at the age of 30. And his half-sister was diagnosed with atrial ventricular block at the age of 17. So as we discussed the limitations of expanded carrier screening, and since the couple will be going through IVF and considering PGT and reviewing their reproductive risk, the husband is keen to test for cardiac gene that may be related to his symptoms. Also, from the family history gathered, husband's mother, uncle, and maternal grandfather had their gallbladder removed, and there was an unconfirmed diagnosis of cystic fibrosis in the late maternal granduncle. So after considering the options, the test that was presented to the husband was to consider whole exome sequencing to identify potential cardiac genes related to his symptoms, and also this whole exome sequencing actually covers carrier status for autosomal recessive conditions, and we've made a point to request for the lab to look out for variants in the F11 gene. So 1 month later, the whole exome sequencing report was issued and reviewed with the couple in a post-test genetic counseling session. So the whole exome sequencing reported no pathogenic variants detected related to patient symptoms of arrhythmia, premature atrial, and premature ventricular contractions. So the variant detected in ATP7B was detected and classified as VUS. husband was not detected to have the pathogenic variant in F11 gene, similar to the egg donor. However, he was detected to be a heterozygous carrier of cystic fibrosis in the CFTR gene, which was classified as a missense pathogenic class 1. And this was not reported in his first carrier screening report. So now this is actually the fourth issue on why this pathogenic variant was not reported in the first expanded carrier screening conducted. So looking at this case, we can see that there were conflicting variant classifications detected between the two laboratories, and it may be possible that the cystic fibrosis variant was not reported by the first US lab, maybe because it was actually at a VUS status in February 2023, which was possibly later reclassified to a pathogenic status, but the updated report may not have been shared with the husband because they ordered it from a website, and there was limited follow-up provided to them. So on the hindsight, if the whole exome sequencing was not conducted and expanded carrier screening was not reported in another lab because they were considering an egg donor, the husband would not have known he is actually also a carrier of cystic fibrosis. So looking back, the couple were thankful that they are now clear of their reproductive risk, and they can proceed with IVF using the egg donor. So the reproductive risk was given at 50% chance of potentially having a child that may be a carrier of cystic fibrosis, Wilson disease, and or hemophilia C, based on the variants detected in the husband and the egg donor. So it is recommended as well to test their potential child for his or her carrier status when they reach adulthood, and if they are found to be carriers, to also test their reproductive partners for the variants concerned. It is also recommended to share the carrier status detected in the husband with his half-sister and extended family members for their consideration of carrier screening. So following that, the limitations of expanded carrier screening were explained, followed by prenatal screening and test options when a patient is pregnant.
Okay, Dr. Juliana, you have
his presentation. Here are some key points in genetic counseling to be considered when offering carrier screening.
Dr. Juliana? How many more? 5 minutes. 2 minutes. Yeah, this is a recorded session, so there's 5 more minutes. We'll take 2 minutes. That's fine. We'll cut down. We'll run through the slides and then.
So as you can see, family history is still a valuable assessment tool for genetic risk. So even though the case has been just referred for carrier screening, should pay out of pocket to have a carrier screening conducted either from private laboratories locally or to send it out to overseas laboratories. And with the introduction of direct-to-consumer testing by ordering expanded carrier screening on the website, some couples find ways to purchase testing online, but eventually if the providers are also not responsible enough, the patients will actually encounter issues like having reports that are not of clinical standards and also limited access to genetic counseling support. So lastly, the public health setting is actually not prioritizing the importance and the need of developing reproductive genetic services, especially in developing countries. And this includes advocating for expanded carrier screening to reduce the incidence of genetic conditions and rare diseases in our community. So in summary, genetic counselors need to be updated and informed as always on the latest advances in genetic testing. not just on carrier screening, but also on various genetic tests to know its benefits, its limitations, and the gold standards of testing for different genetic disorders. So we should create more awareness on expanded carrier screening, also to help couples evaluate their reproductive risk of having a child affected with serious genetic conditions. And lastly, healthcare providers not trained in genetic counseling should refer patients for pre- and post-test genetic counseling for a more holistic clinical management.
Thank you, thank you. Thank you, Dr. Juliana. Is she coming online for the panel? Yeah, fine, thank you. That's fine. So I think now I would ask the moderators to take over.
Thank you, ma'am. Thank you, speakers. I would request the speakers to please come on stage for the panel discussion.
A quick question for Vandana. Like Juliana spoke about screening, do you all recommend SMA, DMD, Fragile X, and sickle and thal, at least for the donors? Please do it because we are getting...
Hand over the mics to the speakers here. You keep that with you.
Yes, so what we do is, we do counsel patients who opt for donor gametes regarding this risk. We follow the ART law, the ART Act of India. So as per the ART Act, there is no recommendation to test at all. Yes, but we do counsel the patients an option to test. In fact, we do advise carrier screening itself, where we can match the couple with the male partner with the female or any donor gamete. But these specific... Yeah, anything, donor gamete. Yeah. If they're opting for donor gamete, we do counsel them. Just a female going for an IVF cycle in general. So usually we don't tell all the patients unless there is a prior family history. So patients, what we do is we do a regular electrophoresis for all patients, the couple. We do a karyotype for all the couples prior to their IVF. So these two things, and then if by their family or history we have any indication that they may require, we send to the genetist to have a counseling session with them. But we don't do for all.
So for all couples who come for PGT, a general carrier screening is not recommended or offered, is what you are telling.
Yes, yes.
So don't you think because they already have lost 2 or 3, couple with either infertility or RPL, any pregnancy that they are going
RPL, yes. RPL we do evaluate like that.
general
Carrier screening based on their history, because consanguinity
definitely there is no history, no consanguinity.
Yeah.
Because any pregnancy they are going to have will be considered as a precious pregnancy. So by any chance, if the couple is a carrier for a condition that cannot be picked up on ultrasound, so would it not be better to, at least offer them or tell them that there's a carrier screening?
Yeah, we do tell affordable patients. See, we do tell because then it heads towards PGTM also. So that is something that we just give an option to patients. And today many patients are asking for it. We need not even tell because...
Is there any guideline in ART that you should not offer?
No, they're not mentioned whether you should or should not. Both words are not there.
Okay, so negatively
it's all left to the clinic. It is all left to the clinic.
I think it takes time for the guidelines to come. It's just in the evolving stage. These guidelines will come later. Like yesterday when one of them suggested that whole genome should be done as a newborn screening. So what, for pre-implantation itself, again the whole genome sequence should be done or what? In one of Juliana's slides, she also put it up. So those recommendations, guidelines, it will take time for us to come formally. But right now, on case-to-case basis, it depends on the treating obstetrician or the genetic counselor or the geneticist who has the whole pedigree with them and know the condition, and offer it on case-wise basis. So don't stick to guidelines. If you feel there is a need, you discuss with the team and then go ahead. That will help.
Juliana, can you please give me your opinion? Like, if a couple comes to, to you for counseling, opting for a PGT, opting for IVF, not in general about PGT, but they're going for an IVF itself. So do you want to offer them a carrier screening or an expanded carrier screening for that matter?
Be a reason for you to do that. Do you
Yeah.
have the justification for that?
To rule out recessive
No, you
conditions.
can't just say rule out. How many will you rule out? Some justification should be there. When we are practicing evidence-based medicine,
See, we got one case just last week where they came to us for the delivery, okay? Because India, anybody can go anywhere. When she came for the delivery, we were doing the routine antenatal because she has come for the first time to Kammanahalli, and we found that she is a sickle carrier. Now, 32, 33 weeks, we naturally said that we'll ask your partner to get tested also. Then they said that this is a donor sperm. So you tell me, how do we help them at that point in time? Because we can't find the donor.
Yeah, so ART Bank of India, the donor sample that we get for semen is tested for thalassemia, sickle cell anemia. They do. That is what they claim at least. So the lab that we are getting it from does tell that this is a negative thalassemia, sickle cell anemia, and I think we should keep very
True, ma'am. But the thing is, see, as genetic counselors and geneticists,
Actually, we don't screen donor gametes. So now the law tells we are a ART clinic. There's an ART bank. Now ART bank, we are not supposed to be doing it. So we have to source the gamete through the ART bank. So the responsibility lies now on ART bank, which is not run by doctors. Now that's the truth that we have to accept, that they're not run by doctors.
no, the question why we are asking this is because we want to rule out these rare conditions. See, when
But then going back to the last slide of Dr. Veronica,
is rare is actually common for us every day. Common for us every day.
Yeah, exactly. So what she was telling, cystic fibrosis may not be that common in India.
So that is, ma'am, we do see, because when you do a carrier screening test in general, a couple comes to us, they are carriers sometimes.
I mean, go back to the ethical dilemma, the slide which Dr. Veronica has put in the last. So we are imposing so many things on a couple who is just married. Even the couple's relationship is not well established between both of them. The family members are still not used to, getting adjusted to them. They land up with infertility. We are doing IVF. And then we are landing up with a career screening report. So there are a lot of issues that we need to consider.
So that's why we recommend a pre-test counseling, so that, they understand the implications.
That's something that we do.
Doing the test or not doing the test. See, what I'm asking is not an advice. We should at least offer them, tell them that there is a test available like this in the world.
Yes.
And if you do, these are the pros and cons. If you don't do, these are the pros and cons.
Yeah.
And then let them decide.
We are doing it for PGT now, for women above 35. We just, tell them that this is available. It's not like we are forcing them to do. So we do tell them this is an available test. Career screening, wherever we feel history-wise, it just shows that we definitely advise them.
But, and for consanguinity, all couples.
Consanguinity, 100%.
Yes, we all do it. Okay.
Can I? Yeah. First of all, thank you so much for those wonderful talks. Thank you, Juliana, for joining. This is Suraj here. In this, fast-paced life, of course, the average age of reproduction is increasing, and that's why these services also. I almost ended up having a debate. Let me put my thoughts in coherent because a lot of discussion. Please excuse me. I will quickly just put it across. Yesterday, I had a discussion with one of the scientists on whether to have guidelines to regulate PGT. I mean, I respect his point of view as well. He said, why at all do you need such? Because we have everything available in India, right? So you had stated examples as such. Young couple coming after a PGTA failure, you do not know why it was suggested. This is why we need regulations and guidelines in our country. And what I am afraid is that not just eugenics, I mean, there are groups such as sociogenomics working on, today you do for conditions as you wish. I mean, there should be set of conditions that are approved to be, proceeded by centers to be allowed to do PGTs, right? Not anybody can come and ask, I don't want this child to be having this condition. That is eugenics. So, sociogenomics is coming up. Today we practice this. In future, we'll talk about traits and we screen for that. So, please comment on having guidelines and how do we go about it. Experts in this forum, please.
See, what I would tell you is if you have to do a PGT, one, the clinician should have sufficient knowledge on genetics. They should have an in-house genetist. The embryologist should be well trained and experienced in doing the biopsy procedure. The sample that it should go to a well standard quality controlled genetic lab. So it is when all these fall in place, that is when you have a good euploidy rate and a good pregnancy rate. Now what has happened is with NICS coming in place, there are so many centers which do not have a laser biopsy because the machine itself costs around 18-20 lakhs. Now with non-invasive screening, you don't need that. So what has happened is there's a rapid shift of so many IVF centers towards non-invasive testing. And I've seen patients having a lot of embryos tested with non-invasive and a lot of irregularities in the reporting with respect to the euploidy, mosaic. So it is becoming a haywire in India. I would tell you that that is why that regulations have to come. In fact, we were hoping that with the ART Act, PGT, they thought that the licensing and the regulation, the check mechanism should be more stringent in India, is what even I would feel, because that's what I'm seeing. That they should tell whether.
I'm waiting for for a long time to just interact with you. How do you integrate your PGT results into your overall IVF strategy? And what scientific guidelines do you follow to make informed decision about embryo transfer?
So see here, when we have an infertility couple, the most important is that we do karyotype for all before IVF. So there we screen at least where, the structure PGT-SR is required or not. When it comes to PGT-A, it is only the advanced maternal age. So we take a cutoff of about 37 years actually. 37 above, we definitely counsel every patient who's going for an IVF that you have an option of PGT-A. Recurrent pregnancy loss patient, irrespective of the age, if they have 2 or more losses, we do screen them again, everything, and give them an option of PGT-A. Again, PGT-M is only where it is indicated. So they come with the family history or clinical history based on that, or history of consanguinity, where we do the carrier screening and the whole exome sequencing and decide what it is.
Specific ethics, evidence-based guidelines being followed here, or is it you follow the international guidelines?
Only international, because India doesn't have any, doesn't work here exactly, but we have our own boundaries, because that is the reason. See, what happens is here, at least the center where I'm practicing, we are like a team of doctors. So we have a clinical team of about 10-12 consultants. We have a genetist, we have the embryologist, and we have the genetic lab where we send the sample to. So since multiple, people are involved in decision making, I'm sure ethical boundaries are not, crossed. So that is where team helps. Because as an individual person, when you take decisions, you are biased. Where you push, where you withhold, those things can come into play. So when you have a whole team giving each one's, viewpoint on that particular case, I'm sure we take right decisions with group discussions.
Does the number of embryos play a role in offering PGT, for example, in infertility couples?
Infertility couple, again, it depends on the age of the patient and the reason why they're doing. In recurrent pregnancy loss, if we get even 4 blastocysts, I would say we do get a good enough 1 or 2 good euploid embryos for transfer. The only thing is, as the age is advancing, it may take sometimes multiple stimulation.
So you follow the same guidelines both for RPL and infertility. Even if there are, 2 embryos, do we not take a chance?
See, what is the thing is, if you have single euploid embryo, the chance for pregnancy is about 60%. Okay. If you have 2 euploid embryos, the pregnancy success rate goes to 70 to 80%. With 3 euploid, we have a 90% chance that the patient conceives. So we usually aim at having minimum 2 before we start the transfer.
Yes, Ma, you can go ahead.
Hi, I'm Dr. Presi Joy from AIMS Bhubaneswar. I learned a lot from this session, especially because we are doing prenatal genetics. I have one question for Dr. Vandana. I want to know whether you have come across cases with PGT for familial cancers, because BRCA1, BRCA2 carriers, we saw yesterday there were a lot of carriers, right? So I just wanted to know about that, if you have come across or if there are any guidelines.
No, right now we have not done any PGT specifically for BRCA or familial cancers. No.
I can answer. There are usually because of its incomplete penetrance, you do not offer prenatal, but pre-implantation definitely for high penetrant variants for BRCA1 and 2 only were done actually in our country.
I would also like to add, we do offer for BRCA1.
Yes, BRCA1 and 2.
BRCA1 and 2. I mean, in fact, I have one patient, we are doing the pre-PGTM workup now. She is BRCA positive, very strong family history.
Yeah.
And we can.
So again, as Dr. Bratati said, yes, you can tell that, when you should consult with the lab, look at the variant, because as she said, you're not looking for the high penetrant variants with a strong family history where prenatal cannot be offered. You go for a PGTM.
And we should also confirm that the affected family members, that makes the case stronger, like maybe her mother or sister also affected. The variant has been confirmed in her. This is a strong case for us to rule out that in PGT.
Yes, Upasna.
Dr. Bhatati, I have a question. So how do you handle actually and interpret? How do you handle and interpret your mosaic results and what guidelines you follow?
So,
that is very tricky kind.
Yes, yes. So we have guidelines, the ASHRAE guidelines and PGDIS, which came recently, PGDIS in 2021 and ASHRAE guidelines for these low mosaic. Not only the guidelines, these guidelines were made after certain trials and studies that has been done, following up till the birth of the babies with mosaics. High mosaics, we don't have evidence and no guidelines for that to be transferred. But low mosaic, with the research and trials that has been done, and based on this, the guidelines say that we can go ahead. And with my experience, at least I think 6 or 7 cases where I have done post-test counseling for these low mosaics, and they have healthy babies and successful pregnancies. Very less percentage which led to miscarriage, but I have seen successful pregnancies also.
So Dr. Bratati, then does embryo biopsy play a role on mosaicism?
It may, yes. That is one of the factors that can lead, yes, yes. There are publications on that.
So the question now to Dr. Vandana would be, what is the take-up of couples for mosaic embryos?
Exactly.
No, but do they want to go for the next cycle or?
Till date, we have not transferred a lower level mosaic also.
Even low level,
we prefer transferring euploid. We tell patients to opt for a repeat stimulation instead. Yeah. Because we need to take a consent. They need to sign it that they refuse repeat stimulation. Okay. That's when we can transfer a low level mosaic. Yeah,
Upasana, sorry.
Yeah. This question is for Dr. Verika, ma'am. Actually, just now we spoke about the BRCA variants for, these are all adult onset disease. So since you spoke about the ethical dilemma for genetic disease and diagnosis, what is your view? Because how much ever the high penetrance variant we take up, it is nowhere, especially apart from some few genes like STK11 or something like that. None of the cancer syndromes mostly has 100% penetrance, right? So correct me if I'm wrong here. So for these patients where they can live up to maybe 25 years or 30 years, and if they can be detected earlier in their screening, will you still offer a PGT for them and choose? Again, Suraj mentioned a very important point that the traits are being chosen, later on we can do. And are we not restricting? Are we not going ahead the eugenics era?
Upasana, the first thing which I would tell is, I think the whole counseling part of it for the family should be very, very accurate. They should be known mutations. They should understand how these mutations have come about. We are talking about germline mutations, and we will have to tell them that there is a possibility that it can get inherited. Now, coming to your next point, whether this should serve as a screening for the subsequent generations, either by PGT-M or by prenatal diagnosis, whatever it is. I would leave the choice to the couple, or to the mother. It's a difficult situation, does she really want this to go on to the next generation or not? But if the couple wishes, or if the mother wishes that it should be screened, I will still offer her the
That's fine. I think
So that is where the autonomy comes. We are taking away the autonomy. We have to offer them at least offer them.
Offer it.
Standard point, yes.
Yeah, that's fine. Okay, any more questions from the audience? Just one question.
Just to clarify,
the last candidate over there, the last row, somebody's raising the hand, please. Yeah.
Until then, just to clarify regarding the donor, see what happens in US is egg bank is having it's a centralized banking where they have the entire carrier screening of all the donors and frozen oocytes. So here the problem is the patient has to pay every time for the donor screening. Because we need to match if that comes any risk, again we need to select another donor. So they keep paying again and again. So that is the only
It's very expensive. Please carry on quickly your question.
BGTSR has been offered for the polymorphic.
Louder, please.
No. All right. PGTSR is only for clinically significant rearrangement identified, one of the partner. Also to add to, it's a question for Dr. Vandana. So don't you think these donor banks, they should include these carrier screening? Is that, because I do see some reports from Mumbai where the carrier screening report is also shared with us. And there are 2-3 donor reports that are shared and the recipient male partner. So is it for all the donor banks? Okay. So,
One second.
So can you request these banks, like since you will be asking for the donors to include this, I think this will be very helpful for the couples going for donor?
Yeah, I think if there is a lot of, that's all, that will be coming later, for all answers will be, all the questions will be answered, but anything. I want to ask Juliana to,
One last question.
I'll ask Juliana, and you have anything to talk to us or tell us about?
Also,
you heard the conversations here.
And also with a question, Juliana, to add to, you have some experience in Dubai and Malaysia and also Hong Kong. How are the regulations with PGT in these countries, especially in some Gulf countries? Prenatal abortion is not permitted by law. So how are these regulated in these countries? Could you quickly comment on? Thank you.
Hi. Just to address the earlier question. So for couples before that were seen in IVF centers, most of the cases that refer to me, they do conduct expanded carrier screening, even for the couples, especially for the egg donor cases. And most of the cases that refer to me, I've covered mainly in Malaysia, Singapore, and some referred from Australia because they came to Malaysia for egg donor conception in IVF. So I think like Singapore for PGT, they are only allowed for families that are affected with genetic disorders and not to couples who are not having a family history. So there are some restrictions there. That's why most of the couples actually came over to Malaysia to have egg donors. And same for couples in Australia, most of them are of Chinese descent. And because there are not a lot of egg donors in Australia, so most of the patients were actually seeking for fertility services in Malaysia as well.
Yeah. Thank you. That's fine. Thank you, Juliana.
Thank you, Juliana.
It was a pleasure to chair this session along with Dr. Khan here and the lovely ladies here speaking, all 4 ladies here today as our speakers. All these days it was mixed, and I think all the exam going students here, the genetic counselors, have got the latest information, because you must have paid attention to all the talks here. You are updated with what's happening all over the world. and we all have learned. And one thing I want to say is, Dr. Madhavi, your enthusiasm, eagerness to see that all these things are eliminated is good, but it will not happen.
No, that's
is
not
nature's
possible, ma'am.
way.
We just want to reduce.
Just
We cannot eliminate. There's no extermination here.
nothing will happen.
Yeah,
I'm just saying, there are so many treatments also available after that. So, and then the mindset is totally different across the world. Billions of people, you think so many billions are being born, but why all these tests and all? And this is the miracle, okay? It is such a miracle. Nobody understands that this is a miracle. They take everything for granted because so many billions of people are there. But when you go into the science of it, exactly, see, you end up with so many questions. We need guidelines to do that, to do this. And it is, whatever possible, always keep in mind that the minds are different, though the bodies are the same all over the world, and each mind thinks in a different way. So as a genetic counselor, when you are going and telling them the problem, don't think that their mindset is same as yours. You may think that, oh, this is such a lethal thing, I have to test for it. But the couple may not feel like that. There's so many practical examples Dr. Veronica also was talking about in her thing. They might say it's okay, and some of them believe that this God has given us like this. And I have seen so many patients which surprised me, and I was shocked, and I learned from the patients how to respect their feelings. Okay? So they say it's okay, and they should fully understand. They go through the pain, the child dies, they are okay with that, and they are happy and satisfied. On the other hand, we should not force that this is abnormal and remove it. And there will be so many doubts, not only in her mind, in so many of her family members' minds. So keep all this in your mind when you're counseling. Respect the sentiment of the person or the couple or the people whom you're counseling. Okay, you're loaded with knowledge. That's okay. But you should know where to put your limitation when you're counseling them and telling them. This is what I want to say. But you must keep updated with what is happening latest, recent, and offer them. This is what it is. Okay? Thank you so much. I thank all the speakers and the moderators.
Ma'am, I request you to please give the felicitate our speakers, ma'am, with the Lakshmi ma'am. Yeah.
With the speakers here, especially Dr. Vandana. Her mother-in-law is a very senior gynecologist from Hyderabad, Dr. Hegde, with whom I had the honor of working with her, along with Annie also was there with us. So, and Dr. Veronica says, the first BGCI examination she attended and she passed through. I'm so happy to see all of you all. Thank you.
She's good. Dr. Bhatati? Yeah. I'll ask her to stay for a photograph. Ma'am, please stay for a photograph. I request Dr. Zeena Chauhan to come on to stage to facilitate our chairpersons, please. Juliana, are you still online? Can you please stay online for a photograph? Okay. No, I just saw her online. She was just online.
Oh, she's left now. Yes, come.
Thank you, Dr. Juliana, to stay online. We can all break for lunch. We'll send you the group photograph.
Good afternoon, everyone. I hope you enjoyed a refreshing lunch break. We are ready to drive back into our session with renewed energy. So, myself, Abhinika, certified genetic counselor.
I'm Shruti, board certified level 2 genetic counselor.
In this session, we are discuss about rare disorders in pediatric and adult. To chair the session, I am requesting Dr. Ajay Kammanahalli and to co-chair the session, Mr. Suraj. I'm happy to introduce Mr. Dr. Ajay Kammanahalli. He is an assistant professor in Amity University, genetic health consultant and counselor in Sadar Hospital, and he is a part of National Health Mission, Jharkhand. His academic includes PhD in biomedical engineering from University of Helsinki and another PhD degree from University of California, Berkeley in clinical genetics. His postdoc in MDR cancer from Institute of Curie, Paris, another postdoc in clinical genetics from University of California, and another postdoc in synthetic virus from Technical University Delft, Netherlands. His career highlights include he is a member of WHO Task Force in COVID-19, and he developed therapeutics for pneumothorax disease, synthetic cells, and viruses. He has several publications, includes Journal with IF 132, and his area of interest is public health, diagnostic, and sickle cell anemia. We are welcoming you, sir.
I'm happy to introduce my dear senior, Mr. Suraj. So he is a genetic counselor at KK Women's and Children's Hospital, Singapore. He is a board certified level 2 genetic counselor. He was previously associated with Apollo Cancer Hospital, Chennai. He was also been associated with reputed Germany-based genomics company. He is also associated with MDCRC, where he was leading the genetic counseling team under the State Planning Commission, National Health Mission Project. Welcome you, Mr. Suraj. Now I hand over to the chairperson to introduce the first speaker of the session.
The mic is on? Yes. It gives me a great pleasure to introduce Dr. Arijit Mukhopadhyay, Associate Professor in Precision Health from University of Salford, United Kingdom. His academic credentials goes PhD in Genetics from CSIR, IICB, Kolkata. He is a Marie Curie postdoctoral fellow in Human Genetics, Netherlands. And career highlights goes a co-lead the University of Salford submission towards Research Excellence Framework. I will skip this. So please welcome you. And he's talking about improving health equity by storytelling in rare diseases.
Can I have this mic? Thank you. And thank you to Annie ma'am, Nancy, and the organizing committee for having me here. I've been learning a lot through the past 2 days. So as introduced, I am currently in University of Salford, which is in Manchester. If you like and follow international sport, I live 10 minutes from Old Trafford Stadium. So for me, a football match means a traffic nightmare. It's not a good news. So, and this gorgeous building you see, that's the main building of the university, and I'm very happy that my office is in this building. So I enter this building almost every day. And for those who are interested, that would take you to my LinkedIn page. But what I'll talk to you today is not what I'm doing in the UK. I'll talk to you today as an Indian story, an Indian family, an Indian case, and how I have tried a little bit of my capacity to change lives of common people that live in our country. Before I get into that, just
few basics and what drives my motivation. So all of us, whether we are a geneticist like myself, or you are a genetic counselor, or you are a clinician using genetics for diagnosis and treatment, all of us are trying to do one thing. That is, how does the pointer work? Sorry. That is we are trying to relate genotype to phenotype, G2P. Okay, thank you. All of us are trying to do G2P, genotype to phenotype, right? We are trying to relate genotype to phenotype. And a phenotype is an overall outcome of multiple layers of information, DNA being the most stable and first layer of information. So DNA encodes the information, RNA and protein decodes the information, and everything put together, not in isolation, using environment and cues and effects, results into a phenotype. So you will be able to only do a direct genotype to phenotype correlation when the DNA variations alone will be able to explain it. In other words, in technical terms, when you are dealing with high penetrance mutations, right? So you have heard a lot from Thangaraj and Vinod and all those people for the past 2 days, and you have heard about what genomes can do, genomics can do, and all that. So I work on all these layers of information. So I work on DNA, I work on RNA, I work on protein, I work on phenotype, I work on environment as well. And I use all those toys that you heard about, right? All the fancy toys that you can have in the lab, I use all of them. But today, I'm not going to show off my academic scientific achievements and credentials. I'm going to use a very simple science, how that can change, make a real social impact, and can change somebody's life using social aspects into account and using genetic counseling as one of the features, right? So I'll work with this layer of information, how that relates to phenotype, so a high penetrating mutation. So scientifically, it is a very simple case study. So the focus is on a disease called glaucoma. Just to highlight that I'm using glaucoma as a representative example, right? Just for the storytelling, because that's the story is about, but this is applicable to a large number of genetic diseases, what I'm going to talk about. So a little bit about the disease. Glaucoma is a disease of the eye. You may have heard about it, and that people go blind. It's a neurodegenerative disease. This is a neurodegenerative disease that causes loss of retinal ganglion cells, which are at the back of the eye. So the back of the eye is a retina, and the retina has 9 layers of different types of cells. All of them are different types of neuronal cells. And the last layer of those cells are called ganglion cells, and those are the cells that take the visual input to the brain. And that's how you know what you are looking at. Rest of it is just a camera. Without the ganglion cell sending the signals to the brain, you cannot make sense of what you are seeing, right? So those are the cells that die in glaucoma. But the problem is actually in the front of the eye, where the light enters. In front of the eye, a major subset, is not 100%, but major subset of glaucoma, the front of the eye in this corner, there is a blockage, so liquid cannot come out. That increases the pressure, and as it increases the pressure here, the lens is pushed backwards, and because there's a viscous jelly inside, that push gets transferred to the retina, and because the ganglion cells are the first layer from inside, they die. And that's the neuronal loss, and that's why you lose vision. So you see everything, camera works fine, but camera doesn't make sense of anything you are seeing. So that's the disease we are talking about. So what happens if this is what you are seeing, a beautiful day on a beach? A person with glaucoma will see peripheral sight diminishing. And then finally, so this is called a tunnel vision, and then finally they will go blind. Okay, that's what happens in the disease. If you have heard about glaucoma, if you knew about glaucoma, and you might be wondering that this is a common disease, why I'm talking about in the rare disease session, right? The reason, so yes, you are right, glaucoma is a common disease. It affects 2% of any population in the world, about 2%. So currently the estimate is that 105 million people are affected in the world. And there are primary and secondary forms, and the primary forms are mostly genetic, secondary forms are non-genetic. And in the primary form, there is this one subtype. You don't have to worry about the acronyms. There's one subtype that is the 50% of the total causes, right? They're called primary open angle glaucoma. And that has another subtype called juvenile open angle glaucoma, right? That is Mendelian and a rare disorder and affects children. That's the one I'm going to focus on. That's the subtype I'm going to focus on. So this is the largest cause of irreversible blindness. Why I specify irreversible blindness? So overall, the largest cause of blindness is cataract, right? But that is reversible. You can surgically operate. Glaucoma, you do surgery, but whatever they lost doesn't come back. It's a neuronal loss. So irreversible blindness, this is the largest cause in the world. So as I said, this is a complex disease, has a complex genetics, and as all complex diseases, this is common disease, not a rare disease. But like every other common complex disease, it has a Mendelian component. There's a subset of the disease that works as pure monogenic forms. So for glaucoma, these are the two subsets. One, the monogenic side of it, classically as all monogenic diseases, genes were found by linkage analysis. And these are the few genes that are known. I have published on all these genes, and there are association studies like GWAS and all that. You do that. We have also done the first GWAS on glaucoma in India. Mansi has done her PhD on that, so who is now a genetic counselor. So that's Mansi's PhD project. So that we have done that. Scientifically, something intrigues me, which I am not able to find out an answer yet. For not a single disease that I am aware of, glaucoma included, the genes that you find from the Mendelian component never comes up in a GWAS. And genes that you find in GWAS never comes up as a monogenic disease, right? I don't understand why, but that's a fact for all diseases. So which means you can look at each one of them as a rare disease. Okay, and that's what I'm going to show you, what we did. So this particular gene, myocilin, that was highlighted in the previous slide, that is so far the largest mutated gene in juvenile open angle glaucoma. What is largest? Largest is about 5%. If it sounds small to you, that is higher than what BRCA1 and BRCA2 combined does for breast cancer, right? Why do you don't know about this? Because we don't have an Angelina Jolie for glaucoma, right? We have an Angelina for breast cancer. So 5% is small, but it is large, given the genetic complexities we have. So this particular gene has 3 exons. Even this gene is by itself is a very mysterious molecule, because if you have mutations in exon 3, that always end up giving you a Mendelian form of the disease. If you have mutations in exon 1, that always gives you a complex form of the disease, right? Again, we don't understand why. I published papers on the structure of the protein, why can that be? But frankly, I don't know the reason. And what we have published in the past, that only 20 nucleotides in this gene, only 20 nucleotides, 11 in this band and 9 on this band, they account for 30% of all the mutations. They're like mutational hotspots. And one of those mutations is what I'm going to talk about. So this is the mutation, right? That's C there, that's a wild type variant, which we found to be a T, right? I am deliberately putting up this very dry, boring stretch of sequences to highlight that as a lab researcher who is not a genetic counselor, this is what we see, is devoid, stripped off of the human face, right? We see either in spreadsheets, if I'm having multiple individuals, or for one individual analyzing, this is what we see, is devoid of any emotional connect. That is important. That is important to maintain the unbiased, objective, scientific aspect of it. But you never have that emotional connect if you are not doing a genetic counseling, right? So we found this mutation in a family. I used to go to the clinic, talk to the doctor, sometimes meet the patients. So these patients I met. So this is how that person's eye looks like. If you don't know how to look at eye disease, probably you don't. I learned from clinicians, so I'm also did not know before. So this area here, there is a thinning of the optic cup, it is called. the thinning indicates the neurons are dying off, right? In healthy eye, this yellowish layer will be much thicker. Okay, this indicates that significant cupping. That means the ganglion cells are mostly gone, right? And mutation, so the objective signs without the human face. This was the heterozygous mutation, right? It's a C2T change, as I already told you, and that was in exon 3. It's a proline to leucine change in one of the amino acids, in one of those two bands that I showed you in the gene. And we also developed a simple RFLP assay to quickly and cheaply screen for this mutation. But this is a mutational hotspot, right? This happens very, very commonly. It's a rare disease, but within that rare community, it's a very prevalent mutation all over the world, not only in India. So after we saw the patient and we found the mutation, we went back to the family, right? So in this audience, I don't have to tell you that it's an autosomal dominant inheritance, classical. Right, a little bit phenotype. So this is the proband, right? You know how to read a pedigree. And I want to highlight that at this stage, no genetic counselor was involved. This was in 2002, 22 years ago, right? No GC was involved. So we found this. I traveled to the village. It's a rural family from West Bengal. I traveled to the village with a doctor, with a phlebotomist. We collected blood sample from few individuals and we confirmed that everybody who was blind had the mutation, right? As you could imagine. So it's 100% penetrance in this particular family for that particular mutation. And whoever had it was blind by the time they were 18 years old. They did not have any problem until they were 12. From 12 to 14, they started fall behind in school. Then they stopped going to school and by 18, they were blind. This is so. Imagine this scenario, it's a very poor family. Child is happy, healthy, going to school, playing around. From 12-year-old, they start having problem. And in 5 years, their life is completely robbed of them. So this is the family which I saw. And another thing I realized after attending that family, that because they live in close quarters, and because they see so many people having the disease, they think it's an infectious disease, right? So the husband ran away, right? So this poor woman, when she came to us, she was blind, her middle child was already blind, and she was worried that the eldest child, who was 18, 19 years at the time, was the only earning member of that unit of the family, will go blind, and because they had no way to predict, and then they will starve to death or they will beg on the road. So this was the situation. First thing we were able to tell her that this son will not go blind because of this mutation. Okay? Of course, there's no GC. I did that. I don't know how accurately I did it, but that's the best I could do 22 years ago. Okay? But we also found out that this particular son and then two other children in the family who were below the age of onset, they had the mutation, right? And hence my struggle began to convince the other stakeholders that please intervene them to save their sight. Nothing I could do. So 2002 is the discovery. I talked about a lot, published multiple papers, spoke to many conferences, got even awards, but I could not do anything. I waited and saw those children going blind, as you would imagine. They grew older and they went blind. Then something changed last year, after 21 years. And that's what I want to highlight for the rest of the talk, and which finally created the impact. So until last year, I was using the lens of an objective scientist. Right? Objective data scientist talks with data, talk to other scientists, try to convince that you do something, nothing worked. Then we went back to the village again in 2023. So that's the real house they live in. I'll not show you the name or face of the patient because of confidentiality. That's the house they live in. So imagine, and this is 2 hours away from the nearest hospital by bus, right? So imagine a blind person, how does she get the right bus, gets the right stop, goes to the hospital, comes back. And if one of the member of the family who can see accompanies them, they lose that day's earning, right? So what is the result? The result is they don't go for follow-up, they don't go to hospital, right? Health inequity. The health equity is completely not there. They're not able to access, even if we can tell them the genetic mutation. There's nothing going to change in their life. So what we did, I managed to find some money from the university, so that's me. Last year, I managed to find some money to do something about it, to travel to the village. So I took a genetic counselor, a doctor, to that village, and what you see me doing, we are developing a script to tell a story through a film. Right? We developed the script with the family. And then we came back and modified the script with my students back in the university. Right? So I tried to include all stakeholders, including a genetic counselor. So I did that. I found a team who makes films professionally, and thankful to my university who funded this whole project. And then I made the film. The film is called The Vision of the Blind Lady. Right? Some of you have seen the film, who's in the audience. I'll encourage you to please go and see the film. It's only 25 minutes long. That's the link. It's on free, available on YouTube. And I have also given a TED talk recently about this film and how it is changing society. And that's the link for the TED talk. Both are freely available on YouTube. I'm happy to share the slides for later on. So this is what I did, and that changed things in the last 8 to 10 months, which I never imagined it is possible. What I'll do, I hope I'm doing okay with time, I'll play the trailer for the film to give you a flavor, which is 2 minutes long, right? So I'll shut up for 2 minutes and let you watch the trailer. Glaucoma can take away your vision silently. Worldwide, up to 100 million people are affected with this disease. I tell you the story of a family that I met in Calcutta Medical College 20 years ago. তুমি কি আমায় দেখতে পাচ্ছ? ওই একটু বেশি অন্ধকার. Did not know how to read or write, but she was one of the most aware and educated patients I have ever come across. Madam, আমি এদের কেসটা দেখলাম. For a detailed survey, আমি এদের গ্রামে যাব. আমাদের গ্রামের আর কোনো ঘরে চোখের এই সমস্যা রয়েছে? না, শুধু আমার মায়ের দিকের ফ্যামিলি। আমজাদ, তোমাদের গ্রাম খুব সুন্দর। আমি আবার আসবো। আজ আসি। স্যার, আমার নজর কবে যাবে? I was in agony. After we recorded the family history, we were certain that genetics is the cause of glaucoma in this family. একজন তো অলরেডি গ্লুকোমা এফেক্টেড, তাকে তো ওষুধ দেওয়া হচ্ছে। আমাদের ফাইন্ডিং অনুযায়ী কিন্তু আরেকজন প্রি-সিমটোম্যাটিক, মানে ওদের ভবিষ্যতে তার গ্লুকোমা হবে। তাকে কি আগে থেকেই কোনো ওষুধ দেওয়া যায় না? রোগ ছাড়া আমি তো কোনো মেডিসিন দিতে পারবো না। ক্লিনিক্যালি it is not ethical to start treatment before someone shows symptoms of the disease. এই মিউটেশন থাকার দরুন এই ছেলেটির আগামী 3-4 বছরের মধ্যে গ্লুকোমা হবে, এবং তখন যদি ট্রিটমেন্ট না হয় তাহলে মায়ের মতো ও বন্ধ হয়ে যাবে। We wanted the doctors to start treating these children, which was not ethically possible. So today I pose a question. I ask the medical professionals, the researchers and the public, should we revisit the ethical boundaries? আমাদের তো বাচ্চাদের জানার না, কিন্তু আল্লাহ দিনটাকে ধরে রাখতে পারবেন না।
In precisely 30 minutes you did. If there's some more, can yes conclude.
So that's what we did, and then we had a screening in Kolkata and 2-3 screenings in Delhi, and then we put it on YouTube. And first thing that changed, Emily Banerjee, who many of you know, one of your trained counselors, she was in the Kolkata screening event, and Dr. Oshim Shil is an ophthalmologist, eminent ophthalmologist, glaucoma specialist in Kolkata. He was also on the screening, and both of them also we consulted for making the film. We got them as experts. They got emotionally motivated, the first 2 people. And they offered on the stage that they are offering their services for this family for as long as it takes. First thing that happened. Rotary was also present in the venue. So Rotary offered to make it a lifelong program. So now this has become a program under Rotary. Only thing I did differently was I left the objective scientific community and I told it through a story. I made it fictionalized. so that I am not breaching any confidentiality, but I'm still telling the story. So that's the novelty on this. Scientifically, there is no novelty. So we went to the village. Emily had done pre-test counseling. Then we did clinical evaluation after they consented. And imagine this was not a trivial counseling. No counseling is trivial, but this was even more complicated because these were blind, illiterate, uneducated family. Right? So it's a pretty challenging job. She did a great job. Then Asim Sheel has evaluated them. And then I have two of my juniors who are now respected scientists themselves, Moinak and Molinath, one in Calcutta University, one is NIBMG in West Bengal. They took up the genetic testing, post-clinical evaluation. Then based on the mutation, Emily again did counseling. We traveled to the village, I was with them, counseling, and then if they consented, we did surgery for those who carried the mutation. Okay, this is what he done last year, last August, September, that time around. So these are some of the views. You can see we are in the village. You can see the Rotary placard, banner, and then you can see the doctor. This is happening in their house in the village. The doctor is testing. Emily was counseling on the side in another room, and then whoever consulted is happening in the eye surgery happening in the eye hospital, all free of charge from their point of view, but otherwise funded by Rotary. The best thing is I am not involved. It is an autopilot mode. That's what I love about it the most. People got motivated and they started taking their own initiative. I don't have to do anything anymore. So I showed you the family. Now I'm shorting it out. Remember these three children who had the mutation, we could not do anything for them. They are now grown up 20 years later. So as you could imagine, they are all blind now. But they have their children. Sorry. Yeah, they have their children. And now these three children next slide, just click next. So now, of course, with GC involved, that is happening. So they have these three children. These three, I'm not showing the entire pedigree, but these three carry the mutation. Right? So now, with GC involved, with the doctor involved, with this film in the background, now we are able to change life as the impact of the storytelling. How we are changing life? This is one example of the three. So again, if you are familiar with eye diseases and glaucoma in particular, there is a small bleb here, the inflammation here. That's the surgery, right? That's the surgery. So this is a 13-year-old boy, one of those children. So again, I remind you, age of onset is about 12, 13. They go blind by 18. So this boy just started showing an elevated intraocular pressure, no vision loss yet. So at that point, he was surgically intervened in last August. Medical ethics dictates that we have to follow this boy up for 2 years, which we will do. But for now, 6 months, for every month follow-up, Asim Sheel, the clinician, is very confident that we have saved blindness of these three children. Right? So this is the impact. Thank you. This is the impact of storytelling. Right? Science is very, very simple. Right? How the science is communicated, that what made the difference. Right? That's what I want to highlight. And this is important because this is a health economic study that came out. This is in the US. The reference is at the bottom, where they showed that the cost of delayed diagnosis per patient on average is $220,000 in one lifetime. That's the cost of delayed diagnosis. That's what you can save. And every such saving does not need whole genome sequencing. Right? So I wanted to highlight that I do whole genome sequencing, believe me, I love it as a basic scientist, but it is not needed in every case, right? So actionable genomics is different from predictive genomics. You don't hit everything with a hammer just because you have a hammer, right? You use whole genome sequencing when everything else fails, right? If you can do something by one RFLP, you don't need to do whole genome sequencing. So actionable genomics is what I am interested in. And we should have different strategy for different levels of genetic involvements. We don't need genome screens for everything. Right? We can do it. That means we have multiple tools in our box. Which tool you use when depends on the situation. Depends on what is the time scale I need to make a real impact on the patient and family. That should be the decider, not the technology. Right? So that's what I think. So guys, summarizing, I'm bringing you back with, to my title, the subjective storytelling with objective genetic data can improve health equity in rare diseases. So it is a reality, it is possible. We are trying to scale it up now and to approach many families. But bringing you back, why this happened? Right? This happened because something different happens to your brain when you listen to stories. Right? You feel things. Logic doesn't make you feel. The part of the brain that understands logic doesn't understand emotion. You take action when you feel, not when you understand. You first have to feel, then you'll be motivated to understand, then you will be motivated to take action, right? That's why I wanted to tell a story, right? I want to make you feel first so that you are motivated to take action. And this is because stories are data with a soul. So you need to put soul into the data to make a real impact for people. So these are the people who made it possible. You have seen already Oshim Shil and Emily Banerjee, Moinak Sengupta and Molinath Acharya are the genetic partners in this journey. Kunal Ray has been my PhD mentors as well as PhD mentors of these two guys. So he was the person who led all these projects 20 years ago. I want to highlight these two people here. They own a company called 7th Art Communication. So Dr. Rumpa Gui is not a medical or genetic doctor. She is a doctor of filmmaking, right? So she and they specialize on making documentary films for public awareness. So we partnered with them in this journey. And of course, funding for the film came from Salford University, my university. And in India, a lot of logistics help is provided to me by my alma mater, IGIB. And Premas has been a long time friend and collaborator that helping me. That's it. That's the end of my talk. Thank you very much.
Wonderful presentation and inspiring story with the collaboration, Dr. Arijit. In fact, genetic counselors try to present information as an education model in such stories to convey the information. Believe me or not, I have to confess, I was in class 7 when you drew that pedigree, and that was more than 3 generations. Thank you so much for that inspiring talk. We would request you to wait for the panel discussion later. Without much ado, I would like to introduce the next speaker to you all. It is my pleasure to introduce Dr. P. Govindaraj. He is the Assistant Professor at the Department of Nephrology, National Institute of Mental Health and Neuroscience, Bangalore. And he has done his PhD in Biomedical Science at the Bharatidasan University, Tiruchirappalli, and postdoc in NIMHANS, Bangalore. And he has additional qualifications as a Secretary of the Society of Mitochondrial Research and Medicine. And his career highlights are that he has published several research articles, CSIR, DBT, and SERB fellowship for PhD and postdoctoral work, and he has got Young Researcher Award from SMRM India in 2012, and his research focuses on understanding the mitochondrial dysfunction in human health and disease, in particular with a specific aim to explore the new genes associated with mitochondrial disorders, understand the molecular mechanism, and develop therapeutic diagnostics. Over to you, sir.
Okay. So, good afternoon. Thanks for the nice introduction. First, I would like to thank the organizers for inviting me to share our work. So, today I will be talking about the mitochondrial disorders in the omics era. Currently, where do we stand? With some work which we are doing for the last 2 decades. So, going back to the history of mitochondrial medicine, this gentleman has the first lived in 1962. He has reported that Swedish woman with hypermetabolic disorder is due to mitochondrial dysfunction. He explained this case report with clinical, biochemical, and morphological evidence. Later, in 1970s to 80s, the field has the basic mitochondrial functions has been described. And in addition to this, in 1981, the first complete mitochondrial genome was sequenced, and the field got ignited after the first identification of mitochondrial DNA mutation in LHOM following this, in 1990s, we thought that these mitochondrial disorders are only due to mitochondrial DNA mutations. But there are several nuclear encoded mitochondrial proteins also involved to be the cause for the disease. If you see the graph here, the mitochondrial, the publications are started moving after this discovery. And later, there are few neuroscientists, these gentlemen, they have identified several phenotypic classifications of mitochondrial diseases. So, mitochondrial diseases are just, it's a clinically heterogeneous and genetically, which can be characterized by mitochondrial dysfunction, and is one of the largest class of inborn error of metabolism, and it is caused either due to mutation in nuclear DNA or mitochondrial DNA, and the onset of the disease starts from infancy to adult onset. And then either it can include, as I mean, like it can be a single or multiple organ involvement, and further the dominance, I mean the pattern of inheritance would be dominant, recessive, and maternal mode. In addition to this, because of this phenotypic changes, and there is, it's not moving. Okay, so it leads to lots of clinical heterogeneity. If you see here, these are all some of the photographs which I have given. This is only stroke-like episodes where you can see the phenotype of MELAS patient, and this is adult onset CPO where you can see ptosis in the eyes, and Leak's disease where you can see the hypertonia, hypertrichosis, and albinetic fundus. And there are some kind of a classification related to complex 1 disorders, which I will be coming in the subsequent slides. The problem with there are several non-syndromic groups in these mitochondrial disorders, which makes a complex in diagnosis. And currently, if you know, there are 400 genes have been identified, but still more than 50% still we don't know what is the diagnosis for these patients. So this leads to poor genetic, I mean, poor genotype-phenotype correlation. So if you see, the epidemiology of this particular disease is like in across the world is 1 in 4,000 to 5,000 individual, but this changes from the population to population. But however, in India, if you see, there are like exactly we don't know what is the prevalence of disease. But based on our experience, what we feel is that the prevalence will be more when compared to the rest of the population, because still few populations are maintaining the endogamy. That could be one of the reasons where we can see some kind of a recessive pattern in this mitochondrial disease. So once these are all the complete, evaluations needs to be done. But the point I want to stress is there is no single test has a capacity to provide a definite diagnosis. So in such case, we need to see based on the phenotype, and we need to go for completely whichever is required. It requires comprehensive investigations. So coming back to this, what is the current status of this omic technology in the mitochondrial medicine? As we all know that genomics has played a vital role in identifying the causative genes, and many number of novel genes have been identified. But the role of the particular variant still was not established when it comes to the clinical setting. So now this omics technology has come into the picture where it can complement the genomic findings or the genetic causes, and it can generate some kind of data where it can lead to improve the patient health, and it can particularly target the insight of the mitochondria, and further it can improve the diagnostics and the therapeutics, which can lead to personalized medicine. So end of my talk, I'm going to give some kind of the glance what so far have been available in this omics tools. Yeah. So as I said, last 2 decades we are working in this particular disease, where almost more than 1,500 samples have been worked on. And if you see here, the prevalence of this syndromic groups are like LHON is predominant with 25%, followed by CPO, Leak's disease, then MELAS, MOF, KSS, and remaining 43% are non-syndromic groups, which cannot be put it into either which syndrome types. So this makes a complete, which requires the complete evaluation with respect to the clinics, clinical side or the genetics. So usually the first line of evidence is to say biochemically whether these are all the mitochondrial is lactate, pyruvate and CPK. But if you see here, not all the patients showing the elevated lactate, and of course here also there are few group for patients showing normal, likewise CPA. So, but these complete cases are all showing the classical mitochondrial signatures on clinical characterization. So why do they differ? Because it depends on this, when it comes to specificity and sensitivity, all the particular syndromic groups may not fall into the biochemical parameters. And in addition to this, in humans we do histopathological stains where we look into the muscle pathology, and this is the RRF, that is rag red fibers, there is subsummal accumulation of mitochondria. This is the golden standard stain which is used for diagnosing mitochondrial disorders, and followed by like rag blue fibers and COX deficient fibers. In this data also, if you see, more than 50% of the patient is not diagnosed. So there is something behind this. Then in addition to this, to complement the histopathological findings where the patients may fail to show the abnormality, in humans we do this respiratory chain complex assay for this mitochondrial patients, where we'll be isolating mitochondria from the muscle and we'll be seeing the complex activity, and based on this we'll have like less than 20% with CETUB deficiency. So accordingly we'll give a report, and finally this will be helpful for taking to the genomic study. And if you see here also, there are like several phenotypes are associated with this complex deficiency, and the first thing is complex 1 deficiency is more, and followed by multiple complex deficiency, likewise in adults also. And if you see, there is several overlapping phenotypes are there in the complex 1 or irrespective of the other complex deficiency. So this makes a complex. So far whatever I told, it gives clues us for going for the further genetics workup. And as I said, almost more than 400 genes have been described so far because of this advances in the technology. Particularly, most of the gene discovery has been done after this NGS came into the market. And in this last 2 decades, particularly one and a half decade, you found more genes that could be caused for the mitochondrial disease. So before going into our experience, just I want to highlight there are several salient features of this mitochondrial DNA, but one thing is heteroplasmy. Like usually we'll be talking about heterozygous mutations, that one is inherited from father, another is from mother, but here it is a plasmid. That means the mitochondrial number is different into the cell types and organ or cell to cell types, so that will varies. So here there is like a what you call the phenomena which taking place inside this bottleneck. We don't know how this phenomena is happening or the segregation is happening. If there is a low level of mutation, then you may not be able to see the clinical expression. But if it is a 50% or less than 40 to 50%, you can see the intermediate phenotype. And if it is more than 80%, you can see the patient high level affected offsprings. The point I want to stress here, always the clinical symptoms evolves when it reaches to a certain threshold. Then only we can see the phenotype. Yeah. So this is the data of what we have been, which we have developed the algorithm for analyzing this case, which we have done like a complete mitochondrial genome for tissues as well as the blood DNA, because we always prefer the tissue DNA because it's a post-mitotic tissue where you can see even detect the low level of heteroplasmy in the post-mitotic tissue. But if when you take the blood DNA, there is a chance for selective elimination of the mutant that could not be able to detect even the low level of heteroplasmy. So there could be possibility of missing the variants. So here we identify around 13% across the genome, and also we have identified several hotspot region in our data sets. And further, once you identify a variant, how you can say that it's a causative. So for that, what we are trying currently, we are using is trans mitochondrial hybrid model for characterizing this variant. So I'm just going to show some 3 mutations how we characterize. So this is the trans mitochondrial hybrid for validating the functional characterization for mitochondrial DNA variations. So we'll be growing the patient-derived fibroblast and we'll be removing the nucleus and we'll be fusing it with the parental cell line of 143B where there is a mitochondrial DNA depleted. So that we'll be putting the mitochondrial DNA of the patient to the 143B row 0 and further the selection is done on uridine minus and BRD. So whatever the cells that mitochondrial DNA entered into the 143B row will be the colony will be selected and we again further the mutations will sequence it and later we'll go for the functional characterization. So in order to see how whether this particular mutation is affecting the mitochondrial function, the simple thing is just to see the cell viability and proliferation. Instead of glucose, we'll be supplementing with the galactose media so that usually the supplementing galactose media we cannot directly convert into pyruvate. So it has to depend on, it has to convert to glucose, then it has to convert into pyruvate. That time it will be relying on the mitochondrial respiration. If there is any defect, automatically the cell number will come down, as you see from this bar graph. So this shows that there is a decreased proliferation rate, indicates that there is a reduction in the mitochondrial respiration. Further, using these hybrids, we'll be measuring the mitochondrial respiration using Seahorse technology, where you can able to see here this oxygen consumption rate is reduced in the patient cell line when compared to the wild type of, this is a blue color indicator. So overall, if you see the basal mitochondrial respiration, ATP production and the maximal respiratory capacity is lowered when compared to the control. So this also indicates that there is some problem in the mitochondrial functions, which also supplemented with membrane potential. This is usually, as I said, there are several mitochondrial subunits are present in the inner mitochondrial membrane space. If there is any, because of this mutation, if there is any communication problem, automatically the membrane potential will get affected. So here we can able to see the membrane potential also is reduced. In addition to this, as we all know that the mitochondrial DNA is present near the vicinity of ROS rich environment. If there is any damage, obviously we can expect the ROS production is high. So this also here we can see the patient cell line is ROS is high. So with this line of assays or the functional evidence, which we can tell that mitochondrial DNA variant is possibly pathogenic, how we can, that we need to correlate with the phenotype. Coming to this, one example which I want to give. So this is the phenotype which has a patient is having both like malas and leaks overlap. So if you see here, pathology is like NDP, there's no diagnostic pathology, and Oxford complex also normal. But however, the mitochondrial DNA sequencing has revealed there is a homoplasmic in patient and there is low level in mother and in sister. But one thing is in Sanger sequencing, there is a probability of missing less than 5 to 10% of the mutant. So in such case, we need to go for the NGS technology where you will be requiring to indicate for more coverage. So if you, again we have done NGS and we identified here, if you see, the patient is carrying 100% in blood and tissue, but mother 2.3% and the mild symptomatic sister, she is having 35%. So the same mutation can also lead to different clinical phenotypes. In our case, if you see, this is a malas and some other mitochondrial disease looks like, but it is not clear. But if you see these two patients, one is LHON, another one is only leaks. So this all depends on how much the mitochondrial mutation load is present in the particular patient. So coming to this, as I said, there are more than 1,200 proteins are encoded for mitochondria. So there are several nuclear genes which are also involved in the mitochondrial disorders. So these are all some of the genes which are present inside the mitochondrial and their function. Coming to this, previously we also described the prevalence of poly mutation. This is the polymerase gamma, and in this we have seen that the L304R is more prevalent, and following but surf, it is mainly caused due to the Leaks disease, and here also we have shown in our previous study what are the mutations prevalent. In addition to this, there are some clinical exome panel was developed, around 7,000 genes. In this also we could not able to detect 50% of the causative. However, the further we went and we have done the whole exome sequencing and we identified several mutations. In addition to this, recently also we have described how this primary, I mean, this is the 3243 is the primary mitochondrial DNA mutations which leads to MELAS. Because this 3243 has not only restricted to MELAS, it can also cause cardiomyopathy, hearing impairment, and additional phenotypes. So here, the MELAS symptoms, along with that, there will be additional symptoms. So which we identified, there are several mitochondrial nuclear genes which could be the modifier for this 3243. That was also recently, I mean, published in Journal of Neurology. So based on this, our experience, so now we are like, since the exome cost is more, so what we are trying to do is just trying to identify some set of panel for this mitochondrial disease, particularly in Indian population, using this mission program on rare pediatric genetics, which was funded by DBT. So we are currently in the process of that one. In addition,
sorry, another 2 minutes.
Yeah, I complete. Just another 2 slides. Okay. So this genomics has given the lots of information, but end of the day, when it comes to the interpretation like novel variants or VUS variants, it is really challenging. And in terms of the functional lacking. So in such cases, there is no global data sets available. Mainly it is on like either a cell line or the animal models, but is not replicating to when you go for any particular drug for the patients, because the genetic background is different. In such case, some kind of omics tools will be helpful for identifying some tissue specific manifestations which could be which can contribute to the field of mitochondrial medicine. So with this, 2 studies are available on transcriptomics, where they have done more than 100 samples and they identified one particular, 10% of the cases they identified diagnostic markers, and they also told that the complex 1 assembly factor TIMM-DC1 establishing a novel associated with based on this RNA-seq. Following this, there is another one study which is not yet still published, it is in binary. So they also identified what are all the, particularly the mitochondrial toxicity, vitamin bone, transport metabolism, and having a different expression. In addition to this, this particular study also showed there is a shared enrichment model, like particularly in the Parkinson's disease, epilepsy, and schizophrenia. And few studies, like particularly on metabolomics, also has shown that they identified a multi-biomarker. Of course, this arginine is one of the metabolite which will be reduced in the MELAS group, and they also shared there are some kind of metabolic signatures has overlapped with the primary mitochondriopathies with the inclusion body myositis, and of course this taurine, that is taurine and vitamin D3, there is a decrease in the concentration of this LHO in patients, and which could be the predictive metabolic signatures for the LHO in patients, which can lead to therapeutic targets. So yeah, so this is another 2 biomarker, that is FGF21 and GDF15, where we have analyzed with our patients with having a neuromuscular as a disease control. We could able to see there is a elevated FGF and GDF15. This is a which further together the sensitivity and specificity is high in this OCQ. So this is mainly for sensitive for muscle manifesting mitochondrial disorders versus non-muscle mitochondrial manifesting disorders. So with this, previously the algorithm was followed by this, but now this muscle invasive procedure is reduced because of this, and where the yield of diagnosis is also more. So this is what is helping, and with this, due to time limitation, I'm just skipping off the proteomics work also. So
some time to summarize.
Yeah, just 1 minute. Anyway, skip. So the take-home message is what we follow is correlation of clinical, pathological, biochemical, and genetics should be considered for better diagnosis and treatment. But what I feel is this genomic has, as I said, the genomic has given a lots of information. Something is closed. Okay, so genomic has given the lots of information, but now the step for the omics tools. So now this transcriptomics, metabolomics, and proteomics is trying to answer for few things. But in this context of mitochondrial disease, individual patients is different. So, for example, if you take MELAS, only 3243 is like 80% of the cases will carry it. At least we know that what is the genetic defect. But few other cases, if you see, it is randomly, I mean, the functions of the genes which has mutated is different. In such case, it would be difficult. And also, it is appropriate to choose what kind of tissues which involves to be researched and how that can complement to the genetic findings. With this, yeah, what's up? It's okay. So this is what the take-home message which I want to give, and I like to thank my mentor Dr. Tangaraj, who has introduced me in the field of mitochondrial genomics. Later, I started my research career, and in addition to this, I also acknowledge my NUMANS team and CCND colleagues and Keshav Singh from University of Alabama, and patients and funding agencies like your DST, DBT, and also. Thank you.
Thank you very much.
Thank you so much, Dr. Govindaraj. We always look out for scientists like you, genetic counselors, when we have variants of uncertain significances, because you have wonderfully integrated all the omics to correlate the clinical phenotype. Thank you so much. We would further discuss in the panel discussion later.
Okay, let me introduce the next speaker, Dr. Divya Gupta. She is Assistant Professor, Department of Dermatology at Dr. B.R. Ambedkar Medical College, Bangalore, Consultant in Pediatric Dermatology, Manipal Hospital, Bangalore, Consultant Epidermolysis Bullosa Registry Program, Center of Human Genetics, Bangalore. Her academic credentials scores MBBS from Zipmer, MD from Jipmer and DNB, and career highlights course conveyor, a special interest group, dermatology clinical research, recipient of research grant, senior editor IGDVL, associate editor IGPD, Eric Sankey scholarship from Brussels, Belgium, double scholarship for international post-graduation, executive education program at IIM Bangalore, and she has 15 publication and 23 invited guest speaker. Dr. Divya Gupta.
So at the outset, I would really like to thank the organizers of this conference for having me here today. And I've been listening to all these lectures, wonderful lectures. And so I'm a clinician, okay? I'm a dermatologist, and I definitely do not know as much about genetics and genetic counseling as most of you in this room. But I hope to give you some kind of perspective from the other side, you know, from the clinician side. So as I said, I'm a dermatologist, and my area of interest is pediatric dermatology, specifically inherited disorders of the skin. And the disease that we work at specifically is epidermolysis bullosa. So the scope of my talk today will be fourfold. Just a few quick slides on genetic counseling. I don't think I need to really focus on that. An overview of genodermatosis, how I do my genetic counseling, you know. I mean, definitely, I'm sure I have so much to learn from you all. And the focus would be on what Professor Mukhopadhyay said, you know, actually the genomics is what we are looking at as clinicians rather than predictive genomics. So steps in genetic counseling, you're all, you know, familiar with all this. When a patient or when a child, so mostly I see children with these inherited disorders, and when the family walks into my OPD, okay, my work starts with a detailed history and examination, okay. So we ask about past history of any affected family members, any bad obstetric history, ethnicity, you know, any exposure to any chemicals, toxins, pedigree charting. Again, I'm not going to dwell on that. In this, the most important for me is this, you know, management, okay? As you can see, all of this are in green, but I have highlighted the management in red, because when a child comes or when the parents come, at the end of the consultation, what they ask me is, doctor, you know, can we treat this? You know, do you have something for my child? What do we do after this? So
I'm going to be talking more about that in subsequent slides. So just a quick overview of the common genodermatosis that we see. Now, when I say genodermatosis, see there are other conditions also, like, you have psoriasis, vitiligo, eczema. These disorders are characterized by a polygenic inheritance, or under ACMG criteria, they would come under common complex disorders, okay? This is not what I am talking about. When I talk about genodermatosis, I am talking about disorders which are characterized by single gene inheritance, okay? So that would be, sorry, I think it's going the other way. Yeah, so it would be, it could be epithelial adhesion disorders like epidermolysis bullosa. It could be disorders of keratinization, ichthyosis, pigmentary disorders, primary immunodeficiencies, disorders associated with malignancies or development of various tumors like xeroderma pigmentosum, various familial cancer syndromes, and of course we have various metabolic disorders like porphyria, phenylketonuria, and alkaptonuria. In dermatology, usually we do not come across conditions with chromosomal inheritance. I mean, there are other disorders with chromosomal inheritance, like maybe Down syndrome, which may have a minor dermatological component, okay? But primarily, most of our conditions would be disorders which follow the Mendelian laws of inheritance, okay? So when I take history, it's going the other way. The previous slide, please. Yeah. So when I take history from a clinician's point of view, that's what I'm trying to say, okay? What I look at is, how is it inherited? What is the pattern of inheritance, okay? So again, for me, taking family history is very important so that I can figure out whether it's an autosomal dominant or whether it's an autosomal recessive, whether it's an X-linked dominant, X-linked recessive. Next slide, please. The other thing that I want to look at is the severity. See, there was a discussion on eugenomics in the morning, I think, okay? Everything need not be treated. A child who is afflicted with, say, ichthyosis vulgaris is likely to just have dry skin, okay? I mean, there is no need to even test for that. Even though there is a family history of such a thing, there is no need to test, okay? And the child can get by with just a liberal application of moisturizer throughout his life. Or suppose within epidermolysis bullosa, if someone is suffering with the simplex variant, which is relatively mild, okay, then you can just, so from a clinician's point of view, it's important to ascertain what is the severity of the genodermatosis that we are looking at. On the other hand, something like, say, xeroderma pigmentosum, okay, which is characterized by multiple squamous cell carcinomas or basal cell carcinomas, malignant melanomas, which can significantly shorten the life. Or say even dystrophic epidermolysis bullosa, where you have multiple complications like contractures, difficulty in swallowing. Obviously, there the patient and the family needs a lot of support. Okay, so for me as a clinician, that is important to know. Okay, what is the severity of the genetic condition that I'm dealing with? Next slide, please. Now, this is the most important slide. Okay, now, with science progressing at the pace that it is, okay, we have more and more therapies which are becoming available. Okay, we have drugs targeting cell signaling pathways. We have drugs which target inflammatory mediators. For example, losartan in epidermolysis bullosa. We have protein replacement therapies. We have translational read-through inducing drugs like gentamicin. And we also even have gene therapy, okay? For those of you who follow these developments, last year gene therapy was FDA approved for dystrophic epidermolysis bullosa in the US, okay? And patients who come to you, they are aware of it, okay? They will come and ask you, can this be used for me or can this be used for my child? Next slide, please. Yeah, so this is the genetic counseling as I do it, okay? I know this is probably not perfect, but so my counseling with patients starts with, as I mentioned, history and examination, okay? Next slide, please. Next. Yeah, so these are the things that I look at when I am examining a proband, okay? I look at the skin, nail, hair, mucosa, teeth, okay? And these are the other tests that I order. Previous slide, please. Previous. Okay, so
After a detailed clinical history and examination, you sort of know that, okay, this is likely to be a genodermatosis and this is going to require further testing, okay? So then I sort of decide that which test I'm going to order, whether I'm going to order a targeted panel or a clinical exome or a whole exome, okay? Now, conditions like epidermolysis bullosa or xeroderma pigmentosum, they are very well characterized phenotypically, okay? So a targeted panel of 22 genes is enough to diagnose epidermolysis bullosa. You don't need to do a West over there. But say something like, where you have, say, disorders of pigmentation, or somebody presents to you just with cafe-au-lait macules, okay? There could be so many things, okay? So there you would want as much information as possible. So there you would likely go for a WES. So here what I'm trying to highlight is that for us as clinicians, it is important to know the basics of genetics, the, inheritance patterns, and that science is important for us to know, okay? Now, once I have decided which genetic test I'm going to do, previous slide, please. Yeah. These are the things that I'm going to tell the patient, okay? What is it that they want to know? They want to know how much it is going to cost, okay? What sample are we going to send? Are we going to send blood? Are we going to send a, saliva sample? Are we going to send a biopsy, okay? How many people have to be tested? Is it just the proband? Are we going to do a trio testing? Any other member of the family who's affected, unaffected, who has to be tested, okay? What is the turnaround time? Believe me, most patients are actually surprised to know that an exome may take 4 to 6 weeks, or minimum at least 3 or 4 weeks. And they're like, doctor, it's going to take that long? Because most things are available within like 3 or 4 days. So you have to explain. And ours is a referral center. We get patients from Bangladesh, we get from Maldives, we get from West Bengal. And they're like, I'm going to go away. If this test is going to take 6 weeks to come, then what after that? Okay, so those are the important things that we have to sort of inform them when we are talking to them in the clinic. And then we have to also sort of tell them about the limitations, that even if we do this very expensive test, which is going to come after 6 weeks, there is still a possibility that we may not be able to diagnose what your child has, okay? So it is important to, and even if we diagnose, say if we get a VOUS, okay, then I will not be the right person to sort of counsel them about a VOUS, then I will have to send you to a geneticist or a genetic counselor. That will take more time. Okay, so these are all the things which I touch upon when I do a genetic counseling for my patients. And the most important thing that they want to know, how is this going to help? Okay, is it really needed? Is it going to help my child? So then I tell them why it is important to diagnose, why it is important to prognosticate the condition. Remember, we just spoke about the severity. Okay, some conditions are mild, some conditions are severe. So say if you're having a severe type of epidermolysis bullosa, we can do some screening for you. If you are having some, maybe neurofibromatosis or xeroderma pigmentosum, we can send you to like maybe another specialist to see if you have any malignancy in situ or something like that. Prognostication becomes important, okay? Treatment, I'm going to speak about that. And sometimes I can tell them that, look, we may not be able to do much for this child, but maybe I can sort of, if you do this genetic test now, then maybe we can help you to have a second healthy child, okay? So prenatal diagnostic services, we can refer you to specialists for that, okay? And when the results come, okay, it is important for us. So every time a geneticist or a genetic counselor may not be available, or, your appointment may take some time. So we also end up having to sort of counsel them or explain to them a little bit about the results, like if it is a PTC, what does it mean? It may be more severe phenotype or, accordingly. Next slide, please. Next. Yeah. So this is very important for us, a test requisition form. And when I started my sort of journey in genodermatosis, unfortunately, I never paid much importance to this form. Okay, and I realized that it was very important to give an important, a detailed clinical history. And I will sort of discuss that in one of my case discussions later on. Next slide, please. Yeah. So one thing that we do take a biobanking consent, but we never inform the patients that we are taking this consent, okay? I think it is important to inform. Next. Yeah, so this, again, as I said, see, some disorders are look at the strike rate of NGS, okay? Average net, if you combine all genodermatoses, the strike rate of NGS is less than 50%, okay? That is like a wake-up call for all dermatologists around. people who work in this field, okay? Because at least I know in my community, okay, among the dermatologists, there is a feeling ki, acha, this is some kind of genodermatosis, let us send for a genetic test and something will come back. But no, it doesn't happen, okay? In some cases, yes, but in a lot of other conditions, you do not really get a variant which would really explain the phenotype. Next slide, please. So I'm going to spend some time. Now, how much time do I have? Okay. Oh. Yeah, so I'm going to just spend some time talking about therapeutics, okay, because this is something which is very close to my heart. So, see, last year gene therapy was made available, okay, for epidermolysis bullosa, and currently the cost of gene therapy per patient is between 15 to 20 US million dollars, okay. It is prohibitively expensive, but at some point of time, it is going to come to India, okay? And we have patients who can afford it, who want to go to US and who want to enroll in trials over there, okay? So now, is every patient of EB a candidate for gene therapy? No. Only those who have collagen 7 mutation will be a candidate for this gene therapy, okay? Next slide, please. So if I'm seeing an EB patient, I need to know, there are 22 genes which can cause EB, okay? I need to know whether the patient has a mutation in collagen 7 or not. Every patient of EB is not a candidate for this gene therapy, okay? Next slide, please. Gentamicin. Some of you may be aware that gentamicin works as a PTC read-through, okay? PTC, that is a premature termination codon, okay? So, and there has been some evidence regarding its use in various lysosomal disorders, and we have been trying in some of our patients with PTC mutations in LAMB3, which causes junctional EB, and as well as collagen 7 with PTC mutations. But for that, I need to know the exome report of the patient. I need to know what gene the mutation is in and what type of mutation it is. If I do not know that, then I cannot offer this therapy. Okay, so as a clinician, it is directly important for me to know what mutation in which gene is causing his EB. Okay, next slide, please. The other thing is losartan. Now, this is a trial which I have been running at Center for Human Genetics, and we have been trying losartan for severe variants of dystrophic epidermolysis bullosa. It works by down-regulating TGF-beta signaling pathway, and thus it ameliorates the fibrotic sequelae of EB, okay? Next slide, please. So this was the preclinical work which was done, which showed in mice model, which showed that the mice who were treated with losartan in their diet actually managed to sort of preserve their digits as opposed to those who weren't, and they ended up sort of developing a mutant hand deformity within 7 weeks. Next slide, please. Okay, now this is a report from our very own Karnataka. Okay, now which parent of EB will not want this? Tell me. Okay, they will all want some kind of relief of symptoms. Okay, but then again, can losartan be tried in every patient with EB? No, it can be tried only in dystrophic variants with mutation in collagen 7. So again, I need to know what is the mutation and in which gene to be able to give this treatment. Next slide, please. That was about EB. But then we have other therapeutics as well, which are kind of coming up, specifically selumetinib for neurofibromatosis. I think Dr. Vaikunth Raju has some experience in it. And sirolimus gel. Okay, these are all FDA approved. Okay, and this is just a very small list. Okay, I mean, I couldn't put everything because, I mean, it's not possible. And the ones which I've highlighted in yellow are actually present in India. Okay, now if you have patients, wouldn't you want to give this? Okay, but again, I need to confirm what mutation, do an exome, and then be able to get this treatment. Next slide, please. Similarly, for ichthyosis, we have various biologicals, okay, which can be tried. And although they are not FDA approved, there have been a lot of supporting reports in the literature which support their use. Next slide, please. So this is how I look at it, okay? I mean, I would love to have a geneticist or genetic counselor with me all the time, but practically speaking, it doesn't happen, okay? So I end up pretty much probably very imperfectly doing from step 1 to the last step, okay? So the other thing is, so a couple of other things that I want to touch upon is that what I have seen is that an exome test, okay, is not like your other routine lab tests, and we as clinicians should give adequate importance.
to the test, when you're talking about it, we should spend at least 30 to 60 minutes explaining about the whole process to the patients. And we have to be very clear that even if you get the results, you may not really be able to treat the child, okay? Because that is very important to stress upon. And whenever you have complicated cases, like you get a negative report or a VOUS, it is always better to refer to a specialist rather than try to do it yourself. Next slide, please. Do I have time for
About 2 minutes left.
2 minutes, okay. So we have these last 4 cases. I'm going to run through it quickly. So this was a case of palmoplantar keratoderma. We did a clinical exome and we found a mutation in keratin 16. Next slide, please. Now, palmoplantar keratodermas are characterized by thickening of palms and soles, and they result in very painful callosities where the patient is not able to walk. Okay, we confirmed it with Sanger. Next. And this is what the medication could do. Okay, so here, doing a clinical exome, okay, helped me to confirm which variant of PPK it was and helped me to decide what treatment I could give and directly make an impact on the patient's life, okay? Because acetretin cannot be tried for every variant of PPK. In some types of PPK, it can actually worsen it. Next slide, please. So this was one of my first cases where I started my genodermatosis journey. This was a patient with reticulate pigmentation, growth retardation. There were 2 children in the same family, undescended testes. I did not know much about genetics at that point of time, okay? And in my ignorance, I think I ended up ordering just a clinical exome. And the report came back negative, okay? The family was a poor laborer from Bihar, and when they spend around 15,000 doing a test and they don't get any answer, it really pinches you as a doctor, okay? Next slide, please. Yes. And no significant variant related, okay? So I was quite disappointed, but then it kind of made me wiser, Now, if I had known as much about genetics as I do now, probably I wouldn't have made those mistakes at that point of time. So when this came...
Time to summarize.
Yes. So in this case, I was a little wiser. This case was a peeling skin syndrome. Again, the West through something else, which was totally inconsistent with the phenotype. But this time we asked the lab to sort of reanalyze the data. Next slide. And when they did that, they actually found a mutation in filaggrin. It was a termination mutation, and it fitted perfectly with the phenotype. Okay, so I had the presence of mind this time to request the lab to sort of reanalyze the data and have a look at it again. Next slide, please. This is the last one, and this is where I want to leave it. This was a patient with very sparse hair since birth, with some mild CNS features, precious baby. And I told the parents that even if you do a genetic test, or whether you do or you do not do, the child will probably have to wear a wig for the rest of his life. And they said that they didn't want to do a genetic test. And it is fine. I said, it's okay, because I'm not going to be able to make any difference in this child's life, even if I do a genetic test. Next slide. This is my take-home message, okay? Yeah, thank you. Thank you for the patient listening.
Thank you, Dr. Gupta, for bringing out the depth of information in genoderm in this medical specialty. And it's also promising to see a clinician talk about the importance of genetic counseling. Thank you so much. Please join us in the panel discussion later. We are moving forward to the next talk by Dr. Shweta Yudar. I would like to introduce the speaker. She's a professor, director, adult genetics service at the Department of Molecular and Human Genetics. And she also is affiliated at the Michael E. Debye KVA Medical Center, Department of Internal Medicine, as a professor and section chief of genetic medicine. And her qualifications after medicine, she has residency in internal medicine and pathology. In addition to that, she has master's in biotechnology in the United States. Her career highlights, as you can see, she specializes in evaluation, diagnosis, and treatment of adults with genetic disorders. She has many publications and book chapters. She's also an elected clinical director of the ACMG Board of Directors and also course director for ACMG Genetics 101 course. And she has many publications as well in peer-reviewed journals. We will now listen to her recorded talk.
Hello, and thank you for inviting me today to give this talk on cardiovascular genetics. I'm going to explore some single gene etiologies and polygenic risk scores as it pertains to cardiovascular genetics. I'm happy to be here. I wish I was there in person. But of course, we'll make do with the best that we can through this virtual platform. Let me now share my screen so I can show you my slides. All right. So, thank you for the introduction. As was mentioned, I run the adult genetics program at Baylor College of Medicine in Houston, and I also work at the Veterans Affairs, which is our military department, and I'm the National Program Director for Genomics for the country. And I also am involved in the American College of Medical Genetics and Genomics on the board of directors, as well as on the education side as their vice president. So many hats that I wear. And today I'm going to talk about, cardiovascular genetics. We'll first kind of approach genetics versus genomics, the difference between the two, and if there is any difference. We'll talk about comparing single gene monogenic disorders and polygenic disorders as it pertains to cardiovascular conditions, and then we'll talk about some future directions. So, we keep changing this term genetics and genomics. We use it interchangeably. And the traditional sort of definition of genetic medicine focuses on rare monogenic variants. But as we learn more and more about polygenic inheritance and, various different other genes, we now kind of have moved towards calling them sort of genomic disorders. And, this figure kind of illustrates where you can have polymorphisms, and then that score ranging from 0 to 2n, or the number of polymorphisms for each person, is kind of what determines your polygenic risk score. And this is done using statistical models, and kind of you're collecting a person's variance and how it affects their risk for a particular disease. So this picture here, I kind of wanted to delve a little bit more into what we call polymorphisms, how is that different from our single gene variants? So you can see like these orange V's, these are shared variants, and then some of these bigger ones, so these smaller V's actually are the shared variants, and then these big V's that you see here are the unique variants. So in this example of cystic fibrosis, you see that they have a lot of these little small gray V's or variants, and then you have this individual with on chromosome 7 has these two orange big V's. And so that's kind of the mutation or pathogenic variant in the CFTR gene, which causes cystic fibrosis in this individual. So those are our single gene disorders, Mendelian pattern of inheritance. And then we look at these coronary artery disease, and you can see there are 60 variants causing coronary artery disease. So there's a lot of different V's in the background of the gray V's or polymorphisms that all of us have, and then you have these risk factors or risk alleles. Before we kind of talk more about PRS, I wanted to establish sort of our traditional genetics of cardiovascular disorders, which is our single gene etiologies. And, you're probably all familiar with these conditions, Ehlers-Danlos syndrome or EDS, Marfan syndrome, familial hypercholesterolemia, and cardiomyopathies. I am going to talk about Ehlers-Danlos syndrome. I only have 20 minutes for this talk today. It's a lot to cover, but I do see a lot of patients with EDS in clinic, and I'm going to focus on that today. So EDS is a group of disorders that share a common decrease in the tensile strength and integrity of the skin, joints, and other connective tissue. So there is looseness of the connective tissue. There were 10 different types that were established, and that's kind of what we followed until 1997. Then we moved them to 6 types. And then recently, they now moved them to 13 subtypes of EDS. And so collectively, there's 1 in 5,000 individuals who have a diagnosis of EDS. The most common one, and you guys may have seen this in your clinics, is the hypermobile type. And then the other common ones are classical and vascular. And hypermobile and classical type account for 90% of all cases of EDS. And I'm going to kind of go through some of these big ones in subsequent slides. So this is the 2017 EDS International Classification that came out, and this sort of also tells us the genes involved in these different subtypes. So as you can see, and you can read this at your own time, but the most common ones, like I said, is the classical, which traditionally we used to call type 1 and type 2. We have now stopped using the terms type 1 and 2, and we actually call them by their names. So classical EDS or CEDS. We have the genes that are associated with it. Then you have your vascular type, which is the most rare, but which is the most concerning. You don't want to have missed a vascular EDS in a patient because that leads to really grave consequences, such as hollow organ ruptures. For example, a pregnant woman would rupture her uterus during the pregnancy, or rupture of blood vessels or dissections, which are spontaneous and can sometimes also be fatal. And then, of course, the most common one, and we see a lot of this, is the hypermobile type of EDS. It's the one that's most challenging to diagnose because the gene or genes associated with hypermobile EDS are not known. And so that's kind of where these patients come in with a lot of different conditions and problems and complaints, and you kind of have to sift through that and sort of make the diagnosis. And we'll talk about this in detail as well. These are the other types, and note here how there's different major criteria. And then, of course, these are all on a spectrum. EDS is on a spectrum, so you can have sort of overlapping features, but you go by the most prominent symptom in that particular category to give them that the diagnosis of that particular condition. So let's first talk about vascular EDS. Like I said, this is the one that is the most concerning for patients as well as for providers. You don't want to have missed this. It's only about 5 to 10% of cases that actually end up being vascular. You do have the characteristic faces. So the acrogeria, kind of person who looks kind of much more older than your age. You can see the thin translucent skin with highly visible subcutaneous vessels. You can see this kind of bruising leading to ecchymosis, hematomas. And then, of course, like I said, the organ ruptures. Typically, they don't have as much skin hyperelasticity as you see with the other types of EDS. Now, mind you, these are all very, very classical features. I've seen several vascular EDS patients, and they actually do not have these kind of typical features. So the best way to make the diagnosis in them is through genetic testing. The good news is that the gene COL3A1, 99% of individuals who have vascular EDS, you'll actually pick up a mutation or pathogenic variant in that gene. So the best thing is to kind of, you don't want to wait for a hollow organ rupture to make the diagnosis. I typically will run an EDS panel and rule out vascular EDS when I see a patient with a suspicion of some sort of Ehlers-Danlos syndrome. And especially because, like I said, most of these patients are hypermobile EDS, so you want to sort of rule out the other types of EDS and then settle by exclusion. You settle down with the diagnosis of hypermobile EDS, and then you can manage them accordingly. Moving on to classical EDS, the major diagnostic criteria. So there's skin hyperextensibility, they have wide atrophic scars. So because this tissue will not be able to oppose very easily because it's just so loose. And they, of course, also have joint hypermobility. And then sometimes also with a positive family history. But the typical criteria or the major manifestation in classical EDS are the skin findings. So, for vascular EDS, you're looking at all of the dissections and aneurysms. With classical, you're looking at more of the skin hyperextensibility, like really loose, stretchy skin. That kind of is way more than you would expect to see, kind of like as you can see here. If they have any kind of scars, they tend to gape or split after slight trauma. The wounds tend to split And so the scars really look really bad. Now, the best place to test for skin hyperextensibility is on the volar surface of the forearm. So not kind of this skin here, where, you know, all of us kind of have loose skin over here. People kind of pull here and show you. But the best place to test is on the volar surface of the forearm. So as you can see here in my hand, you would actually pick up, you know, pinch the skin here and see if that is loose. So that's the best place. And it has to be more than a centimeter to say that this is actually hyperelastic skin. Moving on to hypermobility, yes, like I said, that's the most common one. And we see this a lot in clinic. We calculate or examine the patient to see if they meet the Bateman score. Now, the Bateman score, it's a score from 0 to 9. If they meet the score more than 5, if they have 5 points out of 9, then we say that they truly are hypermobile. And so typically what we would do is, one is your thumb, you want to try and touch it to the forearm. Mine, of course, I do not have hypermobility, so my hands aren't going to touch. Or my thumb is not going to touch, but if they touch, then each of those, in both of those hands, is point of 1. You put your pinky or your little finger, you try to push it back, and it has to go more than 90 degrees. So mine is kind of at 90 degrees, but what I typically try to see is the give of the joint. So not so much as the angle, but when you've done a lot of these exams, you can actually tell by how the joint is, does not have much resistance. And when I'm examining these patients, I have my trainees with me. I will have them do this Bateman score on my hand and show them what the resistance of the joint should feel like. And then I have them examine the patient, and then you can tell that that's kind of really loose in the patient. So that's, each one is a score of 1. So you have your 1, 2, 3, 4. Then you have them extend the elbow. And again, that's the carrying angle, and it'll be sort of 180 degrees. As you can see in my hand here, this is just plain 180. With them, it's more than 180 degrees, as shown in the figure F here. And so each one, again, is a point on both hands, so that's 6. Your knees have to hyperextend, and that's the hardest one, I find, to kind of see that hyperextensibility of the knees. And then they have to touch the floor, as in the picture here. They have to touch the floor with your hands flat on the floor without bending your knees. So that's your score of more than 5, then you know that they are truly hypermobile. There's a lot of different clinical features of hypermobility EDS. Obviously, the major predominant one is the recurrent dislocations and subluxations, chronic pain, autonomic dysfunction. So you kind of have your postural orthostatic tachycardia syndrome, or kind of where your blood pressure is kind of going down and your heart rate's going up. They have the TMJ joint function. So this is the TMJ, and that kind of locks up. They have dental caries, or cavities in their teeth, migraines, irritable bowel, blurry vision, increased allergies, and heavy menses and endometriosis. So there are all of these different features. Not all of these are part of your diagnostic criteria. So if you look at the 2017 hypermobile diagnostic criteria, the main one is the hypermobility and the Bateman score, but then a lot of these other things are not part of the criteria. As on a side note, the criteria are being revisited by the EDS consortium, and maybe they'll come up with newer criteria that may include some of these things. But when I start seeing these patients, I kind of see the gestalt of the patient as they're kind of, you kind of see them in with these different features very easily. Now, only one type of EDS runs in the family, so you don't have hypermobile EDS and then vascular EDS in the same family for the most part. Classical is 50% inherited, 50% de novo. Hypermobile also most have an affected parent, but we don't know, again, because we don't do genetic testing for that. And then vascular is also 50% inherited, 50% de novo. Management of EDS, we typically do an echocardiogram, we do a bone density scan because their bone density tends to be low because of their hypermobility. We check the vitamin D levels, eye exams, physical therapy, pain control. So some of these things, again, can be done with no matter what diagnosis we give them, because the management is purely symptom-based. And you can go to the EDS Society to get more information if you'd like. So with that sort of introduction into single gene disorders, let's talk about some polygenic etiologies in cardiovascular disease. This is kind of each polygenic risk score is put on a bell curve distribution. So you find that most people will find their scores to be in the middle, so they kind of have average risk for developing disease. And if you either are on the tail ends, to either on this sort of low risk or high risk, then those on the high risk will actually benefit from some discussions about lifestyle modifications and medications and things like that. Now, this example here shows you monogenic familial hypercholesterolemia mutations or variants are identified in 1.7% patients, leading to a 3.8-fold increased risk for heart disease. So your increased risk with that monogenic disorder is 3.8-fold, and then you can look at this. You take 100 patients with myocardial infarction, and they have a high polygenic risk score, and that increases the risk by 3.7-fold. So together, all of those variants will also increase your score. Okay? There are polygenic score catalogs that are available, which is an open database, and there's then this number keeps growing, but you can see here many different traits have been established in this catalog. What's the point of PRS? And you find PRS everywhere. This is developed for more than 15 disorders. It determines, like I said, the low, moderate, or high risk of a disease, which can be minimized by diet and lifestyle. This is much more accurate in European populations because that's where the studies have been done. The accuracy is lower currently for non-European populations, but as more research gets done, I'm sure that will improve. It modifies penetrance and leads to variable expressivity, and then different PRSs may have higher utilities at different ages. So depending upon the age of onset of those conditions, your utility may be different when you're doing that in a child versus an adult versus an older individual. And then, of course, people are accessing their own PRS through direct-to-consumer testing, and these are, you have to obviously use them in context. There's a lot of literature out there for how this can be done. And I like this quote. This is from the Office of Public Health Genomics. Just like other areas in medicine and public health, there is no one-size-fits-all for PRS. Their eventual use in clinical practice will be specific to clinical scenarios and population groups. These are defined by race, ethnicity, ancestry, age, gender, disease risk factors. And then there's a lot we still don't know, and more research is needed. Okay? PRS can add to clinical risk scores as it pertains to cardiovascular disease. So, you have the Framingham risk score, Q risk, ASCVD. So there's lots of these different clinical risk scores that have been established for myocardial infarction, for atrial fibrillation, things like that. And so your polygenic risk scores will add to those scores. And so you can kind of have a more personalized approach to that patient, to their condition. But widespread clinical use is still ongoing, undergoing evaluation and research. And so it's not used commonplace. We don't use it commonplace yet in our clinics. So in summary, I kind of wanted to give you a flavor for what we consider monogenic disorders and then what we, mean by polygenic disorders. And I hope I've emphasized sort of your inheritance patterns are different. Obviously, with monogenic disorders, we kind of know the specific inheritance patterns, your predictive power, somebody has Marfan based on the penetrance, whether they will, if they have a mutation in the FBN1 gene, whether they will have Marfan syndrome and, penetrance, expressivity, and things like that. And then you have very specific management strategies for those single gene disorders. When you start talking about complex disorders like hypertension, diabetes, CAD, or coronary artery disease, then you start kind of thinking about the overlap with environmental factors, lifestyle, all of those that also play a role along with your genetic burden.
No doubt this is a very important and interesting session. In the interest of time, we stop at this conclusion and move on to the next talk. Thank you, Dr. Shweta, if you're available online. Over to Chairperson for the introduction of the next speaker.
Okay, let me introduce next speaker, Arjun Gupta. He is a founder and CEO of Genetico Research and Diagnostic Private Limited. His academic credentials course Masters in Entrepreneurship from Rose School of Business, University of Michigan, and BTech Biotech from Amity University. His career highlights: he received Direct Grant in 2019 to develop India's first AI-enabled clinical genetic software and patient matching platform to support diagnosis of complex and undiagnosed rare and genetic disorders in India. Founded a vaccine company to commercialize novel vaccine technology targeting mutating viruses such as HIV, hepatitis C, and influenza. Worked in a $100 million venture capital fund in Houston, Texas to evaluate high potential biotech companies for investment. Worked as a product manager in Advaita Bioinformatics to develop multi-omics analysis platform. Worked as a product manager in Genomenon. Worked as a molecular diagnostics and drug testing lab in India. Trained in ISA 10725 for quality system testing laboratories. You too, Arjun.
Thank you.
Mr. Gupta, we are not wrong. You have 10 minutes time for this presentation.
Right. Thank you. At the outset, thank you, ma'am, for having me here and the organizers. My topic is around the role of deep phenotyping and the use of human phenotype ontology in the diagnosis of genetic disorders. So deep phenotyping, mainly two aspects I would like to highlight, which is the comprehensive examination from head to toe. Many speakers before me have spoken about the importance of this. And then there is evidence, there is hard literature to show that more number of phenotypic terms tend to increase the diagnostic yield of the test. And talking about the second aspect, which is the precise phenotyping. So instead of giving up, observing a high level, phenotype, as deep as you go, it's going to help you in many aspects going forward. So to this end, human phenotype ontology was developed, and this was mainly inspired by, the ability to communicate effectively across the globe about specific phenotypes, so that especially in the field of rare diseases where we have a lot of undiagnosed conditions, we could understand that, two clinicians are basically talking about a similar phenotype. And one of the biggest aspects of this is the hierarchical structure that it has brought to the phenotypes. And that makes a huge difference, which I would just speak about. So just in a common workflow, when a patient comes to a clinic, I think this is the most critical point where the examination needs to happen. And then when a sample is sent to a lab, typically we see in India that genetic counselors would extract, if it is not already in the HPO formats, it would be expected by the genetic counselors a lot that, you would extract the HPO terms, manually research the differential diagnosis, which is then provided to the genome analysts, and then they would use it in their software. So almost all software today are using, HPO phenotype. So what I would like to share here is that if we all, come on to HPO phenotype, HPO-based deep phenotyping, there are tools that, technologists like us can build to reduce your time, automate your workflow. So here you would see that, in our software, if you've written a summary of a clinical summary, we have algorithms to, extract the HPO terms, and we even go as far as, covering a set of synonyms. But there's active work going on where we are using natural language processing to even convert your general terms that you have been using for the clinical terms to convert them into the HPO. But this is just to cut down some of the time for your research. The end decision lies in your hands to see that this is the correct phenotype that we are choosing. And then there are ways to automate the research that, many times you might find yourself doing by going onto various websites to see which are the relevant differentials. So there are, we've built one algorithm. There are publicly available algorithms like Phenomizer, but we've added more parameters to this algorithm by understanding which are the specific and non-specific phenotypes that are associated with the condition, so that the ranking could be improved. But then we don't say that this is the ground truth. I mean, it's very difficult to come to a diagnosis from this. But then there are additional modules that can be built on top of it, and this is the one that we have recently built. So if you're suspecting between 3 or 4 conditions, this module would basically help you compare across various organ systems or various systems at what kind of phenotypes I can observe to really resolve between 2 closely related syndromes. And in a workflow, you can even see that which were observed, which were unobserved, which are very high frequency phenotypes with a particular disease. This is just an example where we are talking of a patient matching, how HPO really enables patient matching. And patient matching is very important for genetic conditions where you are unable to diagnose a condition. So in case you can find a similar patient with a similar phenotypic profile and a variant in the same gene, that actually increases the probability of diagnosis multifold, or at least it will give you an opportunity to discover a new disease gene or characterize a new disease phenotype to genotype relationship. So in this particular example, so there are 3 patients, I'm sorry, it's a little mismatched. So on the right, on the left, you see that these are the hierarchical structures, and right at the bottom, there is arachnodactyly and there is long fingers. So if I were to match 2 similar patients, one say in the north in J&K and one in Kerala, so these are not word-to-word similar patients. So you would not get a match by finding a similar word. But this is what HBO brings to the table, that, you know, it identifies that these 2 are related, and hence there are algorithms to really compute that similarity without having 2 similar terms. So it makes a huge difference when you have huge set of undiagnosed patients and you want to stratify those patients based on algorithms. So there the HPO plays a very, very major role. And interestingly, you know, this is not something, this is something which is happening globally at a good scale. There's a consortium, all these come together for patient matching. So when I saw that we don't have one in India, I thought we must build it. So BIRAC was gracious enough to give a grant, and we've actually built it, we've tested it. We are hoping that, you know, we can launch it soon. So if there is interest here to kind of beta test it with us, that'll be great. Now, the last piece, and it's a very deep piece, so I have 10 minutes, but I will just touch on this. I think Sir mentioned that for mitochondrial DNA disorders, it was not very helpful, but there are a lot of good examples that, you know, where there is a gap in the knowledge that you are not able to diagnose, but there can be, you know, clues in the animal model. So with the help of HPO, it has made possible for technologies like us to build pipelines that can bridge that knowledge gap. So if you can find, you know, orthologs in relevant animal models, there is a way for you to kind of understand that is it really mimicking the disease, and it will give you clues to go and do the functional studies, and then of course, you know, you can probably establish some new discoveries. So I think just in summary, dephenotyping is critical. There's a lot of time I've heard, you know, some peers talking about now we have a genotype first approach. But no matter which approach, it is really, really critical that you deeply phenotype the patient at the time of examination, because you can always go higher and cover more and more genes. That is always an option, but you cannot go lower once you have selected a higher phenotype. So a message to fellow, you know, peers here that it's really critical that you do a really good job of phenotyping up front. And then there are, if you are using HPO and you're doing a good job at it, then, you know, it gives a lot of potential for us to give you the tools that can automate a lot of labor work and things that you may not be able to do it in a short amount of time. So, and then patient matching could potentially, you know, solve undiagnosed cases if you can find similar cases. So I think we need to find a way to start doing this in India. I've seen talks where clinicians have done this with the international, you know, tools and platforms that are available, but I think, you know, being such a large country, we should have our own platform. And then, you know, projects such as Monarch Initiative offers tremendous ways to kind of bridge this knowledge gaps, and could be a great interdisciplinary study, especially for India, because, you know, our genetics are different, and even I've seen some of the data with our collaborators that even the phenotypic variability is also there when we compare to the Caucasian population. So this is the team behind doing this kind of work, and a couple of our team members are present here. So if you would like to know more about the work that we're doing, please connect with us. Thank you.
Thank you, Dr. Arjun, for highlighting the importance of technology in clinical service delivery. Certainly agree that we need such databases in practice. We move on to the panel discussion now. I would like to invite all our speakers onto the dais to join us for panel discussion. In the meantime, I'll summarize. This session was on rare disorders in pediatrics and adult setting. And we had started with a very nice story of, at least 2 decades of how a family was followed, and then the success of that by Dr. Arijit. And followed by that, we had good omics integration in mitochondrial disorders. And we also did see in specialties like dermatology and vascular medicine, how genomics plays an important role. And of course, Mr. Arjun gave the final touch of how technology innovation can support clinics. On that note, I would like to hand it over to the moderators to facilitate this session. Thank you.
The panel is open for discussion. Any questions from the audience?
Do we have the online speaker?
I don't think so.
Okay, all right. She is unable to join us. Sorry.
You have done about 1,500 cases of mitochondrial.
Yeah.
You yourself said about heteroplasmy, differential penetrance, and so on. So where is the genetic counseling in all this?
Yeah.
I know it's one of the most difficult things to counsel
Yes,
mitochondrial
for mitochondrial disease, particularly for mitochondrial DNA variations. For say example, if you see, as I said, only 10 to 15% of the patients what we see, there is the role of this particularly mitochondrial DNA variants. If you see LHOM, there are 3 primary variants.
No, that is all fine.
Yeah. I'm
counseling, how are we doing? All these cases you have done.
Yeah. I'm just
published.
Yeah. It's published.
But have you gone back to the patients?
Yeah, but few patients are in follow-up, few are passed away. So this is just a last 2 decades data which I clubbed and just presented. For example, if you see in terms of genetic counseling with respect to mitochondrial mutation is really difficult. For example, if there is a LHON, only the LHON phenotype only exists in homoplasmic condition. That irrespective of the family pedigree, if you see, maternal people also will carry the same homoplasmic mutation. But for example, few of the maternal relatives may not show the clinical expression. But there the male is a predominant factor for causing the LHON phenotype. In addition to this, while counseling, for example, mother is there, she is carrying the homoplasmic mutation. Child is having also the homoplasmic mutation, but there is a clinical expression in the child, but not in the mother. Because there the other factors will play a role, like mitochondrial haplogroups, nuclear genetic background, and sometimes this heteroplasmic factor they say, but as per our experience, more than 95% is the heteroplasmic cutoff for this LHON. Likewise, for other phenotypes, it's very difficult to say that, for example, if patient is carrying some 80% mutant, but we cannot say that the next child will also have the 80% mutant. Yeah.
He showed in a film a simple thing that how do we help this patient families, clinicians, counselors to handle such a situation?
No,
because for you it is 80%, for me it is 10%.
Yeah, fine.
But it's a family.
Yes. So for that reason only, what I'm saying is, in case if it is a known cause, we can counsel it. That is the cause for this. In case if it is if it is a VUS or like any novel variants, we need to go back the functional validation. Then only we can get. Otherwise, we cannot immediately tell to the family that this is what it is carrying. So you will, and the next child also will get, we cannot say, because the heteroplasmic may not be stable. That is one of the major constraints in the mitochondrial disease.
No, I think so this is very difficult, and as you said, mitochondrial is much more difficult than nuclear, right? What he's talking about lawn and any other diseases. Only thing I would comment probably differently from what he said, that this is we are very comfortable communicating like this, an academic with another academic, right? But if we say the same thing to the patient, they will understand, and they won't understand anything. So only thing I would say probably, like the example you gave, that both the mother and the child has homoplasmy in lawn. Child has the disease, mother doesn't, because of variable expressivity. So I think what she's trying to ask is, when you see that, do you explain it to the mother that, look, there are reasons in simple language, which is why you have the same genetic feature, but you show your child shows the phenotype or might show the phenotype and you are not showing the phenotype. That's, I think, what you are trying to say. That communication is missing. Yeah.
Communication, if you say, I'm in case if people can start to really understand what we explain in simple term, few people we will explain. Few people, they may not be understanding what exactly we explain. But
in failure if we can't make them accountable.
No.
Agree with me?
But see, in case if you can convince the family in terms of
Is it our failure that we are not able to convince them?
I think that if such is the case, like we don't have any specific genetic counselor or something which most of us what we work on. For example, if you consider our patients, whatever we have learned, even clinicians or the pathologists or this thing, based on the discussion of the team, we are trying to address each cases. In such case, we'll be trying to explain them, this is the reason for it. But I understand. I'm just talking about the difficulties in conveying them to reach to the family. I know that where you're coming, but
yeah, if I can, to be very honest, I don't know much about mitochondrial inheritance, but I know that it is very difficult to explain. So in such a situation, possibly something that might help would be a multidisciplinary clinic where the clinician, the counselor, the geneticist, they sit together. Not you go from this person to this person, you sit together at the same table, and also depends on the level of understanding of the patient, like somebody who's
we are talking of communication here, right? You are talking of communication. Talking about conveying.
Yes.
This is all. They take convey.
I'll share.
How much they can understand the different
To add to that, in a council, I wear the genetic counselor's hat most of the time. It is the information that is conveyed that we are discussing here. We use different models with different colored balls within a cell and show them that, okay, these many colors represent the severity in it. So that's how we explain to them as counselors. But beyond that, genetic counseling is that the implications. A mother would ask, can I not have children at all? So these kind of discussions come after we explain these things. So that's what we are asking. Do you have such, model system to?
Okay. So in case of mitochondrial DNA variants, really it is difficult. We will say the facts, what is happening. But the same thing, one case which due to time constraint I didn't present. If you see, there is one patient called ethylmalonic encephalopathy phenotype. We have been observing like 8 years of the patient since birth. Later, based on the MRI profile, biochemical was normal, but still exome sequencing has identified VES. Further, we characterize that mutation and we inform to the patient father, this is what it is happening. Further, what happens is we have given the counseling because that is a recessive mutation because it's a nuclear gene. Later, we have told this is the fact, and he also supported for us to do the research. Such kind of parents also there to want to know what is the cause for the disease for his son. So we could able to explain by doing the lab experiment, which further help us to counsel him. Then later, the second baby born, she is a carrier. But that patient, the first patient died in the 8 years. So such kind of experience is also there. But until unless we have a, like a, yeah, until unless we have a proper evidence with this, still we can educate the family members. That's what I meant.
You have the burden of lab and all that.
Yeah.
The
So
message
everybody
is
has a
that-
different-
The message is that that's just make use of counselors. All right. So moving on to the next topic, I would like to post a question to our Dr. Gupta. Do you come across patient families asking for prevention? I mean, for treatable conditions. For example, a woman comes and asks that I have nevi, and then I do not want to have my child. In fact, Dr. Annie, and we had one case like that. One came and said that I don't want my child to have this. It's not that life-threatening and serious. So how do you deal with such conditions? Do you offer genetic testing or?
No, so see, the thing is, nevi is most likely secondary due to a somatic mutation. It's not a germline mutation. Even if you would want to do a genetic test, I don't think it would be possible, or then you would have to take a skin biopsy, send for ultra deep sequencing and those things, okay? So for nevi, definitely the only thing that I do is I just reassure, okay, for the patient that, see, if the patient wants treatment for herself, there are a lot of things that we can do, possibly involve the plastic surgeon, do some laser. Some nevi cannot be treated. And then we have to sort of counsel them that, this is something that you have to live with it. But with nevi and things, it's very difficult to really predict, whether there are very few conditions, some types of epidermolytic ichthyosis, which can be inherited as a nevi and have a germline mutation. Okay, those things are extremely rare. So,
Thank you. Thank you for explaining that.
Yes, ma'am. So in that case, then what we would do is that probably we would do an exome for the mother. I don't think I would really advise them. Or I would probably refer them to a geneticist or a genetic counselor.
Just quickly, in your experience, has pedigree helped you arrive at a clinical diagnosis looking at the pattern of inheritance?
Yes, it has. Okay, especially so as I said, most of my work is in epidermolysis bullosa, and we have dominant dystrophic epidermolysis bullosa, and we do get those families. So it is easier to counsel them. We take a 3-generation pedigree, typically, not beyond that. And dominant dystrophic is typically milder in phenotype, okay, as compared to recessive dystrophic. So it has helped.
Okay, I have a comment and suggestions to ask from Dr. Arijit. Okay, we don't have an Angelina Jolie, but we have Amitabh Bachchan, Rajni Khan, and a lot of Dr. Vaniyar is here, Dr. Aini is here. They are the role model of us. So can we go for, in your case, tried any of these role models to come and taste and can do this thing? And the second thing, it's very nice that the movie, cartoon, like the people, Jerry, Tom and Jerry, these cartoons does let understand the visual documentary. So GCs, should we start training the GCs, the visual AI assisted tools or something like that, please?
Yeah, so I'll answer the second part first, because that's the easier to answer. So my first thought was to make an animation film, not to involve humans, because I did not know anything about making film. I thought that would be easy. I don't have to find actors and people like that, right? But I found out that making this film was 1/4 of the cost that I would have to pay to make a cartoon or an animation film. So I learned that. So that's a good idea. But you can find, so there is a journal called Journal of Visualized Experiments, JOVE, right? JOVE has a section for educational resource. And because of this is a TikTok generation, all JOVE videos are 2.5 minutes long, bite-sized videos as they call it, right? So they have, if you're looking for educational resource, then JOVE has covered most of the molecular biology genetics resources. Some of them are free, some of them are paid because that's part of their business. That is one. for this, as I said, for this kind of disease-specific or family-specific issues, if you want to convert into a educational resource or animation or cartoon, they actually cost more than hiring human actors. So that's the answer. The second part of the question about your role, first part of your question about the role model, as I said, we don't have Angelina Jolie in glaucoma. Without offending any seniors in the audience, Annie Hassan as a role model in this hall is very different from Angelina Jolie as a role model for breast cancer, right? So you need a Aishwarya Rai, you need a Priyanka Chopra, you need a Vidya Balan, you need that kind of people. Yeah.
Bachchan and Karishma Kapoor, they both are carriers of thalassemia, and that was one of the reasons why the Bachchan family was not happy with them getting married.
Started by Mithun Chakraborty.
But that's why I always say that they didn't have a good genetic counseling.
So we are not for glaucoma though, but we did approach from IGIB 2008, long back when I was a young faculty there. We approached Amitabh Bachchan for him being the first human genome to be sequenced. He refused because of privacy issues, right? So, well, so that's, but we do need faces, spokesperson who will say this. As was presented yesterday, that after Angelina Jolie's double mastectomy, people just volunteered for mastectomy in the UK, for example, right? So,
I think we can overcome the language barrier.
So that is another thing we were talking before the talk. I wanted to dub it in Hindi, have the complete thing in English for the UK people to also understand, and a few key major languages in India. Also, I figured out dubbing will cost me more than what it take me to make the whole film. So voiceover, what is possible, cheaper, still expensive but cheaper, is voiceover using AI. That we are trying to do, but not done yet.
Thank you. Definitely, it's not an easy task to collaborate and produce such a movie. Before we move on to the last question in the audience, you have the time to raise your hands for the last question. I would like to pose this question to Mr. Arjun. What do you have to say to our genetic counselors who wish to be entrepreneur like you? What is the future of technology in genomics, and your insights to them?
Okay, great. Lessons on entrepreneurship are common for everybody. I would say that really get to know your field, really do the hard work, and really understand the lay of the land. And technology is a big enabler. I've seen the clinical audience sometimes shy away from it. So you can really bring that to the table, I feel. That if you get really well versed with technology, what you can essentially do is cut down the time for many stakeholders in the process. And I can already see that the industry is very reliant, like they have to rely on you, they're already relying on you, and they will continue to do so. It will grow even bigger and bigger. So if you can embrace technology, use it, cut down times, bring out good insights. And one big thing that I would say is that at least build your own databases. If Upasana yesterday was saying she's been counseling for 8 to 10 years, she would have done probably 2,000 cases. Why aren't you guys publishing with your clinicians? So that's a way to bring forward your capability as a counselor. And then coming specifically on entrepreneurship, I think when opportunity will meet, when you will come across opportunity, and if it will really drive you to do something about it, you will find a way to go about it. I mean, I'm not a successful entrepreneur. I have been surviving. But yeah, so so if you've done right, absolutely, find a good mentor for sure, but then really big stress on do the hard yards right now, and that will really pay off. Right, I mean, sure, after the talk, yeah, sure, yeah.
Can I take the last audience question? If there is nobody.
Yes, one more question from the audience to the
I
panel.
actually have a question for Arjun, if that's okay. So it's a very straightforward technical question. Your app or your platform, does it also do pedigree drawing and analysis?
Yes, it does pedigree drawing. And as far as analysis is concerned, if you're asking about inferring inheritance patterns, what the idea was that to have, once we have large number of pedigrees, we have the ability to store clinical data onto the pedigrees itself, whether these are phenotypes, whether these are genes. So that gets stored in a data structure in the backend. So once there is a large database, we can run, unsupervised algorithms also, models also, and find out some interesting insights. Or it can be a question-driven, kind of a thing. So our app does pedigree. In fact, we are the first in India, probably the first in Asia also, to have a homegrown, home-built pedigree editor. And yeah, so to answer your question.
Thank you. Okay.
He's developing as well. Okay, thank you. Do we have any question from the audience? One last chance. Okay, if not, we will close this session. Thank you so much, all the panelists and chairperson, to facilitate this. Thank you.
Now I kindly request our chairperson, Dr. Ajay K. Mahalka, to present the memento to Dr. Arjit and P. Govindarajan.
I request Mr. Suraj to felicitate Dr. Divya and Mr. Arjun. I request Dr. Dilla Froze, ma'am, to felicitate our chairperson. I request the panel members to be on stage for a photograph. We'll break for tea for 10 minutes and we'll be back for the last session of the conference. Thank you.
Good afternoon, everyone. I'm Kankana Goswami, genetic counselor from Northeast India, and I'll be moderating the session with our senior genetic counselor, Dr. Mansi Vishal, and she is a genetic counselor from East India. And we welcome you to the last session of BGCI, which is on early and late neurodevelopmental disorders, genetic testing, and genetic counseling aspects.
Actually, Kankana is from East India and I'm from West India. So with that, I would like to call upon the stage our chairperson, Dr. Amarjeet Singh Bhavar. Sir is one of our patron at BGCI. You can see on the screen that he is professor and head, Department of Genetics, Shri Guru Ram Das University of Health Sciences, Amritsar, India. He got retired from GNDU, Guru Nanak Dev University, Amritsar, as a professor, Department of Human Genetics. Yes, he has done PhD in human genetics from Punjabi University, Patiala, and till date, he has more than 70 publications in human genetics to his credit. And he has been former president, Indian Society of Human Genetics, former dean, Faculty of Life Sciences, former director, Internal Quality Assurance Cell, Guru Nanak Dev University, Amritsar, former coordinator, DRS SAP 1 and SAP 2, University UGC of Department of Human Genetics. Sir, please come on the stage and take your seat.
I would like to invite our co-chair, Dr. Dil Afroz ma'am, on our stage. Dr. Afroz is the professor and HOD of Advanced Center for Human Genetics in schemes. She has done her PhD in life sciences from Jamia Millia Ismailia, New Delhi. And she is also the vice president for India Immunology Society. Her current ongoing project is on establishment of cancer diagnostic center at tertiary care hospital schemes. She is also the visiting professor of University of Louisville and University of Lincoln. Ma'am, kindly come on the stage. The session is over to the chairpersons and the co-chairs.
Welcome all of you once again to this last session. And this is on early and late neurodevelopmental disorders. So for the first talk, I would like to invite Dr. Bangtu Raju KN. He is professor of the pediatric neurology at Indian Indira Gandhi Institute of Child Health, Bangalore. And he has his academic claims include around 200 PubMed Index publications and 75 of them in the peer-reviewed journals in around 10 projects. His additional qualifications include DM in pediatric neurology from AIIMS, New Delhi. Career highlights that include textbooks on cerebral palsy and 25 chapters in other textbooks, besides having other outstanding contribution to the child neurology.
Shall I start?
Yeah, sure.
Yeah, so good evening to all. Thank you, madam. At the outset, I thanks organizers for giving the opportunity to speak on this subject. So I'll be speaking on role of genetic testing in counseling in pediatric neurological disorders. Maybe a lot of overlap can be there, so I will be not going in detail, but specifically I will talking some common disorders that that commonly seen in pediatric neurology disorders. So basically, I'll be talking on case-based approach. So I'll be dividing into these following headings. Initially, neurometabolic disorder, that is commonly common condition, we see genetic condition, followed by developmental delay and autism, and sometimes cerebral palsy mimics and epilepsy and neuromuscular disorder. So I'll be talking about each cases and giving some clue for how to help the children and family. So as you know that there is a wide variety of inherited neurometabolic disorder, neurogenetic disorders are discovered with advancement in NGS technology, and there is a lot of scope for genetic counseling after discovering these disorders, and detailed clinical phenotyping is a must before going to genetic counseling. And earlier days, now also most of the people think that genetic test should be considered as a last resort. But recently, almost 1 decade back, many people suggested gene panel, including whole exam, should be considered the first test when you are considering these disorders, because it is, nowadays it is more cost effective and easily available also. This is a chart showing that where you are. So earlier in 2000s, we used to mainly focus genetic basically for testing, especially with karyotyping and CMA and PCR. And in the later 2020s, we got facilities of NGS technology, where there is some scope for management of some disorders which are commonly seen in neurology also. So later now that you can know that a lot of improved technology with whole group of tests available. So and also many of them are treatable, especially precision medicine plays an important role. This shows that importance of various tests currently available. So there are two types of tests. Maybe we already gone through all these. Cytogenetics include characteristic microarrays, plays important role in neurology children, and molecular genetic gene panels and whole exome, whole genome, and sometimes multiple genome also. This is a chart where you consider, we'll be giving some of the examples later. So microRF plays an important role in neurology, pediatric neurology, especially as looking at children with developmental delay, dysmorphic feature, autistic feature, and multiple congenital anomalies. And single gene disorders rarely advised nowadays because of not much cost effective, so maybe a phenotype variation can be there. So now commonly consider the technique is NGS, where if you are not sure that what exactly you are dealing and various heterogeneity is in phenotype, then you can consider NGS, and repeat expansion is required. Some condition I will be telling in a few slides after this. These are the advantage disadvantage. I am not going to details. So it should have been already covered by previous speakers. Some of the advanced single gene tests nowadays they use, unless if you are sure that you are thinking of specific disorders. The problem is it is a time consuming, and gene panel commonly used. The yield may be high, but sometimes you are not sure the phenotype. That time the yield may be less. So whole exome is commonly considered test nowadays. This is a monogenic disorders where you consider still a single gene disorders. One is spinal muscular atrophy and Duchenne's muscular dystrophy, and hereditary neuropathy with pleasure palsies and Charcot-Marie-Tooth disease and Rett syndrome. Where if you are sure the diagnosis, you can go for single gene test instead of going for gene panel and whole exome sequencing. Similarly, repeat expansion should be considered as the current techniques may not detect repeat expansion with our NGS-based techniques. So clinical phenotyping is very important. Some of the common examples you will see in children is Fragile X syndrome, Friedreich's ataxia, mitochondrial dystrophy, and spinocerebellar ataxia. So the clinical diagnosis is important to consider what test should be done. And this is the test for various techniques that yield test various neurological disorders. So coming to the importance of phenotype by clinician and also the importance of genetic counselors, and give you some of the examples. So I'll be telling initially the group of disorder which present basically with a developmental or intellectual disability or with artistic features, sometimes hyperactivity disorders also. So clinical typing, you can see that it's very obvious that the all clinical features suggest that a single disorder, the Down syndrome. So in this setting, the testing is very simple. We can go for karyotyping. So that is the important clinical diagnosis. Similarly, the various other disorder, you can see this various behavior of this child. So classically, these children, the female children, girl child with initial normal development in the first 6 weeks, first 6 months, followed by artistic regression with Migren stereotypies and microcephaly, suggests that is a specific condition like Rett syndrome. So you can go for MLPA or MCP, or you can go for directly whole exome sequencing also. Similarly, this is the other child with developing delayed artistic feature with excessive laughing, so that indicates the Angelman syndrome. So you can select the test based on this phenotype. You can go for microarray, or you can go for MLP also. So that the importance of clinical phenotyping is very important. The similar patient with excessive laughing and happy perfect appearance with the microcephaly with artistic feature ataxia with characteristic phenotype of Angelman syndrome. Similarly, male children with intellectual disability with artistic feature with large head and prognotism, large ears, large test indicate the most common is Fragile X syndrome, where you need to do a triple repeats rather than whole exome sequence karyotyping. So where it is a clinical diagnosis is important, and genetic counseling also plays important role, the mode of inheritance and the problems carriers, mothers, all those gives important clue for the diagnosis. Similarly, one more child with characteristic facial appearance of Fragile X syndrome. And some of the other commonly seen condition in pediatric neurology and developmental neurology is this characteristic children initially with a lot of feeding from the infancy, early part of the infancy, later development of obesity, short stature and intellectual disability, sometimes artistic feature with characteristic faces like almond eyes, that indicates clinical diagnosis Prader-Willi syndrome. So you can ask specific test instead of going for exome sequencing. In the later age, will develop severe obesity. So this characteristic clinical phenotyping is very important to select the type of the test. Again, here characteristic elfin faces. It indicates Williams syndrome, so you can ask for fish, or you can go for micro instead of ask for whole exome sequencing. Similarly, here a child is born with a large baby with a large head with features of Soto syndrome. So again, it tells that genetic testing, you can select the test based on the clinical phenotyping. So this is a flow chart. So when you are suspecting a children with global development delay or intellectual disability, so if you are sure that other causes are ruled out, acquired causes, that is still common in Indian setup like perimeter instant cerebral palsy. Approach if you are suspecting genetic test, and as I told the previous case scenarios, if you are suspecting specific etiology, you can go for single gene or targeted gene panel. Or if it is not able to identify specific cause, then unexplained developmental intellectual disability, you can go for directly exome sequencing with copy number various detection. If it is normal, may require reanalysis, or sometimes require karyotyping for translocation or triple repeats also. If exome sequencing showing VS are equal to, then you need to go for reanalysis or again karyotyping and segregation analysis. If abnormal, you can confirm segregation analysis. This is simple flow chart that where you can go for detailed confirmation of our diagnosis of development delay with available clinical features and also genetic tests available. Coming to next case scenario, commonly seen in pediatric neurology, metabolic disorders. So various metabolic disorders, even though the single disease may be very rare, but if you combine everything, the prevalence may not be very slow, but commonly seen up to 1 in 1,000 children. So even though it is labeled as a rare disorder, but the prevalence is high in the community because may not be recognized and difficulty in availability of diagnosis earlier days. So most of them at least partially treatable, some of them completely treatable. So specific diagnosis is very important in this condition. Before NGS era, I used to face a lot of problems for genetics. This is one of the case examples. This child presented around 1 year of age with refractory seizure development delay and regression, and this is a family pedigree. We can show that 2 children already expired because of similar complaint around 5 years and 4 years, and there is 3 spontaneous abortions, and also this is the unique state came at 1 year of age with again similar complaints of development delay and refractory epilepsy like West syndrome and artistic features, all these features. So they're already tried with many anti-seizure medication, not much improvement with these medications. Most of the available medicine was tried earlier. So examination not shown much clinical finding except small head. And this is different differential diagnosis you have considered based on that you are investigated. All the most of the routine tests, including hemogram, biochemical tests, were normal, including extended metabolic screening also normal. The EEG are showing hypothermia, suggest of West syndrome, and the imaging also showed not specific diagnosis. It showed non-specific cerebral atrophy, and you have gone for enzyme assay because the lysosomal showed disorders can present like this. Again, they're also normal. And you have gone for imaging tests, those are tests available before NGS era. So those tests are also not much informative. So all are negative. So you have thought of glucose deficiency, so where you have done the glucose test where it shows significantly low sugar levels, and reconfirm again once more. So before NGS era, you used to do so many tests, imaging tests, and so much expensive. So to diagnose specific condition. Now, after the advanced genetic test, it is easy to diagnose these rare disorders as treatment is very specific, and so after starting ketogenic diet, child becomes seizure free and child has significant improvement in development. So this case tells that the importance of currently available tests and easy of diagnosis instead of going for so many tests and imaging procedure, and a lot of time is wasted with diagnosis and also getting proper diagnosis and starting the treatment. So this is a flowchart showing that before the genetic era, so you used to go for so many tests. That classical example what I have given. So after the clinical diagnosis, all expensive routine metabolic screening tests and also invasive tests like biopsies. Till that, after that also many times you not get diagnosis. Nowadays, after NGS era, after clinical diagnosis and routine imaging and some routine metabolic tests, if not clear, you can go directly for NGS technique. So where the yield is very high, then if it is not conclusive, then we can go for imaging test. That is the advantage of this testing. As these disorders diagnosis is very important, I already told, because the specific treatments are available in many of the metabolic conditions. So I will not be going into details. So coming to the next case, similarly the metabolic conditions where after the advancement of genetic testing where you are in. So this child again present 4 years child presented developing delay with involuntary movements. They treated treated as an epilepsy with multiple anti-seizure medication, but no much improvement. On examination, child is a small head. So this is a video shows that this child has involuntary movement ataxia or dystonia. These episodes are treated in epilepsy with multiple anti-medicine, but no much improvement. So after the exam, you can see there is more of ataxia and dystonia, not epileptic palm discharges. So you have considered the possibility of cerebral palsy and dystonias and metabolic conditions. So after investigations, the routine investigation like MRI was not conclusive or normal improved routine metabolic scans. Only the TMS shows that phenylalanine was high. So usually normal PK, that PL ketamine, usually will get a phenylalanine level of more than 1000, but here it is only mildly increased. So earlier, before genetic era, used to do terine levels to differentiate what exactly is the PKU or tetrahydrobiopterin defects. So they are not easily available. So you should have been ended with just restricting the diet without going for further specific condition. You should have been treated in all these children with PKU, ketonuria without much improvement. So as we told, there are many conditions, not only PKU, there are many other conditions that can produce increased phenylalanine levels. So these are other conditions. So you have started with the L-DOPA cavular child with significant improvement in the development notice. So you can see within few weeks of starting medicine, child has significant improvement. During the latest follow-up, we can child almost nearly normal. Child has significant improvement in this specific treatment. So this is a chart showing whenever phenylalanine, we should consider very differential diagnosis, and we used to do many tests for proper specific diagnosis. This is a chart showing to differentiate various subclasses of increased phenylalanine. So we used to do metabolic screening tests to find out the subcategories and to start treatment. These tests, the disadvantage not easily available and they're expensive, and sometimes you need to do CSF, the lumbar puncture test. That's the disadvantage. So in this child specifically, we have done these other classical PQ. They clinically you can see hyperpigmented hair, autism, all these features. This is most of times you can diagnose clinically, but whatever I told case scenario is different. He was directly gone for a genetic testing. It confirmed that child is suffering from dihydropyridine defects. So child was significant improvement with these medications. So whenever child comes with involuntary not only it is always seizures, you should consider other involuntary like extrapyramidal symptoms, ataxia also, where clinician plays an important role. So most of these children should have been treated with cerebral palsy earlier days, and they should have a recurrence in the family that should be prevented. And investigations also sometimes causes overlap of different disorders. So where earlier days lot of problem used to face, now availability of this genetic test, you can diagnose specific disorder and you can start specific treatments. So this child is a significant improvement after starting the treatments. As I told, they are not very rare, so you have a series of cases, most of them are specific treatment and some of them are responded very well. Coming to next group of disorders in neurology, you see commonly is in epilepsy. As I told, epilepsy is one of the commonest neurological condition. As you see that almost 65 million people in the world are suffering from this epilepsy in the world. So even though in the developing countries, acquired causes are very common, but you will see commonly genetic etiology, especially in the childhood age group. So there is a lot of advancement in the genetic thing. So when should we consider these genetic tests and what should be considered? So I'll be telling with the case scenarios. Some of the case scenarios. This is a natural history of epilepsy with genetics. So initially discovered gene for Dravet syndrome. And now we have discovered many epileptic encephalopathies. There are more than 100 developmental epileptic encephalopathies with various genes. And there are many disorders with precision medicine also available. That is the importance of testing and counseling in these children.
Dr. Raju, you have three.
So clinical evaluation is important. Clinical profile, including clinical signs, symptoms, should be documented properly to get a proper specific diagnosis and give important clue for lab person to diagnose your subject. Dr. Raju is also important.
Dr.
And you should rule out always acquired causes.
Dr. Raju, you have 3 more minutes, please.
Okay. So this seizures can be sometimes non-epileptic seizures can be mistaken for epileptic condition. So I'll be giving one P degree for genetic counseling. So this is a family showing that same child has both seizures and paroxysmal kinesia and dyskinesia. So both can be mistaken as seizures. And additionally, there is a strong family history of similar company in the family. It is similar history of paroxysmal dyskinesia. So in this family, I wanted to show basically that the 2 types of diseases, the same genotype produces 3 different phenotypes. So it causes a lot of confusion. So you should always be careful in taking history. The same family can have different disorders because of even though it is a similar disorders. So in this, it is a very rare condition. Usually they are present with the heterozygous state autosomal dominant in present in a PKD, but in homozygous state they can present severe phenotype. This both parents are heterozygous state for PR2 mutation, and those 4 parents had PKD, that is paroxysmal kinesia, dyskinesia, and migraine. Both children, because of severe phenotype, they are present with severe developmental delay and seizures and PKD also. So this is one of the chart. You need to remember that even though family degree you are taking, you should ask in detail about various phenotype because the genotype can be same, but based on zygosity says that the variable, the phenotype can vary. So these are videos. Because of lack of time, I'm not going to detail. So again, the same child can have different. This child video showing an epilepsy, this child showing choreoarthrosis, and again showing same child with epilepsy. So same child can have different phenotype. The gene can be same. Other PDB I wanted to show is this is the one. So another common scenario in seen in newborn with the prolonged ventilatory care. This is a family degree. You can see that this child presented with this is a family showing that baby has present newborn period requiring prolonged ventilatory support and prolonged feeding problems requiring gastrostomy and ventilated tracheostomy tube. Also, if we examine the mother, they never complain. You can see classical myotomy dystrophic phase, and the family, if you go details, there is anticipation this history. This is the index case, newborn baby, mother also affected, their sister and brother are also affected, grandmother are also affected. By then, let's see here, they are all walking, but newborn is affected. So this is a classical P-degree suggested participation. So this P-degree phenotype is very important to understand.
Professor Venkatraju, could you please just wind up? Time is just over.
So NGS plays an important role in precision medicine and also diagnosis and genetic counseling, and it avoided the many investigations and like invasive procedures. So this also helps in early diagnosis, early starting the treatment. So it can accurately assess the risk, and also genetic counseling plays an important role in these children, especially in pediatric neurology children. Thank you.
The question will be asked in the line, and kindly stay online. It's my pleasure to introduce the second speaker of the day, Dr. Sonam Shekhar Reddy. He is Professor and Head of Pediatrics, Ramaiah Medical College, Pediatric Pulmonologist and Adolescent Care Specialist. He is Chief Editor, Pulmonologist Scan, Executive Editor, Journal of Pediatric Pulmonology. His career highlights are he's President of IAP State Respiratory Chapter, Treasurer IAP Allergy Chapter, IAP State Allergy Chapter, Joint Secretary IAP Bangalore, Secretary AHA, and he is Secretary of IAP State Allergy Chapter 2021. So I'll invite him to talk on various aspects of genetic counseling and neurodevelopmental disorders.
Good afternoon, or good evening all, and also to thank various Dilman for being chairing the session, and Dr. Usha Dev and all the organizing team for giving me this opportunity. Will be surprising. I am not from the background of neurology. I am from the pulmonology. Maybe in the next forum. But the question is, how come I was introduced? I don't know. But if you take all the topics are big, maybe there is some purpose being me selected because I work for the COMED. I work for the COMED. I do work for a foundation called as Aniketana Foundation and Charitable Society. Past 2 decades we are working for the neurodevelopmental children's, mainly whatever the domiciliary care and other things. And as a teacher, I do give the research and other things to my students. My passion is to work for these children. And with this background, every Wednesday, I have a clinic for this in a temple, Ayyappa Swami temple. We do run this clinic and we take care of comprehensive health care. Maybe it has been shielded. My friend Venkat is one of the best neurologists. He takes care of the neurological part, rest of the nutrition, growth. What are the other issues? We take care of them, infections, everything, we take care of them. And we do for a charitable society. But as a pediatrician, what I should understand from the genetic counseling, because I am not limited myself to pulmonology and seeing the chronic asthma patient. I do see these kind of patients. And what is that we should understand about what is a simple pediatrician we should understand? Let me know, because Venkat has already told that further. Okay, let us understand this aspect with the developmental disorder, we already classified. and importance of genetic counseling as a pediatrician, because he needs to know the genetic counseling part of it. It is not every time that all the scholars are there. Aspect of counseling, how it helps, and future outcome, and let us think of some specific condition. We know it is as per the MBD classification, it's a disorder of typical manifestations. It may start early, late, or any time in between because of the environmental or occupational thing. If you take the overall classification previously, what is the understanding of our pediatrician was any neurodevelopmental delay, it is the cerebral palsy. That's all. That's what we are also, still it is going on, but it is not that, it is more than that, it is the intellectual disorder, sorry. Communication disorder, anytime from the stuttering to social communication, autism, now it is given the importance from the academy. I represent from the Indian Academy of Pediatrics as a president. We did a lot of work, and this year, whole year, we are doing a lot of related activities related to autism. Okay, ADHD, specific learning disorder, motor and tic disorder. These are all we should understand. We should be having a chart of these things as a simple pediatrician to understand, not that only neurologist. Coming to the, let us understand few conditions so that we know how to counseling this patient. It is not limited to oral counseling. See, when it comes to genetic counseling, do some tests, tell them about the prevention, some rehabilitation, some whatever the group forum. That is not the thing. We should know about the condition to address the counseling part of it. Let us see if you take dominant disorder, Huntington's or anything, autism and recessive, let's extinct DND and mitochondrial like MELAS and other various diseases. Genetic counseling as such, why we need this kind of so many tests are there? So many tests. When I was just asking the Prashant from the Medgenome, you just tell us, see, I get the reports from so many our postgraduates or whoever it is, we'll just tell them, just do the genetic test, but we'll be doing for some other things, as madam was telling, this madam, the professor of dermatology, she was telling, for one of the tests, other test was done. It happens with us also. For some tests, they have done something. So it is a big, first of all, we should be oriented ourselves that what test to be done. Like, so many tests are there. Nucleotide, repeated analysis for the X-linked syndrome, maybe kind of other birth defect, we don't know, or unexplained neurodevelopment delay may be there. We should know for that purpose, a genetic counselor or a counselor, the pediatrician with a background of neurology or who works for the neurological patient should know all the things. But it's not that only counseling, he should have the solid background on that. It's not that we should go at the molecular level, we should understand this thing. Counseling, first of all, what is that counseling tell about? It's a concept. We know the patients or relatives are at risk of disorder. We should know what is the probability of transmitting, as well as further management is decided on your genetic counseling. It is not that on that day you talk about empathy, sympathy, all the things. With the further management, it makes a big changes. So what is that finally? The need for genetic counseling is, it's provide the diagnosis. Okay, you are working with a neurologist or you are separately entity you are working, you are confirming with a molecular level and determine the prognosis. That's important. As madam is telling, epidemiology is so much, and there was a treatment option where the patient is recovered. That's why we can prognostication and therapeutic and guide long-term disease management and identify people at risk of developing. It also early identification makes a lot of prevention and helps the couples to decide about the further family planning and positive approach. Finally, it has happened. So further positive approach can be done with need for genetic. That's why we need the genetic as a simple physician. So it's especially when it comes to neurodevelopmental disorder, the decision making is very important because you are not treating the genetic disorder, you are not treating the neurodevelopmental disorder or a regression disorder, you are treating whole human being. So comprehensive healthcare will come with genetic counseling and with a good genetic background. So benefits to the patients and families is very important. They will understand better. Don't think that because the layman will not understand nothing like that, because who are the best understanding patients are from the rural background. They will understand better. Only thing we are not given the time. We should make them understand, talk to them in their local language. They will understand better than what we see. Because in the Saturday evening, we get lot of patients with, unusual poisoning, taking the tablets and taking something. But we don't get from the rural area. The rural area, if at all, we used to get only kerosene poisoning. But in Bangalore, Saturday night, you see fall from first floor, taking thyroxine tablet, antihypertensive diabetic, it is there with the educated class. But as being asthma specialist, the counseling best happen, especially the compromise with the medication, everything, with a really spend, and it definitely reduces the anxiety and assessing the support networks. One mother asked me, you have told me you go to the physiotherapy, she's good, definitely, she's doing a lot of, but I stay in Tumkur. Every day coming all the way from 2 hours, not possible. Then okay, then you tell, you just see in the Tumkur area, where is 100 kilometers from me? She told I can't go. Then she asked me one word, can you tell me at home, without any facilities, how can you improve my babies, what are the stiffening? That was the question, I didn't have any answer. Then I went 2 decades back, somebody, Udani and other things have written, how to work for these cerebral palsy children in the rural area. That I got it, of course. Of later part, we are talking about the high technology, but we should know how to work for these children in the rural with limited resources. So, role of genetic counseling, what is the impact of life? It helps in the, it maximizes the quality of life we are talking about. It maximizes the quality of life when both of a genetic counselor as well as the patient as well as the treating physician understand, and it will be a holistic approach. Then it tells definitely the individual autonomy of the patient himself. One more important, other aspects, it is not the, it's a spectrum of counseling, spectrum of the domains are there when you are understanding genetic counseling. That is important about recurrent risk and it is not only the counseling, you are thinking about the recurrent risk, explanation of gene anywhere, naturalist how this disease prognosis and the overall prognosis, how to limit ourselves in the further. And it also tells when you are doing the counseling, maybe it requires some more tests to come to conclusion or to see other comorbid condition and to other family members to screen them also. Apart from that, prenatal testing, we all know we are right now thorough with prenatal testing, come sometime of adoption, artificial insemination nowadays with a lot of more than the gynecological maternity home, we are getting more of artificial or surrogacy or whatever it is going on. If you take now if I ask my UG student what do you want to become, gynecologist, fine, good. What do you want to become later? Infertility specialist. So, it's a labor and everything has become it's not a natural, some other, and pre-implantation diagnosis is there. Now they do the tests for the what are the embryo and other things. When it comes, they tell so many diagnoses, but things, treatment options also, and whom to refer is also equivalent, and we are all our good understanding with them makes them regular follow-up. The most integral part of genetic counseling is correct diagnosis, which is relatively possible, and better understanding of the diagnosis will provide accurate and appropriate counseling. If whether you do good counseling or royal counseling, don't do wrong counseling, only otherwise don't counsel. That's important. No need to do any counseling. So what happens with the patients? Patient definitely have the grief and sorrow, and sometimes I do the counseling part when we see they are not understanding. They use the word, they are not understanding. They are from village. One more big word is that they are illiterate. That is the best word they use. So better we should come to that one level. When come down and talk to them, they will understand. Definitely everybody will have that because they have to talk about finance, long-term management, everything. One day, one of the child with cerebral palsy at the age of 18 years died. Then we all were thinking that, okay, finally the mother is relieved of her, whatever this thing. But she told, whom I should carry now? Whom I should carry? Tell me. When somebody told, at least now you have done your best part of the story, don't worry, God has taken. But she told, no, I don't want to give up. No way. I will not take this. This is the more burden. I am not holding my baby. So will not take that, okay, fine, so development, regression, nothing will happen. Because every vaccination, when I will tell you, 90%, honestly, by all your blessing, I give free vaccination to these children. But the question, after 9 months, nobody has taken the vaccines. After 9 months, nobody takes the vaccine. Even free vaccines are there from the community. All the good vaccines are given from the government. But they don't give vaccine, but they come to the conclusion that they are not fit for any vaccination. their nutrition is not addressed, their skin is not taken care. Whatever it is, their growth is not been addressed. So these things is important part of any genetic counseling. And parental attitude will be there, what are the triggering fallings will be there. See, but take care of the group help. Makes other patients, definitely successful patients are there, parents are there, take their name and see the successful stories. Maybe group, maybe take one more counselor also, take their help rather than individual attempt, because it's not easy. We can't come with a just a one word and give the diagnosis. That's very important. And understand, appreciate, acknowledge them is very, very important. And how to deliver a genetic diagnosis to the this is important. I already told about the emotions. Give the hope and perspective, and always be scientific and be clear. Be clear and limit the use of difficult. What I'll just tell you, ICU only will be dealing. We can tell the so many things about, suppose a patient comes in a shock, we should know how to tell them. You tell clearly this, overall our experience with this shock is 60% they'll have a lot of issue, but 40% there is a concern, rather than we'll see. I don't think so. So it makes a big barrier. That is the time they'll start throwing and they'll be looking into Google, and all the medical legal issues starts there. I'm sure sir will agree with that. And we should have a balanced information with a good breaker, rather than they should go to not to the Google or such. We should have a balanced information, and written information is always with a good diagrams or whatever it is, rather than talking. Document your counseling, what you have done. Take the signature. I'll tell once again, tell them also, you come tomorrow. I'll give the time. Please speak to them, because for us, maybe it is a 200 cases of cerebral palsy. For them, it is only one case. That we should understand. That's very important. And explore the, whatever the relationship between the genetics and this thing is there. We all understand it's a multifactorial causes. It is not only genetics, neuro, or what are the prenatal, antenatal. So many environmental risk factors are there. Their economical issues are there. Their upbringing also there. Family group is there. Their environment also. Take everything into confidence. Then only try to control the emotions or everything. Don't just give a suggestion. We'll just write some kind of high level proteins. What is the use? Should not take also. We'll say so many proteins have come, they'll tell if DHA goes and stimulate the brain and will become normal. No, please tell them what simple recipes where they'll understand, they'll make the baby growth. If you take any cerebral palsy, their growth is very less than 5th centile. They are malnourished. So these kind of the environment, whatever the explanation will make, and always tell about the prognosis naturalized way in a simple way, and recurrence risk of the disease to be determined, and especially when at the future pregnancy or with other family members or with the SIPs, we should know. Recurrence risk, we should know. Already Dr. Venkataj was telling with the family risk, and we should tell about the first this thing, second circle, as well as the low risk, high risk, and high recurrence rate, and what are the exacerbation of so many disease occurs. Exacerbation, if you take neurodevelopmental delay, they usually come to the respiratory clinic with recurrent aspiration, recurrent pneumonia, gastroesophageal reflux. We should address that first, because they die with prognosis, this high dependency pneumonia or a static pneumonia. That we should address, rather than this thing. How to feed them, how to feed the child also is very important. That makes the difference, especially exacerbation of any skin infection or any respiratory otitis media should be addressed, and always with this thing. First thing, always give a hope with a scientific background rather than a false hope, making them understanding better, and try to, whatever it is, cognitive restriction, whatever the limited, we know depending on the mild, moderate, severity, mild or moderate, catch them, severity, take care of the domiciliary care. See to that they got the hope. We have seen our good hosts make them sit, and we have seen so many children who are treated as severe, though they are making their own work. They are putting their own trust, they are doing some business. All those things we have seen. Only things, long-term assurance and close follow-up and good scientific background is important. Finally, it is a multi-systemic approach or multi-personal approach. That sounds again, it's a stigma is still there. Anybody, if you come to clinic, they'll be very much worried. If they come to my clinic, they'll be sitting in some corner and saying that what somebody will think. They'll not be coming to this thing also. That is there still. But what I do, I just go, because they can't stand for a long time, I just go in front of all, I just bring them to my inside the cabin. Easy. Easy, rather than we can't make them to wait, just bring them, make them up, and just give some talk and just drop them back to the door. That makes they will be very confident, they'll come to you. That's very important, because this stigma is always there. They'll not go to the function, they'll not go to the, but it's a common thing. But making understand, if you make a children's day in your clinic, call them first, make them to cut the ribbon. That makes a big thing. It's small thing, but makes a big difference. And counselor must also assess the need for additional testing sometime, as we mentioned. If it requires, as well in taking the consent of your neurologist or the treating physician, we can definitely prove. Already I told about the recurrence. Pre-counseling is very important because of the referral and medication, family education is important. During counseling session, tell about the test, discuss about the option, risk, emotional, psychological support, informed consent. Always talk about now with the era of lot of medical legal case, take the informed consent, make video or what audio, this thing. Multiple consent is very important, because every patient for come to you as got a litigation, because they'll be very innocent, but surrounding legal advocacy people trigger them, because they'll come antenatally, it is not picked up. It is a problem with your who is a gynecologist or antenatally, postnatally they are not done, they'll come with you. We have seen a lot of, because we go to the KMC for various issues, please take care of that. And post-counseling, result disclosure, care condition and long-term management is important. Why I am telling you, when a patient was taken care by one of our pediatricians, very good, care-free, everything, and documented neurodevelopmental delay in the antenatal, postnatally, hypoxia, hypoglycemia, metabolic disorder, all the IMS screen being done with all the background, at the 9 months baby dead because of some reason, but they put a case on the pediatrician saying that he is not picked up.
So you have 2 minutes more.
Yeah, that's all. Okay, family-centered care is important, and let us understand counseling approaches with a different. Coming to the disease-specific counseling is also important. When it comes to Huntingtoria, which is a Huntington's, it's a dominant. We should know the mutation where it occurs and how the inheritance pattern is important. 50% of children, so you will be rating. So disease-specific counseling is also important. When it comes to Friedrich's, it's also autosomal recessive condition. Here also where the condition can occur when the child inherits one copy of the mutated. So we should know the background, what you are telling to the next child or the sibling, and very important. And in addition, we know mainly boys, so that we can take care of the other issues. So depending on these condition, we can plan this thing also. Further counseling also, rate also we know where it is the X chromosome and single gene. Here also you can plan in such a way that it's a maternally, even in males also, all these conditions you can plan, and the dominant inheritance or whatever it is, plan in so that the other sibling is affected, female is being affected, or the next pregnancy is affected, or the other family members are affected, plan in such a way that you can come to some conclusion. These are the disease specific. What happens, outcomes and future direction of the genetic counseling is improved medical management makes them, and informed family planning is also required when you are planning for the genetic counseling. Its conclusion, all the medicines are there. It's not the medicines or anything, it's a multicentric approach, multiple approaches, and it is not the genetic counseling, maybe in the center, take everybody efforts for the comprehensive growth of the child. Of course, we are dealing with a human being, not a delayed children. Thank you all.
Thank you very much for an excellent talk, sir.
So our next speaker is Dr. Meera Purushottam, who is affiliated to, who is a senior scientific officer at the Molecular Genetics Lab, Department of Psychiatry, NIMHANS, Bangalore. Her academic acclaims include, she has published more than 100 articles in neuropsychiatric illness, on genetic epidemiology of movement disorders, ApoE4 and Alzheimer's disease, epigenetics of exposure and behavior. Her honors include Fulbright, Nehru Academic and Professional Excellence Fellowship at Johns Hopkins. And she will be talking about the genetics testing and counseling in Huntington's disease. Dr. Meera, the stage is yours. I have to just tell you that you have 20 minutes of time and your time starts now.
Right. Thank you very much. Thanks to Gayatri for inviting me for this program. I also thank all the organizers for this invitation. I must thank all of you who have survived and are bravely facing the Sunday evening. I hope I'm able to give you some insights which will help you. I must confess that I'm not a trained genetic counselor, but I've been trying to do genetics at NIMHANS, and I've been seeing patients and doing work with them. So I hope I'll be able to provide some insights which you may find useful. So there have been a lot of, I can't see it from here. Okay. So I think many of the speakers have mentioned about this, but I thought I'd just put it here, that there are a lot of repeat expansion diseases, and many of them share many common properties. Like, you must remember that all of us have these repeats, but when they expand beyond a point is when it becomes pathogenic. And that's because it's the inherent property of the DNA, and it often changes when it's transmitted from one generation to the other. And longer repeats tend to do more harm. They start an earlier age of onset and a more severe disease. So that's what's called as clinical anticipation. So I think somebody else showed this slide, but I thought I'll just put it here to show you that if you look at this is the gene, there are repeats which occur in different parts of the genes, and sometimes the repeat expansions are very high. When it's the coding region, usually the expansions are not that high. I'll basically be talking about Huntington's disease, which is in the coding region of the gene. So a lot of mention has been made about technology. So I just wanted to put this slide here to show you how, if you look at the timeline, just from 1990 to 2020, technology has gone up, and with that, there have been more and more discoveries of genes that cause different kinds of disorders. The most recent one, for example, is the SCA27B, which was just discovered last year. So some years ago, about I think 15 years ago, we decided to start the GCAT clinic on the behest of the neurologists at NIMHANS, that we have a lot of conditions that we know are genetic, and we would like you to start testing. So we conduct this clinic where we do testing and counseling for patients. And these are all the bright young girls who have been helping to do the hard grind of actually doing the experiments. So this is just one diagram just to summarize what we have got so far. So this green region is, so we don't do exome sequencing in-house. So these are only the repeat expansion disorders that we test for in the lab. And we have found so many positive cases so far. And there's still more than 1,000 cases that are undiagnosed. So last year we realized that we went back and genotyped these and found that some of them were positive for 27B. So there's still a lot to be understood, even in the repeat expansion space. So looking at the HD, we have tested some 600 plus cases, and we got almost 400 positive. So I'll now shift gears and talk about Huntington's disease. So what is Huntington's disease? I think I'm sure as part of your coursework, you would have learned what is HD, because it's the most classic genetic disease that one learns about in your course. So basically it's autosomal dominant and it's progressive. Okay. I think I need help with slide movement. Yeah. So basically there are three parts to HD: the behavioral symptoms, the motor symptoms, and the cognitive symptoms. So the motor symptoms are the most classic. So if a patient comes in with the motor symptoms, then the clinician usually is immediately able to say that this is Huntington's chorea, and they will prescribe a HD test. But if you were to start with the behavioral or the cognitive,
when the patient may sometimes be treated for OCD or schizophrenia or something else because you really cannot tell the difference. So this is what happens to the brain. You have enlarged ventricles and there is atrophy of the cerebral nerve tissue and the basic ganglia is affected. So just to tell you a little bit about the gene itself. So this is your Huntington gene, and in the exon 1 you have a CAG insertion. So the CAG, because it's in the exonic region, it codes for glutamine. So all of us have a glutamine stretch in our Huntington gene, but when it becomes higher and expands beyond the point, we have a disease situation. So in our patients, we found a mean CAG of about 45, and the age of onset is usually, in the mid-adult life. So the HD gene is a very big gene with 67 exons, and it's on chromosome 4. So as you would imagine, if you have a gene which has a very high number of glutamines, your protein is going to get affected, and the neurons will not be looking normal, and they will have certain pathology. So I've just put some of the demographics of the things that we've been doing. So I'd like to just look at some of these things. The age of onset, like I mentioned, is in mid-adulthood, and the duration of illness can vary. So this is I'll be showing you another. So some of the remarkable findings that are found is that sometimes you have reduced penetrance alleles. You have juvenile HD causing alleles, we found 14 of them. And sometimes on rare occasions, you can have a contraction even from one generation to the other. And the largest difference in siblings we found was 16 repeats. So one sibling have 32 and another sibling has 48, and they're both inherited from the same affected parent. So there are many things that are not predictable about Huntington's disease. The largest difference from parent to child that we found was 71 repeats. So it's a known fact that when you inherit from the father, there's a higher chance of an allele jump. So juvenile Huntington's disease is when you have very early age of onset. So it is, this is the adult one, and this is the juvenile one. So we have seen children. We have seen a 4-year-old child having HD. So less than 20 is considered a juvenile onset. And the upper allele in a juvenile onset is the highest we have seen is 113 CAG repeats. So how do we do the testing? As you would imagine, because it's a CAG expansion, we do a PCR using primers that flank the repeat region. And this is a typical agarose gel. You can see an upper band which corresponds to the expanded allele. And when you run this on a sequencer, you will see a stutter like this, which corresponds to the expanded allele. So this just shows you a graph which is plotted of the upper allele and the lower allele for a large number of patients. So the lower, this x-axis is the CAG allele length. And you can see that in the control of the lower allele chromosome, the 17 or the 18 are the most common alleles. Whereas if you see that this corresponds to the expanded pathogenic CAG repeat length, and what you see in a very low frequency here, these are all the juvenile HD cases. So we've been doing some science and some research around Huntington's disease. So some years ago, we tested HD for telomere length. So in case you don't know, so telomere is the average repeat sequences that we find at the end of every chromosome. And with age, telomere length generally decreases. So you find that in people with dementia or with Huntington's disease, the length of this telomere gets shorter. I'm telling you these things because they all sort of go towards understanding the biology of HD, and we hope that someday it will help in treatment. So this is a world match which shows you the prevalence of HD. For some strange reason which nobody really understands, HD is more prevalent in Europe,
and in North America. In Venezuela, because people have moved in from Spain to Venezuela, and that's how the gene has traveled. If you see India and the rest of the world, it is in pink. So this graph was made many years ago based on HD patients in the UK. But what the prevalence in India is still not known. It's still up for, to be done. And it is much lower in East Asia. So what decides this prevalence? The belief is that it is because of a certain haplotype, and whether it is, what is the CAG tract size in the normal population. So this is a paper that we published some years ago, which showed that our haplotype in India is similar to the European haplogroup A. So which indirectly is suggesting that HD is probably going to be quite common in India too. We went ahead and did a more detailed haplotype mapping more recently, and you can see that all these red circles, so all these people have a certain haplotype, and China and Japan have a very different haplotype. And in India, we have a combination of both. We have some patients which resemble this and some patients which resemble this, which is again quite expected. Looking at the geography of India, you would have expected some mixing to happen. So if you look at the inheritance pattern of the cases, we found that a large number of them are paternal, some are maternal, and there is a difference in the paternal and maternal, like I told you. There is usually a higher jump when it comes from the father, and so corresponding the age of onset is also earlier. So strangely, when you look at the CAG repeat expansion across generations, it varies a lot. So this is very different parent-offspring pairs that are plotted here. And you can see that sometimes there is no difference at all. Sometimes there is a slight increase, and sometimes there's a very high difference between the parent and offspring. So this is supposed to be because of the inherent nature of the DNA. So at the time of DNA replication, it is hypothesized that some slippage occurs, and therefore you can have this kind of differences. This is another very classical thing which I am sure you have read in your textbook, that in repeat expansion disorders, there is a direct relationship between the age at onset and the repeat length. This is true of many of the repeat expansion disorders. That means that if your CAG repeat length increases, your age of onset is earlier and earlier. But if you look at this graph a little more closely, you see that there are a lot of points which go on either side of this. It's not that everybody is right on that line. So if you take 40, for example, you have this person who is well above 60 when he started developing symptoms, whereas you had somebody at 30 who also started developing symptoms. So I'm pointing this out to you because I'm sure as counselors you may sometimes be expected to make a prediction on when the disease is going to start or what is going to be the prognosis of a particular patient based on repeat length. So these kind of predictions are a little difficult to make because it is not 1 + 2 = 3. There's a lot going on. So in fact, there's a considerable research that has happened around this, and people have done a GWAS. So people will look at what is the deviation from the mean expected, and they'll use that as a trait to do a GWAS, and what genes they find? They find genes in the DNA maintenance pathway. So like I mentioned to you, during DNA replication, there is a chance for errors to happen. So if you have a DNA maintenance mechanism which is on its toes, it is going to prevent those errors from happening. So that is one thing. And it is now gaining a lot of importance because you probably cannot cure HD, but maybe you can delay it. So if you can, hyper-express these guys, you can probably put the disease at bay for a little longer time. So it's now got a lot of focus. So many years ago, when we were still, trying to get the protocols together for doing testing, we thought we should first find out, do people really want this? Because when HD testing first was, people realized first that HD is caused because of something happening on chromosome 4, And then 10 years later, it was discovered what is the gene and what is going on. But it's not just that the moment the test became available, people didn't queue up to get tested. Because you don't want to be labeled that you have a genetic disorder, especially a genetic disorder that doesn't have a cure, and which has psychiatric symptoms, which are going to pass on to your kids, which will not be covered by insurance. So, do you really want to get tested? So when we did this, we got some interesting feedback about family members not wanting to tell other members. So it's very often even spouses may not tell their husbands exactly what is going on, which is quite common in mental illness. So these are things that one has to bear in mind when you're treating adult onset disorders, especially that have neurological and psychiatric symptoms. So we did another study after doing testing. We said, okay, let's call these patients and find out how they're doing. So we were able to contact a reasonable number, about 81 of them, and we were trying to see where are these people, how are they, because in the Indian setup, most of the time, it is the family that's going to take care of the patient. So how are they doing? So we found that the this was the age of death, this was the duration of illness, and the causes of death, like the doctor mentioned just now, are things like falls, aspiration, and things of that sort. So it's very important to tell these patient families that, look, nobody dies of Huntington's disease. One dies because there is severe weight loss and there is aspiration, there are fractures often, and if you take a patient of HD to a hospital, most of the time the doctors don't know what is HD. And they have no idea what to do with this patient who cannot hold still. How do you take an X-ray, or how do you take someone to a dentist if he has a problem because his head is constantly shaking? So these families have a big problem trying to look after their patients. So these are some of the articles that we have published, some lab-based and some clinic-based, but that's not important. I just wanted to put this classic pedigree. So here you can see that there is anticipation. So you have a 65-year-old who has many children who are affected. And then you have this gentleman who has a son. So we got this patient, and the father was still not showing symptoms. What also happens is that the juvenile HD presentation is very different from the classic adult onset presentation. So this grandmother, this gentleman, will not recognize that this is Huntington's, though he has seen his wife, because the presentation is very different. So when we really took the first full family history, realized that this could be the case, and when we tested, unfortunately, it turned out to be true. So there are many things which are atypical about the family history. So for example, first of all, the symptoms are not yet seen in the parent, what I just mentioned. Sometimes an asymptomatic parent may have died in an accident or a cardiac arrest or something like that. So you will never know that, that there was some family history. So you will just assume that there is no family history of this kind of disorder. And sometimes, like I said, the juvenile case presents very differently, so you'll never think it's the same disorder. And there are also sporadic cases that are encountered, which is something that one should remember.
Dr. Meera, sorry to interrupt.
I'm almost done. 2 minutes?
2 more minutes.
Yes, yeah. So I mentioned sporadic, and so I just want to put this slide up. So we said, let's just genotype the general Amjanta, the BC intermediates. So we found that 5% of the population, we did 200 controls and 200 of our mental cases DNA that we had. We found that 5% of them have intermediate repeats. So these 5% are going to, maybe in the next generation after that, you have a HD family. So this morning also I was attending that session, where there is this emphasis. A lot of HD families say, okay, we want to get rid of this disorder. So what I want to say is that even starting today, if everybody does family planning, every HD family, we're not going to eradicate it because the genome is inherently unstable. Imagine if all of us are identical, we will all be looking like robots sitting in this room, sharing the same DNA. But, but we are different, and that's our reason. That's what makes us special. So we should be proud of it and not try to, I'm almost done. So this is an exercise that happens every year to raise awareness for Huntington's disease. So monuments all over the world, and we've also done this. I'm sure those of you from Mumbai will recognize some of the iconic landmarks. And this is our lab here in Manhattan. So this is my acknowledgement slide. And this is the society that we helped form, because we got so involved with the HD families that they came to us and said, you must help us form the society. So we formed the Huntington's Disease Society for India. And I leave you with this. I just have one or two sentences, small things to say, which, so one is that I've been doing counseling, because after I do the testing, I have to give the report. So I run them through, okay, this chromosome has come from your father, this from your mother, and trying to explain autosomal dominant inheritance. So, in Hindi or in Kannada, when you're explaining, you try to say it is blood-borne. It runs खून में चलता है. So one of them just turned out and said, oh, so if we do a blood transfusion, is it going to go in? So you never think of how the listener is going to interpret what you say. So you have to be a little aware. And there have been many instances when I think at least 3 or 4 where people have gone for testing after a pregnancy, and they thought they would abort if it was positive, but when it was positive, they decided not to abort, because they said, the lady says, I've lived fine for 30 years. What right do I have to deny this child alive? Who knows, maybe you people will come up with some cure or medicine by that time. So it is each family's and each individual's decision. We are seeing women who have taken care of their father-in-laws, their husbands, and their children. So they don't need any lesson on autosomal dominant inheritance. They know that this is genetic, and that's what's going on. So they come to you for some kind of support so you need to help them about how they can build their lives. What is the kind of food you can, give your patient so that you can swallow and not aspirate, how to prevent falls, wear a helmet so you don't hit your head because you're constantly, hitting your head against the bed or God knows what. So it is hemorrhage, aspiration, fracture. Those are the things that cause disease. So often it's also important, you may not be a nutritionist, but if you give them that little bit of information as part of their counseling, it may at least trigger that thought that, okay, maybe I should go and check a nutritionist. You give them, we don't give milk. A very simple thing which can be done at home, which may alter somebody's life. So you have to treat the individual, not the disease. That's what I like to say. Thank you.
Thank you so much, ma'am, for a very illuminating talk. Okay, our next speaker is again in an online mode, and she's Dr. Anusha Panigrahi. Is she around? Yeah. So Dr. Anusha Panigrahi is a professor of Genetic Metabolic Unit, Department of Pediatrics, PGI Chandigarh. Her academics are MD Pediatrics, DM in Medical Genetics, Diploma MD Pediatrics. Her career highlights are Handbook on Medical Genetics and Genetic Counseling, and Handbook of Down Syndrome, Both Defects and Disability, a Beginner's Guide. So, Dr. Anusha, your talk is on genetic counseling for neurodevelopmental disorders, and you have 20 minutes for this talk. Thank you.
Now you can hear me? Hello, you can hear me now? Okay. So there are several types of neurodevelopmental disorders. We usually see global developmental delay, autism spectrum disorder, sometimes attention deficit hyperactivity disorder, and many times we face unexplained intellectual disability. And there may be also sometimes microcephaly with or without seizures and dysmorphism, which are seen in our genetic clinic. And we also see neurodevelopmental disorders with malformations like involving the heart or the kidneys, along with the CNS involvement. So depending on the type of disorder we are suspecting, we usually advise the test. So some of the commoner genetic disorders causing intellectual disability are chromosomal disorders, and Down syndrome we all are familiar with. Down syndrome then we have microdeletion syndromes like Williams syndrome. Then we can have Klinefelter syndrome, in which you can get also some dysmorphism and pubertal problems and infertility. And we have single gene disorders like Fragile X syndrome, Rett syndrome, tuberous sclerosis, several epileptic syndromes, some of which were covered by Dr. Raju. And there are other disorders, rare disorders also, in which you can get to some extent of mild to moderate intellectual disability. So in the contiguous gene syndromes, you have Williams syndrome, Sartre-Chopin syndrome. In Sartre-Chopin, you can have mutation in the twist1 gene mostly. And then there are also metabolic disorders, of which homocystinuria and rarely carnitine disorders, pyruvate dehydrogenase deficiency, and sulfite oxidase deficiency are seen. So we also see lysosomal storage disorders or these large molecule disorders, which include mostly mucopolysaccharidosis and also sometimes leukodystrophies. So genetic counseling is important in these disorders, and some of it, this counseling was covered by my previous speaker. And pre-test and post-test counseling are important. Psychosocial and ethical implications are to be considered. In the less educated or less receptive couples, sequential information can be provided. Multiple sittings can be there, and then you can give them some facts every time. And if it is a joint family, you need to counsel keeping those persons who are involved in decision making in front. And then decision for testing and subsequent prenatal diagnosis should be taken by the informed couple. In case of next generation sequencing, you may get sometimes unexpected results, which was not actually asked for. And when you give these results, it may create further undue anxiety, because the couple or the parents are already stressed due to the disorder that the child has. Family pressures can be there sometimes, and financial issues should also be considered when we go for some expensive testing. Here, like microarray costs around 10,000 and NGS costs around 13 to 15,000. And then if some variant is identified, we do a Sanger. So these pictures, many of you will be familiar with Down syndrome, Williams syndrome. So Down syndrome is easy to diagnose with mongoloid eyes, slant, flat faces, and low set ears. And in Williams syndrome, you have antiverted ears, and there may be lack of direct eye contact. But many of these are over-friendly, the Williams syndrome patients. So the risk factors for Down syndrome, advanced maternal age is a major risk factor, and the risk rises exponentially after the age of 35 years. And if there is a previous child with Down syndrome or other chromosomal abnormality, then again the risk is increased. If there is a parental valence translocation involving the heterocentric chromosomes, then also you can have a risk of Down syndrome. Then parent may be having some chromosomal disorder, or if say that mother also has a similar disorder, then it is likely the child will also have. So the risk of having a child with Down syndrome it is 1 in 1300 for 25-year-old woman. At 35, it increases to 1 in 365. And 45, at 45, we have a risk of 1 in 30. And there is some correlation with abnormal folate metabolism or folate deficiency, but more research is needed in this direction. So the recurrence risk usually depends on what type of Down syndrome the child has, whether it is a free trisomy 21 or a translocation 21. If it is a free trisomy 21, the recurrence risk is usually 1% above the age risk. And if it is a translocation 21, then you have to do karyotype of the parents to find whether it is an inherited translocation or a de novo translocation, and accordingly the risk will be different, because when the carrier mother is there, you have recurrence risk of 15%, carrier father is there, the recurrence risk of 5% is there. But if it is a 21-21 translocation and it is inherited, then the recurrence can be 100%. So, however, many of these already Down syndrome patients are lost during the gestation, and so the actual risk in the live born is much lesser. So, we have also other patients like Fragile X syndrome, which is a common cause of male mental retardation, and we have see other also rare disorders like Pura syndrome. I will come to this one by one. And so, clinical features of Fragile X syndrome: characteristic facial features like long face, prominent jaw, large ears. But typical faces, the facial features may not be there in every patient, and the suggested behavioral features should also be looked for in a prepubertal child, like poor eye contact, hand flapping, hand biting, and anxiety. And there should be a high index of suspicion in those males with a preserved head size. So you won't get microcephaly in a child with Fragile X. So one should see what are the features in the child and accordingly ask for investigation. So Fragile X syndrome is said to be X-linked. Males and females though can be affected. Most of the patients we see are males. Females are usually less severely affected than the males, and 1/3 of the carrier females have been found to be affected. So the testing includes TP-PCR for the repeat size, and on TP-PCR you can also test the carrier mother, and you can identify whether what is the repeat size in the mother. So accordingly, counseling can be done. So if there is like recurrent otitis, media, seizures, mitral valve prolapse, these are also some of the features which can be seen in Fragile X syndrome and should be kept in mind. And in males with Fragile X pre-mutations, there is the risk of FX tax or Fragile X tremor ataxia syndrome. So in females with pre-mutation, 20% will have premature menopause or premature ovarian failure, and risk is greater for pre-mutations, 80 to 100 CGG repeat range. So here we have CGG repeats. In some other triplet repeat disorders, you have different repeats. So the number of repeats also varies depending on the causing phenotype varies depending on the disorder. So the steps in genetic counseling for Fragile X: one should first establish a diagnosis by TP-PCR, and then the repeat size count can be tested in the mother, and you can see the chances of pre-mutations and full mutation. It is given on the basis of repeat size. So greater than 200 is the full mutation. So if it is greater than 200, then it will be a full mutation. And you have pre-mutation 60 to 80, 80 to 100, and 100 to 200. So depending on that, the increase in repeat size will occur. So accordingly, the counseling can be done. So screening in siblings and female relatives of the mother is also recommended. So extended family screening can be done if possible. So different phenotypes you have and different symptoms and therapies are available for X disorders. And when it is a Fragile X syndrome, the child can have attention deficit hyperactivity disorder, anxiety, aggressive outbursts, seizures, cognitive deficits, and the therapy includes atypical antipsychotics, carbamazepine, valproate, and several other therapies like occupational therapy and special education, speech therapy. And you can also try the newer therapies like, if available, like mGluR5 antagonists. But there is a restriction in the therapies available, and special neurodevelopmental pediatricians or a pediatric neurologist can also be involved in the therapy. So in premutation, you can have a premature ovarian failure. So in that, reproductive counseling is essential. Hormone replacement therapy may be needed, and cryopreservation of ovarian tissue can be done if early diagnosis is done. In case of especially if the family history is there. Then in Fragile X associated tremor ataxia syndrome, one can have the patient will have intention tremor, Parkinsonism, dementia, anxiety, neuropathic pains, and in those cases you can try different therapies like venlafaxine, gabapentin, beta blockers. So depending on what phenotype is there, one can try different forms of therapies. So we see also Prader-Willi syndrome in which there is obesity also in addition to intellectual disability, and it is a common cause of genetic obesity, and it involves microdeletion syndrome involving the chromosome 15, and it is a prototype of methylation disorders, imprinting disorders, and uniparental disomy. So if it is a maternal UPD, it will cause Prader-Willi syndrome, and paternal UPD leads to Angelman syndrome. And it has been found that in case of IVF pregnancies, babies born out of IVF pregnancy, there is a increased risk of methylation disorders. So methylation sensitive MLP can be done to make a diagnosis, and this can detect the methylation defect and also the deletion. And if you want to see an imprinting defect in case of say Angelman syndrome, then we have to do a separate testing, that is NGS or specific sequencing. So diagnostic criteria, you have major criteria and minor criteria, and depending on what features are present, one can make also a clinical diagnosis of Prader-Willi syndrome. So less than 3 years, 5 points, at least 4 major, more than 3 years, 8 points, at least 5 major should be there to make a diagnosis. And if you have a testing available, it is good to go for the testing and confirm the diagnosis, and so better counseling can be done. Because nowadays FISH and MLPA and microarray, these are available, so many times we are able to make it confirm diagnosis. So different types of features you can take, and accordingly you can identify. So the monitoring in case of Prader-Willi syndrome is like a monitoring for hypogonadism, hypothyroidism, obesity, and other additional deficiencies. One can check do different tests from time to time. And growth hormone is said to decrease the obesity. It is not given to increase the height, but to decrease the obesity in case of Prader-Willi syndrome. And one should obtain a baseline polysomnography and screen for sleep apneas prior to initiation of the growth hormone therapy. So sleep tests are available in different centers, so these can be done. And also when growth hormone therapy is given, one should also do HbA1c in all the patients, because there can be risk of precipitation of diabetes. So then coming to genetic testing for these different types of intellectual disability, we have different types of disorders now available like MLPA, microarray, and next generation sequencing. So basic test nowadays is MLPA, especially for intellectual disability, because you can detect the common deletion duplications related to intellectual disability, microcephaly, and behavioral problems. And this MLPA P245 kit from MRC Holland is available, and this is the child with Williams syndrome. So you can detect the common disorders like Williams syndrome, Wolff-Fershon syndrome, and P36 deletion syndrome, and Smith-Magenis syndrome. So these type of disorders, we have significant behavioral problems in Smith-Magenis syndrome. So MLPA is a good test, especially where the expenses become important, because many of the common disorders can be ruled out. So this is MLPA. You can see picture for 1p36 deletion syndrome, and you can see the region. This is red dots indicate deletion. If it is above the blue line, then it will indicate duplication. Here you can see the drop below this red line, and so these are the genes that is on the top, given on the top. So these genes are deleted, and so this is 1p36 deletion syndrome. So now we have chromosomal microarray, low resolution microarray, and high resolution microarray. Usually in the postnatal cases for a specific diagnosis, we do a high resolution microarray, and for antenatal cases we do a low resolution microarray. And the deletions, duplications across the genome can be detected in this microarray, and you can identify especially deletions more than 100 kb in this test. Actually, it will depend on what filter you are using. One can change the filter, and accordingly the size of deletion duplication obtained can be changed. So and you have high resolution and HD microarray when you want to see smaller deletion or duplication sizes. So so with this also newer regions involving several genes have been identified using this microarray, and it is the indicated or the recommended test for unexplained intellectual disability and autism, and also unexplained dysmorphism. So this is one example. One child with global developmental delay and vigogenitalia facial dysmorphism presented.
Dr. Anisha, you have 2 more minutes, please.
First font, which showed
please come on.
And you can see the size, you can see the number of genes present in that region, and this was interpreted as likely pathogenic. So if already a similar case has been reported earlier, or it is especially a very large deletion, then it is more likely to be pathogenic. So usually a deletion sizes more than 400 kb are expected to be associated with phenotype. So a duplication syndrome, we had a 12-year-old female with macropephyly down-like faces and obesity, but you see here instead of micropephyly there was macropephyly. So in this we got again on the chromosome 12. So you can see the region P13.33 to P11.22, and this is the size of the gain of the duplication, and these are the genes present in the region. And so basically there was a trisomy of this region, and this was reported as pathogen. So duplication 12p was there in this child, and tetrasomy 12p is known to cause Pallister-Killian syndrome. So you can get different types of loss or gain on this microarray. Also, you can get loss of heterozygosity in different regions, and this can give a clue to say that what could be the likely cause if there is a significant consanguinity, depending on the target genes in these regions. You can do further testing if the microarray is negative. And if you have loss or gain, you see if it is a very small size loss, it is likely to be benign, and if it is a larger gain, it is pathogenic. So here you can see this is a smaller gain. Usually gains more than 800 kb are thought to be important, but it will also depend on the genes in the region and the phenotype. So now we are using increasingly next generation sequencing for in neurodevelopmental disorders, and in these cases more likely you will identify de novo mutations on the next generation sequencing. So de novo mutations will be
Dr. Please summarize, your time is already over. Kindly summarize.
But I didn't get much time. I think the previous speakers took more time.
No, ma'am, everybody took 20 minutes. Everybody took 20 minutes, and nobody got
That's fine, that's fine. So these new genes can be identified, newer disorders can be identified, and also different genes related to different phenotypes can be identified. Like in one case, we identified this child with Pura syndrome, and this was the variant which was there in this heterozygous variant in the Pura gene, which causes Pura syndrome with hypotonia and increased creatine kinase, along with intellectual disability. So variants of unknown significance can also be identified. One has to correlate by doing segregation analysis and parental phenotype, and then take a call. So, you can identify different genes. Sometimes you have to reevaluate the patient and see, like you do MRI or X-ray or some other biochemical test to see whether it is matching with the phenotype or not. So, more and more new genes are being identified in intellectual disability and or neurodevelopmental disorder. And you can have also neurodevelopmental disorder with seizures. Some of this was covered by Dr. Raju. And you can see SCN1B, SCN1A, GAB, BRB, this is actually BRB2. So, according to the findings and the mutations in the index case, you go for antenatal testing, screening and diagnosis. So, if there is a previous child with Down syndrome, Klinefelter, fragile legs, balance translocation, and possible neurodevelopmental disorder with malformations, or you have already identified variants, or you have ultrasonic markers suggestive of a chromosomal disorder, one can do a targeted testing and accordingly identify the disorder or the abnormality in the baby, unborn baby. So, preconceptional counseling is much better than the antenatal because you have much time if testing is required in the family or the index case. And one should do in the folate supplementation. Otherwise, you may prevent a disorder, but the patient may have a neurative defect or a cardiac defect on follow. So, ultrasonic findings associated with fetal Down syndrome, most of these are known. So, short film echogenic bowel, pilot test, reflexes cyst. So, in quad screen, you can see what is the risk, and accordingly, one can plan a confirmatory testing by qFPCR or a FISH or a microarray, and then one can give a diagnosis. So, take-home messages are family history can provide important clue regarding the etiological diagnosis. Fragile leg syndrome is common cause of inherited intellectual disability, especially in the males, so should be kept in mind. And chromosomal microarray is a first-line genetic test for unexplained ID, but sometimes one can miss the diagnosis. In such cases, NGS can be helpful. And in children with unexplained intellectual disability, developmental delay, micropathy, or autism spectrum disorder, cheaper test is MLP for common vision applications. And after doing all the tests, it may be that you do not identify any cause, then also you need to counsel the family, and they may have a risk of around 4 to 9% for having another similar child. Thank you.
Thank you, Dr. Anusha. Thank you very much. Please stay on for a panel discussion. Moderators may please take on from here.
I kindly request Dr. Somshekhar and Dr. Meera to come upon on the dais, and I request the online speakers to just stay with us for the question and answer session.
Hello. So my question is to all the panelists. Basically, the issue is that what are the major challenges you face when we are doing genetic counseling in the neurodevelopmental disorders?
As a pediatrician, what I see is, first of all, they are not educated properly from our side, not from their side. They are ready to come, and we should educate in such a way that they'll come to us repeatedly, because it is not one-time show. It requires repeated evaluation from our side, from the physician point of view, as a counselor point of view, to come to some conclusion, so that we can have point care, whatever the evaluation and the counseling can be done. There some link is missed. Even though I'm in a tertiary care center, I don't get the patients like this. Even either to even go to the neurologist, forget about, even we refer to them the neurologist, they don't go. So there is some big gap is there that should be addressed first, so that they'll come and get the maximum benefit from it. I think, ma'am.
So there are a few things. One is often the patient doesn't come to us. It is the caregiver who we are talking to when we are giving the report. So it becomes very difficult sometimes whether you're talking in the interest of the caregiver or the patient, because sometimes it's not the same what you're trying to tell. So most of the time, at least what I do is I try to tell the caregiver how you can improve the quality of life of the patient. That's the route I take most of the time. I don't specifically tell them that your child is going to be at risk, but when you draw the pedigree and you start explaining, it is the logical question that most parents would ask: is my child going to be at risk? And most of the time, because like I explained, it's mid-adulthood, they've already had children. So it's not a question of planning a family at that point. And then they immediately say, I want to test my child. And so if a child is in school or in 10th class or 12th class, we usually say, wait till the child is old enough to understand what's going on. There is no hurry for this. Nothing is going to change. That advice may change a little bit going forward because there are more treatments coming up now which may help somebody who is asymptomatic. So that advice of when to test is something that is an evolving kind of thing. You cannot uniformly say that wait till you're 24. Then there are people who have now reached 24 or 27. They've put it off. They've seen their parents suffer. They are afraid of whether they will be able to take bad news. But then they say, okay, I want to get married. I like this girl. I need to know now. So the mother was telling me, what if he's positive? Then he has to handle two bits of bad news. One that he's positive and the other that he can't marry the girl he loves. So these are realities of adult onset disorders. And then there are young people who say, okay, I'm at risk. I don't want to know whether I'm positive or not. So they went for IVF and they were not able to go through it. It did not work. And the doctor said, no, you have to do one more cycle. By then the woman who was the spouse, she said, I'm fed up, like I'm done with this. Let's just go natural way, we'll see what happens. They conceived and they went for, after 4 or 5 weeks, they started getting afraid that, what if, this is not good. And then they called me and said, what should we do? We said, yeah, you can do a test. So go ahead and get it done. And then I called CCMB and tried to make sure that the test results were available soon. And thankfully it was negative, so they are happy. So these are the real life difficulties faced by families with adult onset disorders. Because like I mentioned earlier, no insurance is available. So you better get insured before you start showing symptoms, because once you are, then you have to declare that you are positive, unlike in the West. So there was this patient who was some kind of a high-tech software engineer in the UK, and his father had HD. He came back, he got diagnosed there. And the day they realized that it is HD, they have told him that, look, this is a terrible disease, bad things can happen. You better get all your estate settled. You don't know when you're going to lose your mind, when you're going to, things like that. Whereas in India, we have a very different approach. You say, okay, you tell people that there is hope in life. There are people who have worked till 60. It's not that the moment I give you the report, your whole life comes crashing down. You can still live a full life. And it depends on a lot of things, that you should make sure you eat well, you exercise, you do meditation, yoga, whatever. You're trying to tell them ways of living a normal life. Whereas in the West, the philosophy is tell them all the bad news so that they know how to deal with it. It's probably because our family systems are a little more supportive, because we don't have a government and an NHS that's going to take care of any patient who has a disability. It's finally the family, whether it's a wife or another big thing that we have faced in HDSI is there are a lot of spouses who feel cheated. Who say, when we got married, they never told me. My father-in-law was not walking well, and they said, koi accident hua tha, something happened, and that's why. And now my wife has, and my 2 kids have. And had I known earlier, we would not have had kids maybe. Had you known earlier, you probably would not have married us. You see, so there is if you are the parent of somebody who has a child who may be at risk, will you test and put a label, or would you say, okay, I'll put it on God, maybe the husband will be good enough and not leave my daughter on the street because I won't be there tomorrow when she starts getting symptoms. So these are very difficult real life situations, and it's very difficult to write a textbook and say, you know, step 1, 2, 3, this is how you counsel, this is what you do, this is what you tell them. In India, most of the time, we don't have the luxury of 3 visits. Somebody has come from West Bengal and has come for the report. Do you have a chance to say, okay, you go back now and come back again? They have to book a train ticket with a patient. It's not going to happen. So you do the best that you can in the time that you have and hope that you've done that.
Great explanation. That's nice.
Dr. Anusha, are you there?
Yes, yes.
Can we have a word from you?
Yes, sometimes in some families it can be very difficult to convince the parents that this is the inherited disorder, or they might directly ask from whom it has come, whether it has come from the father or the mother. So when you say it is from the father, they will not be able to accept it because they don't think that it comes from the father. And then if you tell it is mother, then there can be chances of discrimination against the mother, there can be stigma, and there can be divorce after that. We have seen these families from Haryana having DMD, in which there was so much stigma associated with it, and they did not go to the right doctor, and they were told that all the boys will have the same disease. So we say that 50% risk for males. It is not 100% risk. But if they go to a general physician or something, some medicine specialist, maybe he will not be aware about what is the risk of recurrence. So he may say that all the boys will be affected. So in that case, there are families, 5 to 6 families in the muscular dystrophy meet, Haryana government meet we had discussed. They told in the 5-6 families, both mother and father committed suicide. So you can understand if they don't go to the right doctor, what consequences can be there. So actually all the pediatricians, neurologists and medicine people should be properly trained so that they refer to the right specialist. So if they come to a proper genetic counselor or a geneticist, they will get the right information. Otherwise, they will also be in stress, and their family will also be not in good shape. So, awareness is required among obstetricians, gynecologists, pediatricians, and other physicians, so that they themselves don't do the counseling and just refer to the right person.
Venkat. Raju, are you there?
Yes, I am there.
Yeah. Can we have a word from you on?
Yeah, so as you know that the pediatric neurology, you will see commonly the genetic conditions. So the proper diagnosis is very important. Most of the times, earlier days used to label as a cerebral palsy. That is one mistake. Most of the time, it may not be a cerebral palsy. You need to investigate all causes of developmental delay, not only developmental delay, other neurological condition after ruling out acquired causes. You should try to get a proper specific diagnosis so that it will be very easy to give proper information to the parents, what is the future. Not only if treatment available, that is good, but if treatment are not available, at least you can counsel what is the long-term outcome prognosis, and also the counseling for next pregnancy also very important. So you should have a specific diagnosis that is very useful for proper counseling in the family. Thank you.
So one word, just within our professionals, we should remove the barriers. We should speak to each other. Suppose if I refer to you as a super specialist, you should give me some feedback so that I can have a small same word within ourselves. Absolutely. We speak different word and mix the other. Yes.
So when the salient features are like, the right test, the right diagnosis, the right doctor, the right information going across. So it has to be an integrated approach, otherwise it will not reach to the people whom we want to make a difference for. So any questions from the audiences? The floor has to be more active. You are all saturated, we do understand that.
Still great audience.
It's very seldom we have such bright clinicians here, so we must make best use of it.
In community, because you go to the community, how do you think genetic counselors can be incorporated in a community setting? Do you think some freelance people can join you?
Should.
On
Saturdays, this 13th, I'm going for 30 children are there in Bidadi. I'm going there. So never I thought of genetic counselor.
So now what you could do is approach the BGCI, our board is there, and whoever is in the local area, we'll refer and do it.
13th, I'll take them.
Okay.
Done.
So people in Bangalore. Thank you.
Thank you. Thank you, ma'am. We'll take it.
We are very fortunate to have this whole group here, and we are a R&D company. We are a drug discovery, biomarker discovery company, the Arrest the Genome Project. I'm Dr. Vilu Patel, the founder. And so we are, sequencing the Zoroastrian Parsi genomes for basically where there are a number of neurodegeneration, a lot of, diseases, both rare, yet they live long. So there's the huge longevity. So it's a paradox. But anyway, what will come out of it is a number of novel, single nucleotide polymorphisms or repeats. And we are already seeing it because we've done 350 whole genomes. And we would love to partner with all of you and connect with you all and have you all on to, advise, support, and convert those biomarkers to novel therapies.
Perfect, ma'am.
Both prevention and precision. So I just thought,
sure, ma'am.
Would you all be available, everyone? Yeah, Bangalore. They are in Bangalore, I know. But I think we need to form a team between discovery people, with, people actually in the field, as well as people treating patients and the genetic counselors. There's a whole group to come together. Thank you.
Nice.
Something came up my mind in the sense that you all are part of a referral here in Bangalore, based in Bangalore. So how would you suggest a patient, say, far away from here, say from Kashmir? We live in a landlocked area and we are cut off from the rest of the country. Even Jammu, which is a part of us, for almost 6 months. We don't have a connectivity by road, by air, or, things like that. So how would you think that we can take everybody on board on this, thing to counsel each and every, person who is affected?
Telemedicine, but question is when it comes to the thing. Telemedicine, it's nowadays upcoming and it is already established with its evidence. I think we can take at least some hopes in that.
Medicine is something like, we should know what are the times, how to coordinate, when to talk to you, more oftentimes the patient would like to see the doctor. That is one physical connect is very, very important. So how do you think this gap can be bridged? Can genetic counselors, play a role in this?
Ma'am, I'll tell you one thing. What is that from our center?
Virtual things are a little difficult also. We have been seeing it for the past few days, virtual talks. It was a little difficult to, so in person is always
we have
preferable.
One center in Bangalore nearby Kaivara is a Ashram holistic, this thing. There a lot of, this one center is there. From our center, we started this kind of tele. Initially, there was poor response. Now we have started getting the patients. They are coming and showing. Our physiotherapist, one station there, he demonstrate and one will guide. All those things, the initial, maybe it's a limitation, but something has started. I think we should work it further.
So, great talk from all the speakers, but I have a question for Dr. Meera. You spoke about Huntington disease, and we have LC in, counseling families related to Huntington disease when a child is included. So you have shown one of the cases where there's a phenomenon of anticipation and the onset of the diagnosis from 68 to 60, 42, and then suddenly it came to 4 years. So I really want to know the approach of your counseling methods when you are counseling a family and when you tell them about anticipation. And when you see such cases, should we test children? Should we not? Because there is a whole chaos when Huntington is involved.
We don't suggest to test asymptomatic children. But if a child is showing symptoms, then of course you have to test, because you have to confirm what the diagnosis is and, look after the child in the way possible. What I meant is that if a parent is tested positive, then we don't recommend testing their children who are asymptomatic. So there's a difference between the two situations. Any child who is symptomatic, yes, we go ahead and test that.
Yes, that's there, juvenile. But when we see such cases, there's a light bulb which goes on that whether we should at least inform the patients, or the family that definitely we are going to explain about the anticipation and everything. Yes. But then they would come, and if it happens, then they would come to us. Yes. That you counseled us, you counseled our family, and it was not supposed to happen. That's why we didn't test the children. So how many of such cases you have counseled in your experiences?
No, so that has often happened that they will ask, is there a chance that my child will get? So you explain the inheritance and tell them that, this is the chance that your child can get affected. And you have to just wait for the child to grow up. You cannot, even DMD, I personally don't believe that every sister of a DMD boy should be tested to find out if she's a carrier. Here is a child who's going to school. Why do you want to put an additional label on that child? It doesn't make sense. Though not all urologists agree with me. They feel that, you're going to, a girl is already sort of under pressure, and then on top of it, if later they find out that she has given birth to a DMD boy, that is more of a problem. But when that girl is getting married, it is her decision. I don't think a young girl who's going to school at 9 years should be tested. No, it's very different. It is very difficult. There was a case where a girl had just got married. The father did not think of testing before marriage. The couple are now in Bangalore, staying by themselves. Suddenly the father says, I want to get my daughter tested. I said, now it is a decision between husband and wife. You are out of the picture, right? Either you had done it before the marriage, then it's fine. Now imagine that girl is staying alone with this guy, trying to get to know him, and then suddenly you put another spanner in the works and say, you know what, I am a carrier for DMD. It's not fair. So what I'm saying is that either way it's not. It's not fair, and it's not your fault. I always tell the mothers that, just because it's on the X chromosome, it doesn't mean that you're at fault. You also got it from somebody else. It's not your fault that you're carrying the mutation. Yes, absolutely. In Niman's back in the days, they would first ask the father if there's anybody in the family on your side, just to put people off track. But now, people Google, and I'm sure people are more wiser. Yeah. It's excellent. Yeah. You are confused, sort of. Yeah. And often.
Very important also.
Muddy the waters a little bit. and often in autosomal dominant inheritance, if by chance in a particular family all the men were affected or all the women, they conclude that it is gender linked, even if it is not. So suddenly when their child of the other gender gets it, they say, नहीं but हमारे घर में तो सब males को ही हुआ है, मेरी बेटी को क्यों हो गया? So that also happens. They make their own interpretations of what they have. I think we should stop. We can keep going on. So.
Yeah.
So ma'am actually highlighted the right issues where genetic counselors are needed. Exactly. So here we end our conference and the scientific sessions. So I would like to request Dr. Afroz to felicitate Dr. Somshekhar. And requesting Dr. Bhavar to felicitate Dr. Meera. Thank you. Yes. So I would like to call upon stage Dr. Vilu Patel to felicitate our chairpersons. So, ma'am, please felicitate Dr. Afroz. And requesting Ali ma'am to please come on stage to felicitate Dr. Bhavar. Please stay on stage. We will have a joint picture later. Yes. Yes, ma'am. So we are having our online speakers also. I think we will, with them also we will take one picture.
Ma'am, could you please turn on?
Dr. Vinusha and Dr. Vakund, please switch on your videos so that we can see you and have a photograph together. We'll wait for you to switch on your videos. Not sure if they can hear. Okay. I request our speakers to stay online, everybody to join for the valedictory, and I request Dr. Jyoti to come on guys for the valedictory event.
Hello. Yeah. So good evening, BGCI family and friends. So now we have reached to to our destination, I should say, of this conference. So Dr. Mary Claire King, who discovered the first gene for a hereditary form of breast cancer, BRCA1, has said, genetic counseling exemplifies the blend of compassion and science, ensuring patients to receive accurate genetic information and the support needed to understand and cope with it. Board of Genetic Counseling of India is a platform of experienced and budding genetic counselors of our country to fulfill the unmet need of helping the common person to make an informed decision about choosing the appropriate genetic test and what to do after getting his or her genetic profile. I am honored to get the opportunity to host the valedictory function of 9th International BGCI Conference and invite Dr. Ami Hassan to give the concluding remarks. Ma'am, please.
I promise to be brief. It's always gratifying when a conference goes well, and I feel satisfied now. And on behalf of BGCI, I want to start by thanking Villu, who partnered with us to make this program and offered to host it in Bangalore. The credit goes to the organizing secretary, Zubaida, who with her volunteers from all over India tried to make this happen in Bangalore. So I don't know how she did it, but congratulations. A conference cannot be successful without good speakers and an interested audience. So thank you for being here till the last. I really want to thank the event people because they have been so polite, so patient, really hospitable. I don't want to take your thanks, vote of thanks, Zubaida, I know you'll be doing it, but they've really, I think everybody who stayed here, has come here, has realized that. So I don't know who chose the place, but total credit goes to them. Gayatri and Zubaida? Okay, Gayatri and Zubaida. And also all our sponsors for being with us. And at the end of this, I want to tell the new counselors who are about to get certified, we all want you in our team. We want you all to remember that next year again, it's the first weekend of July when we'll have our 10th BGCI. So mark it, don't tell me my brother's wedding or my engagement. I don't listen. All of you will be here. Okay, and next time you all be here as genetic counselor. Thank you so much.
Thank you, ma'am. Now, I would like to invite Dr. Madhavi to present the key highlights of this 3 days long event.
Thank you. I cannot promise that I'll be short. I'll have to tell a few details. But as we all know, when we were reading, our teachers used to say, you forget 50% within 1 hour, you forget 70% within 24 hours. So this is just a kind of, maybe we'll take it as a revision and just go back to the moments that we were sitting here and some key points that we can take home and then remember, right? So we had workshop 1, which focused on variant analysis, which was on Friday, the day 1 of our session. We had 2 workshops, and there was immense response from the participants. We had eminent variant scientists who talked about the guidelines which are being used for variant analysis and interpretation, the clinical reporting, and added on the methods of clinical decision making in using these guidelines. And we had a parallel workshop 2, which many of you attended, which was on essentials of genetic counseling, which focused on how genetic counseling blends with intricate scientific knowledge and advanced DNA and chromosome testing, the psychological insights, and how genetic counselors bridge the gap between science, patient care, and how this is enhanced by actually empathy, which is distinguishing quality of genetic counselors. And as the demand for proficient genetic counselors is there, you need to have a corresponding need for education, training, and formal recognition. And the highlight of this workshop was, its collaboration with the Healthcare Sector Skill Council of India, where Dr. Megha Agarwal spoke about the professional genetic recognition of genetic counseling as a skill under this. And we also signed an MOU, which happened in the inaugural session with the Skill Council of India. The inaugural session on the second half of the day, where there was a panel discussion of 7 stalwarts from different domains of genetic testing, research, industry, academia, and the discussion took us through different perspectives of genomic testing and diagnosis and the way forward in therapeutics and evolving roles of the skill set that is actually desirable in a genetic counselor. So coming to day 2, where we had session 1, which was on global variomes for healthcare policies. The session covered different topics of genomics like healthcare policies, role of genetic counselors in advancing genomic tests, genotype-phenotype correlation, and especially in cases of there was sick neonates and how healthcare policies can be framed in developing countries in the poor and in the poor countries. An enlightening panel and a very enlightening panel discussion followed, and there was a common vote among the all the group that genetic counselors are indispensable, and it's important in framing healthcare policies and implementing them, because this is a bond. Again, the importance was given regarding the bridge of the counselors between the healthcare providers, that is doctors, and the community. The session 2, which was on personalized medicine, the session compiled talks by experts from different fields, including research industry and academic institutes with genetic counseling units. The expert panel, they had their opinions that personalized medicine approach by unraveling each individual's blueprint at the newborn period itself would be ideal. This was one of the points that came out from that session. And we again had very nice interactive session where including healthy lifestyle of manageable like genetic conditions and nutrition, infertility, lifestyle disorders can all be which are treatable and which are manageable, and all of these things also should be identified early on. The session 3, which was on common disorders, familial and genetic aspects, the take-home, the session had actually on familial hypercholesterolemia, cardiogenomics, familial and genetic aspects of diabetic and peripheral neuropathy, and where we actually saw that familial genetic factors significantly impacted the management of diabetic peripheral neuropathy, familial hypercholesterolemia, and cardiovascular diseases, and emphasize the genetic testing and personalized medicine that can lead to earlier diagnosis, targeted interventions, and better outcomes of the affected individuals. Session 4 was on cancer and somatic germline for treatment and prevention, where the panel emphasized the pivotal role of genomics in cancer treatment and prevention, both in somatic and germline fields. And they explored the advancements in genetic counseling, the potential of liquid biopsies, and the importance of integrating the genomic data into patient care. So collaboration between healthcare professionals will be the key to making personalized and accessible cancer care to everyone was actually highlighted. The day 3, that today we had the first one on session 5, where we had NGS data in the in neurology, where the delicate balance between the speed of delivering NGS results and neurological conditions and comprehensive genetic counseling that is necessary to navigate these complex implications of these findings was discussed, and there was discussions on strategies for effectively integrating counseling and in the NGS process to make sure that there is informed decision making. And there was a speaker, second speaker spoke about genetic counseling in hemato-oncology, highlighting the responsibility of empowering the patients to potentially life-changing information and supporting them through emotional and psychological implications. The ethical considerations in providing equitable access to genetic services in rare neurological disorders was again discussed. And there was a very strong discussion where the families who have access to the support care regardless of geographic and socio-economic barriers should be actually done. And all of these challenges, were addressed, requiring coordinated effort between the government agencies, healthcare providers, NGOs, and communities to improve awareness, affordability, and equality of newborn screening programs was actually highlighted again in this session. In the next session, that was session 6, it featured a captivating panel of speakers who delved into the role of genetic counseling here in prenatal and pre-implantation genetic testing, where we had again engaging discussions, insights into practicing and aspiring genetic counselors, where the speakers spoke about the PGT framework, the diverse perspectives on interplay between PGT, the ART, and the essential role, and speaking about the acts that we have on PCPNDT and surrogacy. Session 7 was on rare disorders, pediatric and adult, which covered again a wide array of topics with genetics and its medical implications, included cardiovascular genetics, focusing on genetic factors that influence heart diseases, personalized treatments, and we had on dermatology genetics, examining the genetic contributions, the diagnostic advancements, and also talking about the treatment for each one of them, and specifically again on mitochondrial disorders, which are again a different kind of genetic mutations with therapeutic advantages. So deep phenotyping, showcasing the role in comprehensive genetic analysis was again discussed. The panel discussion addressed the importance of genetic counselors again in education and decision making. So the session 8, which again all of you are here, we had a very nice session where the speakers highlighted the role and need of genetic counselors in the neurodevelopmental conditions, and the speakers pushed the point that without genetic counselors in the system, it is difficult to address the emotions and their genuine queries. And we had specifically a speaker talking about the haplogroup and talking about Huntington's disease, how there's a lot of repeat disorders, and which is actually the way forward and how to deal with in both adult onset. And in this session, we saw speakers talking about adult onset neurodevelopmental disorders and pediatric onset. So the counseling, actually, all of you, if you could make out that there was a difference in that you counsel for an adult onset condition to an adult, and for a young onset, where Dr. Inushi spoke about interpretation of CMA reports, MLPA, NGS, where the affected individuals, the index is a child, but you're actually counseling the adult or the parents, and how different the counseling session would be. So all in all, as Annie ma'am said, really satisfying 3 days. We again learned a lot, interacted with everyone, and I want to use words of Dr. Somshekhar to say that genetic counselors need to accept, acknowledge, and appreciate the patients. Okay, and again, taking from Dr. Meera, I want to say that for for all of us, it may be we may have done. You may say that I have done 100 or 1,000 or tomorrow 6,000 cases of so many, but for that family, it's just one. So every case is different. So keep that in mind always, as long as you're a genetic counselor. Thank you, ma'am, for this opportunity.
Thank you, Madhavi. You did not take much time. This was, thank you for giving a concise and precise summary of 3 days long journey of thought-provoking talks and discussions. Now I would like to invite Dr. Gayatri to thank all of them who sent the abstracts and the posters to be presented in the conference. Dr. Gayatri will also introduce us with the judges and the selection process for the poster presentation. Please, Gayatri.
Good evening all. We have now reached the valedictory and are about to announce the results of the e-posters submitted to the 9th BGCI conference. I would like to brief about the abstract submission and selection process that we followed. We opened the abstract submission under 3 categories of original research, case reports, and innovative studies. There were 11 sub-branches within these streams. We had 60 abstracts submitted that were screened by the seniors in the abstract committee, Dr. G.K. Madhavilata, Reena Trivedi, and Dr. Deepa Bhatt. About 53 abstracts were shortlisted, and we received 44 e-posters towards the end. Ms. Shruti was in charge of the e-poster submission forms and coordinating as well as intimating to the authors. All the submitted e-posters along with the 3-minute explanatory video were uploaded to the BGC website by Lisha and Shruti, and they will be there on our website forever. You can also go back and look into them. We thank our e-poster judging panel members as well. The e-poster were independently judged by 3 reviewers for their scientific quotient, clarity, and presentation. We had 1 international faculty, Dr. Sucheta Bhatt, a senior medical director of Illumina from California, and 2 national faculties we had, Dr. Dil Afroz, professor and chair at the Advanced Center for Human Genetics, SKIMS, Kashmir, and Dr. Harsh Sheth, assistant professor and head of Advanced Genomic Technologies Division, Bridge, Ahmedabad. Before we move on and announce the 3 best posters, we would like to congratulate all the authors of the submitted e-posters. We request Dr. Villu to come onto the dais and to give away the award to the winners. Based on the marking, the 3rd prize in the e-poster goes to Ms. Nupur Thakur, Genetic Counselor from Newburgh Center for Genomic Medicine. Her topic was a challenging genetic counseling scenario, families opting out of treatment options for a diagnosed genetic condition. Nupur, yeah, she is. Anybody else from Newburgh? The 2nd prize goes to Ms. Nidhi Medhi, a PhD student from Central University of Himachal Pradesh. Her topic was unveiling therapeutic gold, genome mining for L-Donovani drug targets. Though it is for infectious diseases, she has showed how this can be used for rare genetic disorders. Okay, yeah. And the winner with the first prize is Ms. Mahmooda Begum, research scholar from Maulana Azad National Urdu University. Her title was Temporal Dynamics of Soluble HLA-G and its inverse relation with systemic inflammatory index in women with history of recurrent pregnancy loss. Yeah. Yes, yes. Yes, please. Thank you, ma'am.
So after the prizes, now it is the time for attendees to say something. I would like to invite 2 people from the audience to give their input, feedback on the event that they have been enjoying by means of a friendly education. Who would like to say something? 2 people, 2 persons? This is the time, speak up. So, person who has received the award or the prize, would you like to say something? Would you like to say something about the event, the conference? Please, yeah. Please say something. Yeah, please come.
So again, very good evening. Myself, Dr. Sanjay Mishra. I am assistant professor in Department of Anatomy, AIMS, Jammu. Initially, when I was planning to come here, I was like having doubts in my mind. I don't know much about, but I have started reading, and counseling was a part of my clinical practice, because after the MBBS, I was continuously involved in different kinds of this thing. I saw so much of turmoil in my practice also, because we had to break sad news, many patients dying in our hand, giving the CPR and all those. That was a part of our journey. But this genetic was having some kind of a similarity with my subject, anatomy. We teach genetics to the medical students, but we don't practice. In that note, I joined this course to become a counselor, so that we can start seeing more and more patients, and we can sensitize the other medical fraternity, to get integrated approach to the patient. And that's why I keep stressing that consultation is different, counseling is different, and we should have good counselors in our institute, in our departments. And as we came across different kind of neurodevelopment disorders, then there are blood disorders. So counseling is needed, customized counseling needed for each and every kind of disorders. We cannot generalize you have this disease and ask your family to get checked, and that's not the case. So I think I'm very young to, or very small to say anything on the emotional aspect of the families, because very senior people are here. They must have seen so many patients, and also from their life experiences, they can, understand much better what is the situation of the family who is suffering from that particular disorder is going through. So after that, I learned a lot, and I would like to thank our Annie ma'am, and I saw like how she is energetically, she actually addressing to each and every participant. So that is the quality I think she has, and we really, and I'm actually, ma'am, this is to learn from you, because we very shortly I'm also going to conduct a workshop. So I was so like, how I will do, like what will happen if somebody, finds out some loopholes or something. But I'm observing you 3 days here, and then I got some, that patience and that strength that I can do it. For 2 days I can manage it. So that's all. And respected other senior faculty and ma'am, I would like to, on behalf of our participant, I, congratulate all the committee for wonderful workshop, conference, and we will be coming and we will be advocating this conference to other platforms also. So thank you very much.
Thank you. Thank you. Who's the next one? How many are level 1 certified genetic counselors here? Raise the hands. So, who would like to say? Please say something. Come. You also come? Yeah. So you say something. Yeah, I mean, she's also.
Thank you so much, but still people are confused who is Shubham and who is Soham. Yeah, I know, ma'am, you will never. But at first, I'd like to congratulate all the committee members for their enormous effort in last 1 month's journey, and Dr. Zubeda for her guidance and support. So, as I believe this is the 4th BGCI I am attending, and it's my own pleasure to have the entire team, because we only got together once in a year. So it's like a reunion for us. Because, ma'am, I meet my friends once in a year. But, ma'am, the location is pretty tough to come, ma'am. Come to East also, ma'am. So I'd like to congratulate and thanks, because in these last 3 days, we have nearly 35 talks, which endorse us with knowledge and experience as well. And I'd like to a chairperson and co-chairpersons to convey my deep regards for supporting us and cooperating us, and also the team. Thank you so much.
All the dignitaries and all the attendees, on behalf of the participants, I like to congratulate all the organizing persons, and also the effort the organizing persons, and also especially Aliman to provide the latest information to even the students. I mean, all the who are going to attend the interview tomorrow. It's actually. And she actually provided us with all the latest information so that we will be benefiting for the interview. Thank you, ma'am, and thank you all the organizers. Thank you, one and all.
Thank you. Shubham, do you want to say something? Please come. Shubham, where Shubham? Yeah, he's there. Please come say something. I mean, word of appreciation is always welcome. Doesn't matter 2 or 4. Please come.
Okay, brother, okay. So, good evening, everyone. Respected dignitaries, all the participants, aspiring students, all those for tomorrow, all the delegates, and my fellow genetic counselors. So, everyone talked about the conference, what all we did in 3 days. I just want to talk about what all happened for, managing these 3 days, and what all got to, conclude these 3 days. So, no, but to be honest, like, it was the first of experience, like, from all the years we are doing conferences. And I would really request all of you to please give a huge round of applause to, first of all, Dr. Zubeda, and then to Kankana. for really coordinating a lot between everyone. Gayatri ma'am, Aruna ma'am, and to all the volunteers who have made this possible for the entire 3 days running here and there. And as Dr. Yes, every updates, every WhatsApp, managing everything here and there. And yeah, ma'am, like as you said, so we just need to, understand and learn one thing. I don't know, it's not like my right to say this, but there will be a lot of challenges and difficulties that will, come across as genetic counselor that even we are facing. But this is a field that is, really required. There are not a lot of people who can afford. Auto walas also have those diseases that Ambanis and Adanis have. So we are the person who need to take care for them. That's all I have to say. So thank you everyone. Thank you. And sir, we only have one animal, so yeah.
Thank you, Shubham. And I would like to add 2 more names because I think they have already left, Krishnangi and Janani. They also have done a lot of work for workshop and sponsorship. So thanks to them as well. Now I would like to invite Dr. Sayyida Zubeda to give the vote of thanks. She is Sayyida for few of us and she is Zubeda for few of us. Whichever name you give to her, what I want to say, or I can say that that all of us would like to congratulate her and thank her for making the 9th International BGCI Conference a successful and a memorable event.
Thank you, everyone. I'm really happy to see so many participants. I thought only the first 2 rows will be there. But I'm still very happy. It's past 7:15, but bear me for 2 more minutes or 5 more minutes. So also to remind me if I miss some important names. So a very good evening to everyone. I, Dr. Sayyida Zubeda, Organizing Secretary of the 9th Annual International Conference of Board of Genetic Counseling India, extend a heartfelt vote of thanks to all who have contributed to the success of this event. For the numbers, this year's conference has been a remarkable success with over 400 registrations, which includes 279 in-person delegates and 146 online participants. We also had the privilege of hosting 12 international and 27 national speakers. I express my profound gratitude to our esteemed President, Dr. Q. Annie Hassan, and our Chairperson, Dr. Villu Morawala Patel, for their inspiring leadership and unwavering commitment. When ma'am says, I don't know how Zubeda did it, then I would like to quote that statement, Karo bete, hum saath denge. So that support and guidance gave me the strength to come over here, and with all the friends we did it. And special thanks to the organizing committee volunteers, especially Dr. Aruna Priya, Dr. Gayatri Iyer, and Dr. Janani for all their meticulous planning and tireless efforts. These have been very instrumental. I thank our distinguished speakers and panelists, both online and offline, who have enriched our knowledge with their expertise. I deeply appreciate the abstract judges and the scientific committee for ensuring the high quality of abstracts and the research presented. Congratulations to all the winners of the abstract and poster presentations. Your innovative work is truly inspiring. A special big thanks to our volunteers for their dedication behind the scenes, ensuring the smooth running of the conference. Special mention and huge thanks to Kankana, Soham, Krishangi, Sachin, Prema, Abhinika, Shubham, Lisha, Upasna, Vaishnavi, Shruti, Anupama, Priya, Aishwarya, Aditi, Rekha, Meghna, and Sabhya. Of all these persons, there are 2-3 whom I really want to repeat. One is Kankana, handling all the online, offline registrations, missing their emails, again and again sending the emails, all of them. Thanks a lot, Kankana. And Soham for the sponsorships. Soham and Krishangi. Then I thank all the delegates. Your active participation has made this event a dynamic forum of knowledge exchange. And finally, my sincere thanks to our sponsors, Avastajan, our diamond sponsor, Sion Cancer Clinic, Strand Life Sciences, MedGenome Labs, Lalpath Labs, Apollo Diagnostics, Genetico, Unipath Labs, Newberg, Progenics, Redcliffe, Velitix, and other partners for their generous support. I thank Nolin, Pranati, and the entire staff from the CEO Center for their constant coordination throughout the conference. With that, let us carry forward the knowledge and collaborations from this conference to advance the field of genetic counseling and improve healthcare. Thank you all, and we look forward to welcoming you to the next year's BGCI Annual International Conference. I wish safe and happy journey to everyone. Thank you. Yeah, so tomorrow for the interviews, we request all of the candidates to assemble here by 8:30. Dr. Pavani will take care from there.
So let's what? Sure.
Conference in the valedictory, everybody was there. And
yeah.
Last week, 800 delegates was there, valedictory, only we were on stage.
Please don't go.
And true science has been delivered thanks to all the organizers and the delegates. Thank you.